Okay, good morning, everyone. Let's start our session. Laurie Frappier and I will be chairing the session on Epstein EBV, non-coding RNA regulation and other transforming mechanisms. And Laurie will give a topic lecture. She's working at University of Toronto, and she will talk about analysis of changes in the sumo proteome in response to EBV lytic infection, identifies the TRIM 242833 complex as an EBV regulator.
All right, hopefully they're going to start the slides up. Okay, thanks everyone for getting up after all that night of fun dancing and coming to hear my talk. And I'd like to thank the organizers for organizing this meeting in this amazing location and giving me the opportunity to tell you about our work. And I'm going to talk today about a sumo proteomics project that we've been doing with Ron Hayes' lab in Scotland. So just in the way of introduction, sumo ubiquitin-like modifier that's added on to lots of different cellular and viral proteins. And this can be in the form of sumo-1, which is just a single polypeptide that's added on to a protein, or sumo-2,3, which are indistinguishable and form chains on the protein. And sumolation can regulate the protein's activity, stability, and protein interactions. And we know that it's key to controlling pathways that are involved in viral infection and antiviral responses and also oncogenic pathways. So it's important to understand for EBV infection. We also know that regulation of sumolation is important for herpes viruses in general, because some of the herpes viruses actually encode sumo ligases that put sumo onto proteins, and also what we call stubbles, which degrade sumolated proteins. And there's also a variety of reports that high levels of sumolation in the cell is inhibitory for herpes virus lytic infection. So our study with Ron the purpose was to identify changes in sumolation of proteins that occur upon reactivation of EBV from latent to lytic infection. And our rationale, it was that if we could identify these proteins, they may point us to important antiviral responses or modifications of proteins that were really important for efficient lytic infection. And so just to begin with, we want to have a look at the effects of sumolation on EBV lytic infection. There certainly were a number of reports in the literature suggesting that this would be inhibitory for lytic infection, but we wanted to just have a look at our system. So here in the first experiment, we silenced sumo-1 in the gastric carcinoma cells, AGS, EBV, that are latently infected with EBV. So this is just showing silencing of, this is the free sumo that it was silenced. And what we see right away is expression of BZLF1. So this is just spontaneously turning on as soon as you decrease sumolation. And that suggested that some of these cells may be switching over to the lytic cycle. We looked at this further by immunofluorescence microscopy, staining the cells for BZLF1, and consistent with that, we saw that silencing sumo-1 caused a big increase in the number of cells that were spontaneously expressing BZLF1. We also looked at this using a sumolation inhibitor called ML792. And this is an inhibitor of the sumo activating enzyme, which is required to put any form of sumo on a protein. So if we treat the AGS EBV cells with this inhibitor and then do a blot for sumo-1, you can see that we lose all these high molecular weight forms. So those are the sumolated proteins. And instead we accumulate this band here, which is free sumo. So the sumo is accumulating, but it's not getting conjugated onto the protein. And when we do this, we also see an increase in BZLF1 expression, much like we did with silencing sumo-1. We did the same thing in the NPC43 cells, which are the naturally EBV positive cells from George Chow's lab. And again, if you treat with the sumo inhibitor, you see loss of this high molecular weight, sumolated things, and increase of free sumo. And you see a sizable increase in BZLF1 expression. We also did this experiment by inducing these cells into the lytic cycle with sodium butyrate and TPA. And if you do that, we still find that inhibition of sumolation greatly increases the lytic infection according to expression of BZLF1. Okay, so for this project, we needed cells that we could efficiently switch to the lytic infection, and that's a challenge for a lot of EBV positive cell lines. So what we did is we took the AGS EBV cells and we integrated a DOX-inducible BZLF1, which we call AGS EBVZ cells. And so when we add DOX to these cells, they mostly switch over to the lytic cycle. And in these cells, we also had an integrated cassette for either 6-his-sumo-1 or 6-his-sumo-2, and these express low levels of histag sumo constitutively. These sumos were a little bit different because they had a point mutation in them where we converted KGGT to KGGK in the C terminus, and this allows this sequence to be cut by lyse C and release a peptide at the C terminus KGG, which can be recognized by a specific antibody. So not only could we identify the sumolated proteins, but we could purify the actual peptides that were sumo-modified to map the sumolation sites. So this is the workflow. We had the two different cell lines, one with sumo-1, one with sumo-2. We had the 0 time, which is a latent infection, and then we induced them with DOX for either 12 hours or 24 hours. We had triplicate samples of everything. We made whole cell lysates and then purified all the sumolated proteins on nickel resin by virtue of their histag. Then we cut with lyse S lyse C and gluci, and that reveals this glyk peptide, which was then further purified with an antibody and then run tandem mass spectrometry. So in the end, we get all the proteins that are sumolated and identify the sites and where they're sumolated. Okay, so what we ended up with was just over 800 proteins that are detected as sumo-modified. Most of these can be modified either by sumo-1 or sumo-2. Only few were specific for one of them. And many of these had more than one sumo site. So in total, we had over 1,700 sites that we mapped that were sumo-modified. Now, of course, most of these are cellular proteins, but we did identify 7 sumolated EBV proteins. The first 4 here have been reported previously, so that's not a surprise. Then we got 3 more, BALF2, BORF2, and LF2, to our knowledge, had not been previously reported to be sumo-modified, but we find that they are. Okay, so this is a representation. There's a lot of data. This is one way to represent it. So this is the results from all the sumo-1 cells, and these are the sumo-2 cells. And basically, most of this is representing all of the proteins that we detected that were sumo-modified. The spots in gray do not change, and that is most of the sumolated proteins. They actually do not change. The spots in red and yellows do change significantly. And the way this works is this upper quadrant here shows things that increased in sumo modifications in both time points in lytic infection, and this lower quadrant shows things that decreased in sumolation in both time points relative to the latent infection. So I just want to draw your attention to 3 proteins, TRIM 24, 28, and 33, which are related and form a complex. And we saw major changes in all of them. So TRIM 33, for example, we saw increase in sumolation at multiple sites. And this was actually one of the most highly changed proteins in response to EBV infection. We found for TRIM 28 that we saw an increase in one particular sumolation site. And for TRIM 24, it came out as being downregulated in sumolation. Okay, now we don't really know if those changes are due to sumolation or changes in protein abundance unless we look at protein abundance. So we also did this mass spec analysis where we took the same extracts and instead of purifying the sumolated proteins, we just did a TCA precipitation and ran SDS page of all the proteins. Then we do in-gel digestion of particular strips and run tandem mass spectrometry just to identify everything that's there. And this can be done quantitatively, so we can quantify the amount of protein in each sample. And the bottom line was that we identified over 4,000 proteins, and of these, 485 of them were the sumo substrates that we had identified in the last experiment, so we could assess whether they changed in abundance. Almost none of them changed. We only saw a few changes in abundance. The biggest change in abundance, as you might expect, were the EBV lytic proteins, because they go from almost nothing to being highly expressed. And but one protein that was prominently altered was TRIM24, and it was shown to be greatly decreased in abundance after EBV reactivation. So the decrease we saw in simulation was probably just due to loss of the protein in general. Okay, so a bit more about these proteins. TRIM24, 28, and 33 are related proteins. They are ring ubiquitin ligases. They have the ring domain in the N-terminus, and that can ubiquitinate either ubiquitin or, in the case of TRIM28, more SUMO. They all have PhD bromo domains in the C-terminus, which are responsible for chromatin interactions. There's lots of reports about these individual proteins in the literature. They all have some reports of being transcriptional repressors and having some kind of antiviral effects. It was shown recently that TRIM24, 28, and 33 form a ternary complex, and TRIM24 and 33 also form a binary complex. And it's also been shown that TRIM24 is a very labile protein, unless when it's not in complex with these other TRIMs, it is highly ubiquitinated and rapidly degraded. TRIM28, which is also called CAP1, has been published on previously and shown to bind and repress EBV immediate early promoters dependent on its simulation. And you heard the other day from Sumita that it may also have some new role in EBV replication. And so we concentrated actually on the other two proteins, TRIM24 and 33, because to my knowledge, nobody has looked at those in the context of EBV infection. Okay, so first of all, we just want to do some Western blots on these proteins to see, do we really see what our mass spec data was telling us? Okay, so here's the two cell lines we used for the mass spec analysis, and the 0 time is latent infection. You can clearly see TRIM24 there, and then rapidly when we switch to lytic infection, it's just gone. And it starts with a little bit of a smear up, which suggests to us it's probably either being ubiquitinated or simulated or both, and this is triggering degradation. We also looked at TRIM33. So these are the two cell lines on the right here that we use for the mass spec analysis. And you can see as soon as we add DOX and shift to the lytic cycle, we see rapid shift up of TRIM33 with multiple bands. We did the same experiment adding the simulation inhibitor, and you can see that the upper bands all go away. So those are indeed simulated products. And interestingly, there's an initial shift up that is not sensitive to the sumo inhibitor. We don't actually know what that is at this time. We then repeated these studies in the parental cell line, AGS-EBV-Z, because we just wanted to make sure having the extra SUMO in there wasn't messing up our results. And again, you see that rapid shift up of TRIM33, and that's due to simulated products. We also looked at the lytic expression profiles of these cells, just again to make sure that having a little bit of extra SUMO wasn't messing up the lytic cycle progression. But when we looked at various lytic proteins, basically the profiles all look the same. So it seems to be consistent. Okay, so then we asked how does TRIM33 and TRIM24 affect reactivation and lytic infection? So we did this by making CRISPR knockouts, initially in the AGS-EBV cells. So we used two different guides for TRIM33 and TRIM24 to make sure the results were consistent. And here we're just simply running out lysates compared to a control guide that doesn't target anything. And so we're just looking at the level of the various lytic proteins here compared to the knockout. What we see is that as soon as we do the knockout, we see an increase in BZLF1 expression, and then any other lytic proteins that we looked at similarly increased. We did the same thing in Honeacata and NPC43 cells, and all of the results were the same. So if we compare to the control guide, once we do these knockouts, we see a turning on of BZLF1. We reasoned that that was most likely because they were turning on transcription of BZLF1. So we quantified the BZLF1 transcripts with knockout of TRIM33 or TRIM24, and indeed we did see an increase in these transcripts. Okay, so we also did the opposite experiment where we overexpressed TRIM33 and TRIM24 and looked at effect on BZLF1 expression. So these are the AGS-EBV cells where we're overexpressing TRIM33 or TRIM24 relative to a flag tag negative control. And you can see that if we count the number of BZLF positive cells, they do go down dramatically. when we overexpress these proteins, consistent with repression of BZLF1 expression. We looked at the effect on the promoter using a ZP, a luciferase reporter assay, and again, overexpressing TRIM33 suppressed expression of the luciferase, consistent with report effects on that promoter. Okay, the other thing we did was to make a TRIM33 that had mutated sumocytes. So our mass spec data consistently detected 4 positions of sumolation in all of our experiments in TRIM33. And so we mutated each of those sites, and this is just comparing the migration of TRIM33 when you co-express it with HIS-SUMO1. The wild type 1 shifts up highly. The mutant that we made doesn't shift as highly, consistent with disruption of some sumocytes, but we do still see a couple of shifts. So it seems like it's picked some alternative sumocytes, and this is the pull down of the sumolated proteins, and with basically the same result. Okay, so we went on to compare the wild type and the sumo mutant of TRIM in the overexpression assay to look at effect on BZLF1 expression. And we found that the sumo mutant wasn't as effective as the wild type in suppressing BZLF1 expression. So sumolation seems to have something to do with this effect. Okay, the other thing we did is we wanted to know whether this TRIM24-28-33 complex actually formed in the context of EBV lytic infection. So we did this using an affinity purification mass spec experiment where we took flag tag TRIM33, we put it into the AGS EBVZ cells, and then reactivated them to the lytic cycle with DOX. And then we purified out the flag tag TRIM33 under fairly stringent conditions that we know just really reveals specific protein interactions. And then we run mass spec to identify all the interacting proteins. So these are the results from 2 independent experiments, and I'm just showing you the top 5 recovered proteins. And of course, the top one is TRIM33 because that's what was tagged. The next 2 are TRIM28 and TRIM24, clearly showing that this protein does interact in this complex in lytic infection. And interestingly, the next 2 hits were both versions of BZLF1. So there's 2 versions of BZLF1 in these cells because we have the DOX-inducible one, which comes from B958, and then there's the one that's expressed from the virus, which is a CATA. And the sequence is different enough that mass spec identifies them as different proteins. So we thought that's kind of fascinating that BZLF1 might be kind of feeding back and interacting with this complex. So we looked at it again by doing the same experiment, but just performing Western blots for these proteins. So here's the wild type TRIM33 pull down, and you can clearly see TRIM24 and TRIM28, and then there's the BZLF1. And then we did the same thing with the sumo mutant just to determine if sumolation of any of those sites was a requirement for any of these interactions, but it did not seem to be because we still got efficient recovery of these proteins. So we reasoned that if BZLF1 was interacting directly with this complex, we should be able to identify one or more of the TRIM proteins that BZLF1 interacts with. And so we did this by just overexpressing an individual flag tag TRIM protein along with BZLF1, and these were just done in 293 T cells to just produce high levels of the 2 proteins. And then we IP'd with flag and looked at whether we recovered BZLF1. And you can see here that TRIM33 and TRIM24 clearly pulled down BZLF1, and TRIM28 does not. We detect no interaction with it. And this is despite the fact that TRIM28 actually expresses way better than these other TRIMs in these cells. And consistent with the literature, we find if we express TRIM24 on its own, it's really hard to detect because it's so labile. But we do have enough here that if we pull it down, we can still see the BZLF1 interaction. Okay, so then the last thing is we wanted to know why would BZLF1 be interacting with this? Our theory was it may be disrupting the complex to disable it. And so for these experiments, we took AGS cells and made a DOX-inducible BZLF1 in them. So there's no EBV in these cells. We're just turning on BZLF1. So here's the lysate. So you can see with DOX, BZLF1 comes on. And then we immunoprecipitated the endogenous TRIM33 with a TRIM33 antibody. And we looked for recovery of the endogenous TRIM28 and TRIM24. And you can see that we do recover these if we don't have BZLF1 expressed. Once we turn on BZLF1, we lose recovery of these other TRIMs consistent with the disruption of the complex. And then the last thing we looked at is if this complex is disrupted, we should be starting to see loss of TRIM24 if it's that labile. So this is a time course of induction of BZLF1, shown here. And as you can see, we do indeed start to lose TRIM24 consistent with disruption of the complex by BZLF1. Okay, so in conclusion, we have identified changes in simulation of TRIM28 and 33 and loss of TRIM28 that occurs in response to EBV lytic infection. And TRIM24 and 33 have roles in repressing expression of BZLF1 and hence lytic infection. And the suppressive role of TRIM33 seems to be partly dependent on simulation. And we've also shown that BZLF1 interacts with this complex and disrupts it, resulting in a loss of TRIM24. Okay, so just to acknowledge the people who did the work. So most of the experiments I showed you were done by my postdoc, Carlos de la Cruz Herrera, who's shown here, and also by a PhD student, Umama Siddiqui, shown here, and she's actually here in the audience today. And my lab manager, Kathy Shire, made some of the cell lines we used. And as I mentioned, the whole project was done in collaboration with Ron Hayes' lab at the University of Dundee, and Mike Tatham in his lab in particular did all the mass spec work, which was like really a tour de force of unlabeled mass spec. And for our affinity purification mass spec experiments, we worked with Jack Greenblatt and Adita Marcone in his lab. And thanks very much.
So any questions for Lori? Yes, please.
Really nice work, Laurie. So as you mentioned, TRIM28 is sufficient to silence and repress EBV lytic genes. Can you speculate on why you need this ternary complex and also what the possible mechanism of silencing might be?
Yeah, so, in most of these studies, there's lots of studies where they just look at one of these three proteins. They never look at the other ones. So I would say from most of the experiments, you don't know whether they're acting individually or they're acting in a complex, because it's kind of been more recently reported that they're a complex. So it's hard to know how to interpret a lot of the literature. But I think we've clearly shown that the other two TRIMs also have a major effect.
So are you thinking that the repression might be through epigenetic mechanisms?
Sorry?
Do you think the repression might be via epigenetic mechanisms?
It could be. They're all chromatin associated. So yeah, I would say that's probably the simplest interpretation. We don't really know yet. Of course, they all can ubiquitinate or in case of TRIM28, simulate things. So it could be even more indirect.
Thank you.
That was lovely. Do you think it's possible that the level of the complex in different cells will determine the spontaneous lytic reactivation?
Oh, that's a great idea. And we haven't actually done that and compared, but I think we should.
So Lori, very nice talk, Lori. So, I'm not sure if I got that correct. So TRIM28 doesn't interact with BZLF1, but all three of them form a complex. So are you detecting like direct interactions or protein-protein interactions?
So
how
so
does it work
when
exactly?
we pull down BZLF1 out of cells, right, we don't know what's direct and indirect, and we pull down that complex. But
Right.
the way we did that other experiment was to purposely just like swamp out the complex. So we had like a ton of individual subunits, so we could just see which individual subunit it liked to bind to. But of course, in the context of the cell, it would pull down the other ones as well. But we were just trying to get at the mechanism a little more.
Okay. Did you try to put point mutations so you would interrupt like, some of the interactions and see which ones would come down and which ones would not?
Like we don't know where the interacting sites are at this point, like in
Okay.
BZLF1 or that's definitely on our to-do list.
Awesome. Thank you.
Okay, let's go ahead with the next talk. So will be by... Yeah. So will be by Ashley Campbell, and she works, I think with you, Laurie, right? So at the University of Toronto, and she will talk about the BGLF2 interferes with cellular mirror function by binding risk.
Awesome. Hi, can everyone hear me, especially in the back? Yeah, perfect. Okay. Yeah, so just to repeat, my name is Ashley Campbell. I'm a PhD student in Lori's lab, and I'm really interested in how EBV causes RNA dysregulation, or even just how it's affecting the RNA regulation of the cellular system. And so today, I'm going to be telling you about how the EBV lytic protein BGLF2 is it's interfering with the cellular microRNA functions, and it's through its interactions with the RISC complex. So just a little background about BGLF2. So it is a late lytic protein. It can be found in the tegument, and our lab has previously shown through screens that BGLF2 can cause G1S-like cell cycle arrest, as well as increase sumylation of cellular proteins. It was a different assay experiment that was explained by Lori, but it was shown to be there, as well as other groups have worked with BGLF2 and saw that it activates P38, as well as inhibits type 1 interferon signaling and NF-kappa B. So Laura gave a great introduction to sumo, but I kind of want to highlight a few points about sumo and sumylation for the relevance of this talk. So just a reminder, sumylation is a protein modification caused by the small ubiquitin-like modifier called sumo. And as you've seen, sumylation itself can affect cellular and viral protein interactions and functions. On the slide here, I'm just showing you the cyclical pathway in which the sumo protein is able to be conjugated onto a target protein through the E3 ligases. And for the purposes of my talk, I really want to highlight the unconjugated sumo protein. So for us, we can also call it free sumo because it's not on the target. And when you have the sumo conjugated to the target, that's what we see on typically our Western blots as sumylation. So after the initial screen to find BGLF2 inducing sumylation, we wanted to understand what cellular proteins BGLF2 is interacting with. And so for BGLF2, we performed affinity purification coupled to mass spectrometry. And this is a filtered list of some of the top interactors. And we found that BGLF2 seems to be interacting with the microRNA-induced silencing complex. It's also called RISC, so I'll be calling it RISC throughout the rest of this talk. And it's important for microRNA-mediated mRNA regulation. So what that means is that you have the small RNAs called microRNAs that can be incorporated into Argonaute proteins. And this with the help of the scaffolding protein called TNRC6, it can then be recruited to transcripts that have microRNA recognition sites by the partial complementarity between the microRNA to that target transcript. It's typically through the 3' UTR, but there's other examples of binding. And so after we got this data, we wanted to confirm the interaction in multiple cell lines, including EBV-relevant cell lines. So the first one that we did was in 293Ts, where we overexpressed flag-tagged BGLF2, which you can see here, and did the flag IP with or without RNase A to make sure that this interaction isn't through RNA, because these are RNA-binding proteins, Argonaute. So you can see that when BGLF2 is immunoprecipitated, it can co-IP endogenous Argonaute2 and TNR6A, even in the presence of RNase A, supporting that it's not through an RNA interaction. We then want to do it in an EBV-relevant cell line, so we used the gastrocarcinoma cell line AGS, but we modified it so that it has a DOX-inducible flag-tagged BGLF2. So like Lori explained, when you add doxycycline to these cells, you allow for the transcription of the BGLF2 flag transcript. And so you can see that here, when you add dox to the cells, BGLF2 is expressed. And when you pull that down, you can detect, again, endogenous Argonaute2 and TNRC6A. And so we then just wanted to make sure that this interaction can also be seen during EBV lytic infection. So I used the AGS EBV-Z cell lines that was explained previously, as well as the Raji cell line, and showed that during lytic infection, that BGLF2 again can immunoprecipitate the endogenous Argonaute-2 in those cells. We tried also to look at whether the BGLF2 homologs can also interact with the risk complex, and what we found was that the other homologs and other human herpes viruses were unable to immunoprecipitate endogenous Argonaute-2, suggesting that this interaction is not conserved across the homologs, and that BGLF2 might have a unique function compared to its homologs. So after confirming the interactions, we wanted to look at the localization in cells through immunofluorescence microscopy, and risk is known to co-localize into these cytoplasmic bodies, including processing bodies or P bodies and stress granules. They're both important for mRNA regulation, either mRNA decay or mRNA storage. And so in order to look at the potential localization, I use DCP1A as a marker for processing bodies and G3BP1 as a marker for stress granules. So what we found was that when you express BGLF2 in HeLa cell lines, especially, you get these cytoplasmic bodies that you can see. They're brighter compared to the rest of the staining. And they do co-localize with Argonaute. This is endogenous Argonaute-2. And it also co-localizes with G3BP1, suggesting that these bodies have components that are also found in stress granules. When we looked at the P-body marker, DCP1A, we didn't see the co-localization, but moreover, we actually saw a loss of these P-bodies in cells containing BGLF2. And when we looked at this across multiple cell lines, including the gastric carcinoma with inducible BGLF2 and the nasopharyngeal carcinoma cell line HONI1, we saw a consistent decrease in these processing bodies when BGLF2 was expressed. So P bodies are important locations of the risk function. So we wanted to then look at whether BGLF2 is affecting the functions of risk in the microRNA. So one famously studied microRNA is the family of LET7 microRNAs, and we use luciferase reporter assays, the first one being a Ranilla with tandem LET7A microRNA binding site, so that allows the Ranilla transcript to be regulated by LET7A. And as you can see, when BGLF2 is expressed with this Ranilla construct, you get an increase in Ranilla luciferase activity, and that's compared to our empty plasmid control, showing that BGLF2 is interfering with the LET7A repressive activity of this construct. We also looked at a 3' UTR that's known to be regulated by LET7A, that is the 3' UTR of Dicer, and again, it caused an increase in the luciferase activity. What was interesting is that the SUMO transcripts themselves have also previously been shown to have multiple LET7 microRNA binding sites, including LET7A and LET7G. And so when we clone those into luciferase reporters, we again saw that increase, suggesting that BGLF2 is regulating SUMO through these 3' UTRs. So we went back and wanted to understand better how BGLF2 is affecting SUMO and maybe bring it back to that initial screen of the simulation. And so using the BGLF2 inducible cell line, we confirmed that when BGLF2 is expressed, you get an induction of simulation of cellular proteins. It's much more visible with higher molecular weight proteins. And when this lysate was ran on a higher percentage gel, you're able to resolve the free SUMO, so unconjugated SUMO. And again, you see an increase in the free SUMO, suggesting that BGLF2 may be inducing simulation just because there's more free SUMO available. And unlike the proteins, the SUMO transcripts themselves weren't changed, and that really signifies a post-transcriptional regulation such as microRNAs. So we next wanted to see, because BGLF2 BGL2 seems to be interacting with Argonaut, we wanted to see if BGL2 is somehow preventing the incorporation of microRNAs into Argonaut. So to do that, I used the inducible systems again, and this time immunoprecipitated endogenous Argonaut-2 and performed RT-qPCR on multiple microRNAs, including LET7A and LET7G. When BGL2 is expressed, as indicated with the plus dots, you see there's less LET7 microRNAs being incorporated into Argonaut compared to the uninduced controls. And this wasn't due to just the microRNAs being less in the total pool of the cells, because when we looked at the total input sample, there tended to be even actually more microRNAs in the pool. When we did the reciprocal experiment to look at whether or not BGL2 is important for microRNA control during lytic infection, we first generated BGL2 knockouts in the AGS-EBV-Z cells. And so you can see there's not much BGL2 anymore. And when performing the Argonaut-2 IPs again, you can see that when BGL2 is knocked out now, there's an increase in the LET7A and LET7G microRNAs going into Argonaut compared to wild-type lytic infection. So this is really supporting that BGL2 is somehow regulating microRNAs going into Argonaut. So I think I'm almost done. But I really hope you can take home this message of without BGL2, these sumo transcripts and even other transcripts could be being regulated by the Argonauts with microRNAs in them. And when BGL2 is expressed, there's a loss of P bodies in these cells, as well as the microRNAs such as LET7 not being incorporated into Argonaut. This relieves the sumo transcripts to then be translated to produce more free sumo, and this free sumo can be driving the increase in sumolation that we previously saw in our sumolation screen. And so with that, I'd like to thank everyone in the lab, especially Carlos. So he started this project and was really helpful with the sumolation experiments. Again, shout out to Amama and Jaime, who are also here today. The rest of the lab, the Greenbot lab for their APMS, for them handling the APMS data, as well as Shin-Hu Young, who graciously gave us the BGL2 antibody that we used in this study. And so with that, I'm happy to take any questions.
Hi, great talk.
Hi.
So maybe I missed this, or what is the effect on viral microRNAs? Does this have,
Right.
does it decouple that process?
Yeah.
Or?
So we're currently looking into that. For this experiment, I looked at just cellular ones, especially because of the most of the experiments were done in just the BGL2 inducibles. But when I get back, I'm going to be looking at the viral microRNAs. I have the samples. Yeah.
Great, thank you.
Thanks.
There is one question there.
Yeah, very nice. Do you think there is a negative feedback loop so that this increased simulation would then shut down lytic replication?
It could possibly be. I think that is something we can definitely look at, especially because we are looking so much into the simulation, and because BGL2 is like a later gene where it could be doing stuff like that, or the increase in simulation maybe be promoting some form of the later gene expression. Yeah, but that'd be really something that we would like to look at. So thank you.
Okay,
Awesome.
I think that we don't have any questions.
Awesome. Thank you.
So the next talk will be by Jian Liu from University of Hong Kong. And the title of the talk is The Role of Epstein-Barr Virus, Long Non-Coding RNA BARTs, BRD4 axis, in promoting nasopharyngeal carcinoma.
Thank you for the introduction. And EBV is a tumor-related gene and is 100% associated with MPC, but the molecular basis of its role remain elusive. EBV exhibits at least 3 types of latency program with limited viral protein expression. It is notable that BAR RNase is consistently highly expressed in all the latency program. However, little is known about the functions of this BAR RNase. BAR RNA derived from multiple exons in BAR region, and its splicing results in 4 major transcripts. RPMS1 which contains all the BAR exons with a size of 4 kilobases. And I use RPMS1 to describe the primary assignment of link BAR's function in this study. My study will be composed of 2 parts. First, link BAR's functions as a epigenetic regulator modulator to promote MPC progression. Second, link BAR cooperate with BRD4 to regulate the expression of MYC and BCL2 in MPC. Part 1. Previous publications have indicated that BAR is restricted in the nucleus. To define more what's the BAR function in the nucleus, I conduct RNA fish using nuclear spike markers, SC35, and nuclear link markers, pair spike markers, PSPC1 and SFPQ, and I found that link BAR is colocalized with nucleus, which is a highly dynamic subdomain in nucleus, which also work as main regulator of gene expression. So it's assumed that link BAR may involved in the epigenetic regulatory process executed by nuclear spikes. To better characterize the role of link BAR, I generate link BAR knockdown cells by CRISPR into EBV positive cells, CTRL6 and YCCEL1. These 2 cell lines show similar phenomena. The expression of BAR RNAs are significantly decreased, and in addition, the EBV latent genes EBNA1 and LMP1, BZLF1, and EBAR1 are also apparently decreased. To define the influence of link BAR on the cell development, the link BAR knockdown cells were analyzed by flow cytometry, and we can see the link BAR knockdown induce higher apoptosis rate, and it decreased the cell number in G1 phase. To assess the epigenetic function of link BAR, and then I perform RNA-seq. In the heat map, we can see clusters of genes are regulated by link BAR. As representative cancer-related genes, MYC and BCL2 are significantly downregulated by the knockdown of link BAR. And I also verify these 2 results in CTRL6 and YCCEL2 by qPCR and Western blot. So link BAR may regulate some oncogenes like BCL2 and MYC to promote tumorigenesis in NPC. To access the epigenetic function of link BAR, I perform ATAC-seq. This method can map open chromatin, open and accessible chromatin is often related to active gene transcription. In the heat map, we can see the open chromatin become globally inaccessible upon the knockdown of link bars. And the relatively loose of open chromatin region accounted for the majority. And most of this region are located in the promoter. So link bar may regulate gene expression on the promoter region. In addition, a decreased enrichment of signal on EBV latent region of replication is observed in the bar knockdown cells. ORIP region supports the replication and stable maintenance of viral episome in human cells. So link bar not only can regulate the host gene expression, but also play a role in the maintenance of EBV latency. Part 2, link bar cooperate with BRD4 to regulate the expression of MYC and BCL2 in MPC. BRD4 is a epigenetic reader, and it can bind and recognize and binds with acetylated histones. To define the role of BRD4 in MPC cells, I perform IHC in MPC patient samples, and it, as illustrated, BRD4 is dominantly in the cancer cells and very few in the non-cancerous cells. And BRD4 seems to be consistently with the MPC progression. The late stage of MPC samples show more intense BRD4 staining. So BRD4 may play a very important role in the progression of MPC. Then I use RNA fish to find that link bar is colocalized with BRD4, and this interaction can be disrupted by the treatment of GQ1. GQ1 has been reported to as a BRD4 inhibitor, and it can competitively replace BRD4 from the acetylated histones. And RNA immunoprecipitation and RNA pull down further can confirm the interaction between link bar and BRD4. SpyTag-based clip, which is easy to use and highly accurate tool for the systematic study of RNA and protein interaction. And I use SpyClip to define the binding motif of BRD4 and link bars. And this figure shows that the SpyClip defines clusters dominantly localized on the exon region of bars, and the red sticks indicate the location of the motif. Mallet here, which is abundantly expressed non-coding RNA localized to nuclear spikes, is included as a negative control. Then the BRD4 binding motif was analyzed by Homer. To clarify the role, how link bar regulate gene expression, I perform ChIP-qPCR. Both the bar knockdown and treatment of GQ1 can decrease the enrichment of BRD4 on the promoter region of MYC and BCL2. So bar can enhance the enrichment of BRD4 on the promoter region of genes to regulate the expression of MYC and BCL2 and maybe other oncogenes. To summarize, link bar colocalized in the nuclear spikes. Link bars directly associate with BRD4 and GQ1 is a competent inhibitor interrupting this interaction. Link bar can enhance BRD4 enrichment in the promoter region to regulate the expression of MYC and BCL2 in MPC cells. And this is my working model. So link bar can facilitate the maintenance of EBV latency, and on the other hand, can regulate some host genes to promote tumorigenesis. And these results also establish a rationale for the clinical investigation of GQ1 as a drug candidate. And LINK-BAR and BRD4 also can be a potential targets for the treatment of NPC. And thanks my supervisor, Professor Hongling Chen, and my team. Thank you for listening.
Okay, any questions? Okay.
Thank you. When you used CRISPR in the C666.1 and YCCEL1 to knock down the LINK-BART, am I right in thinking that would also have removed the BART microRNAs?
Oh, I use CRISPR, just case RX. It's only target the RNA. So I just designed the guide RNA on the exon region. Yeah, so it will not affect the expression of microRNA.
Thank you.
Thank you.
Nice talk. Do you know if the link RNAs are working in a conventional manner in which BRD4 functions, in that is it functioning by increasing the ability of the RNA messages to be elongated, so to increase the elongation phase of transcription
I
through
think,
PTEF-B?
okay, thank
Or
you.
is it working through some other mechanism?
I think the mechanism will be related to both the enhancer and elongation bits, because I also have some data about the nucleus spical, which is just elongation complex. Yeah, but I didn't show this for the time limitation.
Thank you.
Thank you.
So there are BRD4 inhibitors in clinical trials. Have you, do any of those inhibit the growth of the tumor cell lines?
No, I just used the BRD4 inhibitor. I didn't try other kind of drugs.
You might be able to get those inhibitors and find out.
Yeah, thank you.
Okay, thank you, Gian. So, okay, the next talk, it will be by Suk-Kyung Lee from the Catholic University of Korea, and the title of her talk is Epstein-Barr virus MIR-BART1-3P regulates the MIR-1792 cluster by targeting E2F3.
Okay, thank you. I am also grateful for given to present our data in this beautiful meeting at Siena. TCGA network carried out a molecular biological analysis of clinical samples from 295 global early gastric cancer patients. The gastric cancer cases were classified into 4 subtypes based on the molecular characteristics. Of these, EBV-associated gastric carcinoma accounted 9% of total stomach cancer cases. In EBV-associated gastric carcinoma, EBV establishes latency 1 infection and expresses only a limited number of EBV latent genes shown in blue here. EBNA1, LMP2A, and EBERS, and BART microRNAs, and some more. However, the well-known EBV oncogenic proteins such as LMP1 or EBNA2, EBNA3s are not expressed. thus, it's interesting how EBV can cause cell transformation and cause cell proliferation and block apoptosis. Thus, we were interested in the role of part microRNAs in EBV-associated gastric carcinoma. A long time ago, we showed that EBV microRNAs, only parts microRNAs, but not BHRF1 microRNAs are expressed in EBV-associated gastric carcinomas, as well as in EBV-infected gastric carcinoma cell lines. Since then, we showed that several part microRNAs are suppressing some EBV genes such as BGRF1, BRRF1, or cellular targets like BAD, DAG, DAP2, such, as shown here. MIR part 13P are highly expressed in EBV-associated carcinomas such as NPC and gastric carcinoma. Thus, we tried to find a target of this microRNA, and previously we showed that this microRNA targets DAP2 and blocks gastric carcinoma cell apoptosis and promotes cell migration. As our microarray analysis showed that E2F3, a cell cycle regulator, was inhibited by EBV infection, we hypothesized that this microRNA regulates E2F3. Here we tested whether this microRNA regulates the cell cycle by suppressing E2F3 expression in EBV-associated gastric carcinoma. We also assessed the effect of mere BART13P and E2F3 on the expression of mere 1792 cluster. MTT assays were carried out in AGS and AGS EBV cells transfected with mere BART13P mimic or inhibitor to this microRNA. Proliferation of AGS cells were slightly reduced by this microRNA, and inhibitor boosted the proliferation in EBV-infected AGS cells. And cell cycle regulation was analyzed after propidium iodide staining. Compared with the cells transfected with the scrambled control, microRNA mimic transfection increased the proportion of cells in G0G1 phase a little bit. In contrast, in AGS cells transfected with inhibitor 2 part microRNA had a reduced G0G1 phase population. As the G0G1 transition is regulated by EGF family, we investigated which EGF family member is involved in cell cycle arrest following this microRNA transfection. Expression of E2F1, E2F2, and E2F3 were analyzed in GEO data. And compared to EBV negative gastric carcinoma, E2F1 and E2F3 expression was reduced in EBV-associated gastric carcinoma in tumor tissues. However, E2F2 expression were not different. In cell lines, we found that only E2F3 expression was suppressed by EBV infection. And this panel shows that E2F3 expression in the cell line was suppressed after EBV infection, as shown here. To determine whether E2F3 is a direct target of this microRNA, luciferase reporter assay was carried out using, this is, when we assessed whether the reduction of E2F3 expression was related to about 1-3P, we obtained this result. Expression of E2F3 messenger RNA level was suppressed by part 1,3P mimic, as well as SI E2F3. And the protein level was showed the similar result. So E2F expression was suppressed by MIR BART 1,3P and SI E2F3. In AGS EBV cells, inhibitor to this microRNA increased the expression level of E2F3 messenger RNA as well as protein level. For comparison, the effect of the siRNA specific to E2F3 on cell cycle distribution was assessed following PI staining. Compared to the control, as shown in white bar here, SI-E2F3 transfection increased G0, G1 phase and reduced the proportion of the cells in the other phase cells. To test whether E2F3 is a direct target of this microRNA, luciferase reporter assay was carried out using HEC293 T cells co-transfected with 3' UTR luciferase reporter construct P-site check E2F3 and the microRNA mimic or mutated microRNA. The luciferase activity of P-site check E2F3 was significantly reduced by this microRNA mimic, but not by the mutant form. Similar results was obtained in AGS cells too. Then we wanted to check whether this is really specific result. So we constructed the mutant form of E2F3 3' UTR reporter construct. And when we transfected this mutant form construct, we did not see the effect of this microRNA, while the wild type 3' UTR construct still showed the reduced expression using MIR BART 1,3P. When this mutated construct was transfected into AGS EBV, the inhibitor, first, when the wild type 3' UTR reporter construct was transfected into AGS EBV, we could see that even with using control inhibitor, there was a reduced luciferase activity. This means that endogenously expressed MIR BART1 3P inside the AGS-EBV was enough to suppress the expression of E2F3. However, when the inhibitor was co-transfected, this suppressed enhanced expression was recovered. Suppressed expression was recovered. Thus, we could suggest that this microRNA is really targeting the E2F3 3' UTR. Previous studies have shown that E2F3 binds to the promoter of this MIR1792 cluster, and it activates its transcription. As E2F3 expression was suppressed in AGS-associated gastric carcinoma and EBV-infected AGS cells, we examined whether reduced E2F3 expression affected the expression of MIR1792 cluster. The expression of this cluster host gene, MIR17HG, in AGS-EBV cells were lower than AGS cells. This was also shown in EBV-associated gastric carcinoma compared to EBV-negative gastric carcinoma. So this MIR17HG gene expression was suppressed in EBV-infected cells and gastric carcinoma tissues. The expression of individual microRNAs within this cluster was analyzed using the cell line and tumor tissues. So we extracted RNA from the cell lines and tumor tissues and did the RT-PCR analysis to assess the expression level. As shown here, 4 out of 6 individual microRNAs showed suppressed expression in AGS-infected cells. And here, like 5 out of 6 individual microRNAs showed suppressed expression in EBV-associated gastric carcinoma compared to EBV-negative gastric carcinoma. To determine if this microRNA regulates MIR1792 cluster expression, the effect of this microRNA and the inhibitor to this microRNA were examined. The AGS and AGS-EBV cells were transfected with the microRNA mimic or inhibitor to the microRNA, respectively. Expression of MIR17HG.
Please, we're running out of time. Could
Okay.
you please?
Okay, so the cluster, this cluster expression was also suppressed by the microRNA, and individual microRNA had the similar effect, and inhibitor increased the cluster expression, as well as siRNA to E2F3 showed a similar effect to the microRNA. This just shows that in this microRNA is expressed in EBV-infected cells as well as the tissue. And summary, we showed that this MIR BART1 3P and siRNA specific for E2F3 suppress the expression of MIR17HG and MIR1792 cluster. And As the ETF3 directly binds the transcription of MIR 1792 cluster, while microRNAs encoded within this cluster inhibit the translation of ETF3, we suggest some feedback loop linking between MIR BART13P, ETF3, and MIR 1792 cluster microRNAs. Further studies are required to clarify how it actually works. Thank you very much.
Okay, we will take one question. Okay. Okay. Okay, please. The next one is next speaker, Rena Xian from Johns Hopkins School of Medicine, and she will talk about can EBV CpG methylation in plasma distinguish EBV lymphoma subtypes.
Okay, good morning. All right. So thank you to the organizers for allowing us to share our work with you today. So this is actually my very first EBV meeting. It's been a fascinating few days. So today, switch gears a little bit and talk about something quite translational, which is plasma cell-free DNA as a diagnostic marker in cancer. So some of you may know, some of you may not, that over the past about 10 years or so, this entire field of cell-free DNA research has exploded and has applied to many, many different diseases, mainly cancer. So the detection of circulating tumor DNA has been really informative in the initial screening for cancer, detection, diagnosis, in treatment selection, and even in minimal residual disease monitoring. So our group has been trying to apply this liquid biopsy technique to the diagnosis of HIV-associated lymphomas. And mainly our work is grounded in Africa, South Africa, where HIV is endemic. So approximately 20% of the adult population is HIV positive. And maybe surprisingly to you that the number one cause of mortality is actually not HIV or cancer, but rather TB. And the presenting symptoms and signs of TB and lymphoma are quite similar and very difficult to distinguish clinically, and therefore the diagnosis of lymphoma is often delayed, not only related to the TB confounders, but also related to limited resources, limited skilled physicians. So our group has been focused on trying to develop a molecular technique that can expedite lymphoma diagnosis. And as a molecular pathologist and hematopathologist, I'm very interested in new biomarker discovery and validation. And so we've now applied many techniques, including clonal immunoglobulin sequencing, mutation detection, rearrangements to plasma DNA. But obviously this is an EBV meeting. So I'll tell you about our preliminary findings, our early findings in EBV DNA. Okay, that was the preamble. All right, so to date, we have studied approximately 50 HIV-positive lymphoma patients, and you can see that looking at plasma, so plasma EBV levels, those with EBV-positive tumors obviously have higher levels, but those with EBV-negative tumors also have detectable EBV. And then there's a subset where we're still working on the tissue in situ hybridization studies, but you can see all of these ranges are quite blended. And so we thought that looking at levels of plasma EBV is unlikely to be sufficiently specific to diagnose lymphoma. Now, in thinking about a diagnostic test, we actually focus on two particular types of lymphoma that are highly EBV associated and also represent a large fraction of lymphoma in South Africa, namely Hodgkin lymphoma and plasmablastic lymphoma, which is a type of non-Hodgkin lymphoma. Now, similar to Burkitt, where there are different rates of EBV association, i.e. endemic versus sporadic, in Hodgkin lymphoma, the HIV patients who have Hodgkin lymphoma are virtually all EBV positive. Whereas in immunocompetent people, it's about 20 or 30%. So plasmablastic lymphoma, on the other hand, is a non-Hodgkin lymphoma. It's quite rare, but it's highly enriched in immunocompromised individuals, particularly HIV. So you can see both are highly EBV associated. However, they look quite different under the microscope and actually have quite different EBV biology. So on the left-hand side, you can see Hodgkin lymphoma, which is, many of you know, it's rare tumor cells, that express EBV-EBR, you can see down here, in a sea of immune cells. whereas plasmablastic lymphoma is sheets of tumor cells that express EBV with very little immune cell infiltrates. While both have latency to program EBV, lytic expression, gene expression is never seen in Hodgkin lymphoma. However, actually Lorenzo Leoncini's group right here, thank you very much for this, actually showed that plasmablastic lymphoma, many of the plasmablastic lymphoma tissue tumor cells, so tumor cells not bystander cells, tumor cells have some lytic gene expression, such as BZLF1 and other early genes like BMRF and BHRF. So here, in their study, they show very nicely the RNA expression levels of these transcripts at the top and the corresponding protein expression by immunohistochemistry. And so as we think about this, we were thinking, well, virion DNA should never be methylated. And so if we look at methylated EBV DNA in plasma, we might be able to actually distinguish different tumor types that have a different EBV biology. And so work from our group previously have shown that there is a simple technique that can be applied to plasma, actually to any DNA, to give you a relative fraction of methylated DNA. And so this technique takes advantage of this methyl DNA binding protein, which is conjugated to a magnetic bead. And so any methylated DNA will be captured and unmethylated DNA will flow through. And so if you collect all the different fractions and apply qPCR, for example, for your favorite gene or favorite locus, you can actually derive a relative percent methylation. And so in our study, we looked at patients with Hodgkin lymphoma that are HIV negative, so this is not HIV yet, but Hodgkin lymphoma patients who are HIV negative who either had EBV positive disease or EBV negative disease, we found that while most patients with EBV negative Hodgkin's do not have detectable EBV, when they do, the levels are actually quite similar to those that have EBV positive disease. However, when we look at methylation by this assay, we found that virtually none of the EBV negative Hodgkin patients had any methylated EBV DNA, suggesting that the EBV we saw right in plasma in this group was likely tumor or cell-derived at least, and not virion DNA. So a similar story then was found in HIV patients. So now this is AIDS patients with KS, no lymphoma. Again, most do not have detectable plasma EBV, but when it's there, it's quite high. And so it's quite high, similar to levels found in EBV positive diseases. However, again, you can see none of this is methylated, suggesting that this DNA is likely the result of viral replication rather than cell replication. And so here I show you the data now from our HIV patients who have either Hodgkin or plasmablastic lymphoma, and you can see they're separated by the tumor state. And while the EBV positive tumors usually have higher levels, again, these ranges are quite blended. However, when we look at methylation, we see that the Hodgkin lymphoma patients usually have very high levels of methylation, whereas the plasmablastic lymphomas have more of a range. Now, this is a single locus. This is actually WP, and this is showing you the differences that this potentially could differentiate lymphoma subtypes. Now, we wondered what would happen if you looked at the entire EBV methylome. Well, this is when we discovered the work of Dennis Lowe's group, where they did bisulfite sequencing of the entire EBV genome in plasma. So this is not primary tumors, this is plasma. And they showed then that methylation profile could actually separate different disease etiologies, for example, infectious mononucleosis, lymphoma, and of course, nasopharyngeal carcinoma. They went on to show that select differentially methylated regions of EBV could distinguish healthy controls that screen positive for plasma EBV, but do not have nasopharyngeal carcinoma and could actually be used as a diagnostic test for NPC. Now, they were not particularly interested in lymphoma, and we were, so we actually requested and were granted access to their raw data. So we performed our own bioinformatics analysis of their next-gen sequencing data. And here I'm showing you several genome profiles of EBV. So if you look along the x-axis, this is the linear version of EBV genome. On the y-axis is percent CBG methylation at particular marks. And you can see that this example is a patient with Burkitt lymphoma, sporadic, but EBV positive from Hong Kong. And you can see that the levels of EBV methylation is actually quite high throughout the genome. This is an example of an NKT cell nasotype lymphoma, which has, again, very high levels and quite uniformly high levels. This NKT cell lymphoma is interesting because there's actually a bimodal pattern with the low methyl marks interspersed with the high methyl marks. marks. And then lastly, here's an infectious mononucleosis patient who never really achieves high levels of methylation, and the methylation ranges are quite broad. And so if we look at the entire cohort, we can actually distill this down to two regions of EBV. Firstly, there are several loci that are localized to CP that are highly, highly methylated in these lymphoma patients, but never methylated in the infectious mononucleosis patients. Conversely, there is a different set of CpG loci that localize to a coding region of BMRF1 that are methylated to a somewhat modest degree, but methylated, and never methylated in the tumors. Now, genic methylation, some of you may know, actually correlates with gene expression rather than suppression. And so there may actually, this data suggests, now it cannot be proven yet, but suggests that there may be some BMRF1 expression in infectious mononucleosis. So we were really intrigued by this, and we actually then went on to develop our own next-generation sequencing assay, which actually covers the repeat. So I don't know if you noticed earlier, the genome plots had gaps, and those gaps actually represent the large internal repeats of EBV. But because we were studying W, we really needed to look at the repeat regions. And so to do this, we developed a hybrid capture-based approach, which has probes that are specific for EBV unique sequences and also for repetitive sequences. And since there are several units of a particular repeat, many units, we use the same probe to cover the entire region with junctional probes that offer some specificity. And so what this looks like on the genome plot is you'll have the apparent gaps, right, where the probes do not span. However, once you sequence and bioinformatically align, you can actually randomly assort the sequences to the repeat 1 or 2 or 3 to actually fill in the gaps. And so far we actually have sequenced several cell lines, and obviously with a plan to sequence primary patient samples. So the top and bottom plots are coverage plots running across the EBV genome, and this is AGA76. The top plot shows you native DNA sequencing, whereas the bottom shows you the aggregate coverage of bisulfite sequencing, and you can see that we have relatively uniform coverage of both repetitive sequences and non-repetitive sequences, and those eagle-eyed in the audience will see that there's actually a very large internal deletion in the last third of this R strain of AG8, as represented by this massive drop in coverage. And so what does the methylation look like? So these are early days yet, but you can see that we can plot, again, a genome methylation plot across the entire virus to show you both more methylated and less methylated regions. And so some of you may think that this resembles the infectious mononucleosis patient, and actually you would be right, because AG8 actually has a latent C3 pattern of expression similar to infectious mononucleosis. And so future work will apply this to both patient tumors and also plasma. And so here are some early conclusions we can draw from this. First is that patterns of CpG methylation in plasma are associated with different EBV conditions. Now, these differences in Hodgkin lymphoma and plasmablastic lymphoma may reflect differences in viral gene expression in the tumors. Now, whole genome bisulfite sequencing may actually distinguish the different types of lymphomas, and studying the methylome may lead to novel diagnostic assays in HIV-positive patients. So I'd like to thank our collaborators, lab members, funders, and I'm happy to take questions.
Hey, nice talk. So when you talk about AG876, are you talking about the Burkitt lymphoma line?
Correct.
Because, you know, that genome has been sequenced. And
correct.
And all the genes were there. So what do you think happened in your line?
So we're not quite sure. This was actually very unexpected, but it's not that there's an entire deletion. So I don't know if it's apparent, but there's actually low-level coverage here. So we actually have done this now in both the cell, so EBV extracted from cells, and also lytic-induced EBV, and we have seen the same pattern. So it suggests that some episodes, most episomes have actually lost spontaneously, potentially, this entire region, but there are some competent full-length viral episomes in there. Don't know why or what's happening, but.
Are there other questions?
Yes. Yes, that is, we could talk. Hi.
Very nice talk. Thank you. I was wondering, now you have distinguished different tumor types, and obviously these patients will undergo some sort of treatment. Or what happens to methylation during the treatment? Do you have follow-up samples here before and after treatment?
Yeah, so those are in the works. So we have been following these patients now who obviously have gotten treatment and many cycles. So we have interval plasma samples that we plan to study. Now, your question is probably getting at what happens after treatment, right? The tumor DNA hopefully goes away, i.e. the methylated DNA hopefully goes away, and potentially you have either no EBV DNA or unmethylated, right, EBV DNA. So we're actually studying that area. Thank you.
So hi, Rena, very nice talk. So it's obvious that the methylated DNA would reflect the intracellular version, the tumor version, but the non-methylated DNA, is there a way to distinguish whether or not you're having viral production, or if it's just reflecting unmethylated DNA from tumor cells?
Yeah, absolutely. That's a great question. And again, with Lorenzo's work, we don't actually know if there's some maybe abortive replication that not competent, right, viral, not full particles. So this is actually my collaborator Rich's area of work. So we could actually do some maybe treatments to see if there are viral particles in plasma. We haven't done that yet. This is just whole lysate, whole plasma, but that is potentially an area to look at whether we have true viral particles or not. Yes.
Hi, one last question here. So I think it's very interesting what you've done with the probes to try and cover the repeats.
Yes.
And obviously this is a really important part of the genome. How do you think this will work in cell-free DNA versus plasma DNA, especially considering the number of repeats is so variable amongst different strains and viruses?
Sure. No, and the short answer is we wouldn't know. So this is mapped aligned to the reference EBV genome, right, which has, for example, the W is like 7.5 cassettes or something. So we wouldn't really know. I mean, we might see it as evidence of differences in coverage, but then that'll be aggregate, right? So aggregate from any cell, any EBV positive cell that has died or or virus. So we wouldn't necessarily know, but that is an area we're trying to resolve because, as you can see, so down here, right, this is W, for example, this is randomly assigned methyl marks, right? So we don't actually know what belongs to the first, second, third. I think something like nanopore sequencing in larger fragments could tell you whether the sequence, right, the order of events matters. But so far we're looking at, we don't know random, but we could also look at aggregate. So part of an answer.
Okay, thank you, Rena.
Thanks so
We can
much.
move to the next talk.
Thank you.
Okay, our next speaker is my co-chair, Eleni Anastasiadou. She's at Sapienza University in Rome, and the title of her talk is Epstein-Barr virus, From Kisses to Cancer, an Ingenious Immune Evader.
Okay, thank you. Thank you for the introduction. Thank you to the organizers for giving me the opportunity to talk about how EBV can alter immune checkpoints through EBNA2 and microRNAs. So if there are kisses or cancer, in either way, EBV can escape, must overcome immune recognition, and to do so uses and this is one of the stories that I would like to tell you about. So establishes needs to establish its latency. So here is a table with EBV-associated diseases. Of course, there are not all the diseases. There are many diseases, but you all know about the cell origin and EBV association and latency type. And my studies during all these years are centered in diffuse large B-cell lymphoma, and which constitutes the almost the 30% of non-Hodgkin's lymphomas in immune competent patients. And so EBV can cause cancer in these patients. And so then to do so can induce, as we said before, also yesterday we talked about that EBV can induce PD-L1 to evade immune surveillance. So there are several studies about that. And we focus our research on how microRNAs can target PD-L1. As you can see here, there are several microRNAs that can target PD-L1. And this is what I'm going to talk about today. But in diffuse large B-cell lymphomas, it was not known which latent viral protein was induced high levels of PD-L1. So this is exactly what our study is about. So this is an article which we have already published, so I will go very quickly through it. But I would like to show to you that we have infected DLBCL, Burkitt lymphomas, and with an EBV recombinant virus, and which establishes latency 3, as you can see here, where EBNA2 is highly expressed. There is also an induction of PD-L1. And we, in order to see which of the latent proteins is involved in this induction, so we transfected the DLBCL with EBNA2 and LMAP1, and also BL41, which has an inducible EBNA2 when you add beta-estradiol in the culture medium. So we found that EBNA2 was responsible for the induction of PD-L1 and not LMP1. So as I told you before, show you that in AML, acute myeloid leukemia, MIR34A targeted PD-L1. So we actually have also another paper about that. So I thought that maybe EBNA2 can somehow interferes with MIR34A to induce PD-L1. So I checked the expression of the mature MIR34, the precursor, and also I have studied also the relative luciferase activity of MIR34A promoter. And as you can see here, there is a decrease in not only the mature microRNA, but also in the transcriptional levels. So when I reconstitute the expression of the MIR34A, I noticed that there was a downregulation of PD-L1 through flow cytometry and also by Western blot. And so the reconstitution of MIR34A downregulates PD-L1 in DLBCL. So in order to understand the molecular mechanism behind this regulation, so we studied the chip sequencing dataset, and we used JASPAR-TF analysis, which showed that there was a peak of EBNA2 in EBF1 binding site and coincides with the MIR34A promoter. So we confirmed this result through the use of short hairpin EBF1. And so we saw that in DLBCL EBNA2 transfected cell line, the MIR34A expression was reconstituted and PD-L1 expression was downregulated. So the same thing we observed also by using the relative luciferase activity of MIR34A promoter, and we saw that there was a de-repression of the promoter activity of MIR34A after knockdown of EBF1. So what is the outcome of this? Because since we are talking about EBNA2 induction, PDL1 induction by EBNA2 through downregulation of miR34, we wanted to see what was the immune response. So we measured the interferon gamma production by CD8 and CD4 in a mix in MLR, using MLR. And we saw that when we overexpress mimic miR34A with mimics, so interferon gamma was significantly induced. And in order to see, so I'm going to show to you something very innovative. So in order to see what was the outcome of this, the induction of interferon gamma by CD4 and CD8, we use a more physiological let's say co-culture, which we have put allogenic PBMCs, activated PBMCs, and DLBCL, EBNA2 transfected, and in these microfluidic chip devices. And this is because we want to imitate a more physiological environment. And we saw that when we transduced this cell line with a lentiviral vector, and these in green GFP cells, they are the DLBCLs, without the presence of miR34A and with the presence of miR34A. And we saw that, so if you look at this panel here, CD8 and CD4 cells were remaining outside of this channel, and in the presence of miR34A, which remind to you that downregulates PDL1, CD8 and CD4 will be called inside the channel where the DLBCL are, and will induce caspase-3. And so we use this device in order to demonstrate that there was an increased caspase-3 activity in the tumor cells, so to reveal the T-cell dependent killing. So in order to recapitulate our results in vitro, we used biopsies from DLBCL patients, and as you can see here, I have highlighted the biopsies which express high levels of PDL1, and they're EBV positive and EBNA2 positive activated B-cell lymphoma. So, so there was an increase in not only the positivity of PDL, but also the intensity of the staining, as you can see here. So those are the biopsies with EBV positive EBNA2, and those are the EBV biopsies, positive biopsies with EBNA2 negative, and those are the controls. So we were very happy to see this article because somehow confirms our results, so that the presence of EBNA2 in DLBCL patients induce, let's say, poor overall survival. So the presence of EBNA2 positive comparing to EBV positive, but EBNA2 negative. So EBNA2 positive patients have poor overall survival. We can think now about the fact that EBNA2 induces PDL1, so we could use antibodies against PDL1. But as many of you know that while encouraging, the antibodies alone are not sufficient. As you can see here, the use of, in many types of cancer, the use of only antibodies gives no response in the most of the patients. So we proposed our patent, we proposed a patent in which we can use miR34A and antibodies, anti-PDL1, to give a helping hand in the immune system. And so as you can see here, the combination of miR34A with mimics, with antibodies against PD-L1, induced tumor immunogenicity. So, and here you can see that there is an increase of caspase-3 in the presence of miR34A and antibodies against PD-L1 comparing to the single agents. So I talked about how EBNA2 can alter an co-inhibitory PD-L1, but we were asking if EBNA2 can influence also other immune checkpoints in DLBCL. So we looked at the co-stimulatory, which has the opposite effects comparing to PD-L1, the ICOS ligand. As you can see here from this cartoon, the presence of ICOS ligand in APC or tumor cells may have an anti-tumor T cell response. And there is also a paper, and also another very important point of ICOS ligand is that it is important, essential for the germinal cell formation. And another paper showed that in diffuse large B-cell lymphoma, ICOS ligand high expression is associated with a prolonged survival time. I took the cell lines, DLBCL and B-cell lymphoma transfected with EBNA2 to see whether ICOS ligand, the effect of EBNA2 on ICOS ligand expression. And this is the results by qPCR. And we showed that ICOS ligand is downregulated in both cell lines. And this was also confirmed by flow cytometry, in which we show that there is a downregulation of ICOS ligand in the presence of EBNA2 in both cell lines. EBNA2 reduces ICOS ligand. This is the conclusion of this experiment. And then by searching algorithms that, searching also microRNAs that can target ICOS ligand. We read this paper on BMC bioinformatics, which showed that miR34A was targeting ICOS ligand and also miR17. But we found that also, this is also very interesting, a microRNA, viral microRNA BART3 is targeting ICOS ligand. So to confirm this prediction, started to study the mature expression of miR34A. So in EBNA2, and I saw that in the presence of EBNA2 in all these cell lines was upregulated. So I confirmed the interaction between the 3' UTR of ICOS ligand by using the ICOS ligand 3' UTR look assay. And I saw that when I knocked down miR24 in this cell line, so in the presence of EBNA2, so there is a release of luciferase activity of ICOS ligand. Indeed, ICOS ligand is an authentic target of miR24. So next, I wanted to see if the reconstitution, if I inhibit miR24, so I used as a control the parental cell line, and so if by knocking down miR24, I was able to reconstitute ICOS ligand. Indeed, I found that in the presence of EBNA2, so there was a reconstitution of ICOS ligand expression. And so in collaboration with Professor Leocini's group,
Sorry, Eleni, you're
Yes,
running over.
yeah. So I'm finishing. We found that in EBV positive activated B cell diffuse large B cell lymphoma, so the presence of the virus reduces ICOS ligand, so confirming our in vitro studies. So in conclusion, EBNA2 can act both either ways in order to for the virus to immune escape. EBNA2 can bind the BF1, the promoter of MIR34A, by upregulating, inducing BTL1. And on the other hand, and now we're studying this mechanism, how this works, can induce MIR24 expression in order to downregulate ICOS ligand. So this is the virus that immune escapes. So, and based on our patent, we proposed also the use of MIR34A mimic and MIR24 inhibitor in order to reconstitute the immune cell anti-tumor immunogenicity. Thank you all for your attention. And I can take some questions now.
Okay, I think we just have time for one question. If anybody has one?
So very nice. In the PDL1 targeting that you promote or suggest, right, in the patent, you want to downregulate it once transcriptionally or post-transcriptionally and one by blocking with an antibody. How do you see the synergy working? Because, I mean, if you target by two mechanisms the same molecule.
Yes. I really showed that, okay, so the first question, so the answer in your first question is that, so the idea was to use a microRNA, of course, to downregulate the messenger RNA PDL1. And so by using the antibody, I can block the PDL1 on the surface of the same cell. So, of course, we need, it would be great to see this effect in a single cell, in a single cell experiment that we're planning to do. But it was, let's say that it was a dramatic induced of apoptosis when I combined both, so the mimic and anti-PDL1, because we had an activation of caspase 3 when we used alone MIR34A and anti-PDL1, but both we noticed a massive apoptosis.
Okay, thank you, Eleni. We'll move to the next speaker. So next speaker is Eric Johansson from the University of Wisconsin at Madison. And the title is an EBV mutant derived, deleted for the immediate early locus reveals RTA to be the dominant early gene activator and Zeta acts primarily on ORILIT.
Okay. 1 second. So thank you all for getting up early after, hello. I think we had it. Is somebody helping me or maybe don't do that? Less help is better. Okay. Thanks for the opportunity to speak. I am giving this talk on behalf of my postdoc who would have really relished the opportunity to come but was unable to get a visa. So I'll be giving his talk instead. Here we have the EBV lytic cascade that we're all familiar with and which is controlled by the two immediate early transactivators, RTA and TTA, as was nicely reviewed by my colleague Shannon Kenny in her Henley talk last night. So I won't go into detail except to pose this deceptively simple question: which early genes does RTA and ZTA activate? And cite two studies that really inspired this question, and also illustrate the challenges of trying to get a straight answer. So the first study is this sort of classic by now study by Regina Federley that really put this into context, the idea of which is the more important transactivator, RTA or ZTA, when she was able to show that without both, you don't get past DNA replication. So they're both absolutely essential. But because this was sort of in the pre-RNA-seq and high throughput era, we had relatively little information about exactly which of the early genes RTA versus ETA was responsible for. And the problem that study really highlights is that RTA and ZTA co-induce each other. And so even though it's nice to be able to do this in the context of a whole virus, unless you do what they did, which is delete out one of the other two, you are always sort of studying them or risking studying them both at the same time. The second inspiration is this study by Cora Willey in Shannon's lab that showed this really remarkable finding that ZTA is less important, considerably less important than RTA in epithelial cells, and this is because the EBV genome is hypomethylated. And so we can see here that the BMRF1 gene is sort of the rule where in the methylated state, Z is quite strongly able to upregulate it, and then when hypomethylated, this reporter Z is quite weak. There were a few exceptions. For example, this early gene BHRF1, which encodes EAR, could be activated by Z actually better in the unmethylated state. And then at ORI-LIT, Z was quite good in the unmethylated state. But of course, reporter assays are predictions, and we've all been lied to. So it's really important to verify this. And so the way we chose to do this was to construct a EBV genome that was deleted for the immediate early locus. And so I won't belabor the details, but we were able to do this, and we've derived a number of different cell lines with it, including epithelial cells and some LCLs. The latter I won't be talking about. I'll be focusing on our results in epithelial cells, particularly the Knox cell line, which are sort of a highly physiologic model of EBV epithelial infection. But I first want to try to convince you that this is really important and just give you one illustrative example where, and this is a result that has really been published many, many times in different papers, but ZTA really appears to be, when you do this study in the context of the whole virus, the activator of BMRF1 expression, which I've sort of highlighted in blue here. And RTA doesn't seem to turn it on at all. There is actually a very weak band that's there, but nothing like what you get with ZTA. But when we look at what happens with the Delta RZ virus, it turns out this is a complete lie, and that ZTA can't turn on BMRF1 at all, and that RTA actually is turning it on a very little bit, but not very much. And if we do a longer exposure for our RTA blot, we see the reason for this is that the tiny little bit of RTA that you can easily miss in the Western blot is doing all of the heavy lifting here in terms of what Z is able to apparently turn on. And so this co-induction problem is very real, and it's quantitatively significant. We've done our due diligence. The virus is otherwise replication competent as long as you provide RTA and ZTA and trans. We can get late gene expression. It's got a GFP. We can turn a reporter gene in it, so we can turn various cell lines green when we package virus with transcomplementation. And so this is our experimental design. We've done this in several different cell lines, but again, I'm just going to be talking about the Knox cells, where we simply transfect in either vector, ZTA alone, RTA alone, or both, harvested 24 hours and performed RNA-seq. And then we want to determine which early genes are activated by RTA versus ETA in epithelial cells. And so in the latent state, without transfection, we really only see EBER expression, and these are actually our reporter genes over here to the right. And then BNLF2A, interestingly, is sort of an interesting example of a keratinocyte-specific gene. So we don't see this turned on when we do this in 293 cells, but it is there in the latent state in this, and of course it's a latency gene in gastric cancer and other keratinocyte models. And here's the big reveal. So Coral was correct, but the dichotomy is much more stark. So ZTA is not relatively less important for early gene turn on. It's incapable of early gene turn on. And all it's really doing is activating the ORI-LITs. I'll point out here that we have deleted the R and Z locus. So the signal that you see here is just is mapping our transfected Z. And we see on both sides of the genome, the ORI-LITs are coming on right and left. And ZTA, as long as BNLF2A is on, ZTA can upregulate it further. But we don't see this peak at all in 293 cells, for example, where BNLF2A expression isn't already on. RTA by itself can turn on most of the early genes. And of course, when we put both in, it's synergistic. And so we really saw three patterns overall of RTA and ZTA dependency. Now I'm switching to 293 cells just for convenience, but we've also confirmed this in Knox. And so some genes, like these two examples here, BSLF1 and BARF1, RTA turns them on quite substantially, and you don't see that much more of an increase when you add Z. On the other hand, there are some genes that are slightly turned on by RTA alone. BMRF1 is sort of a recurring example of this, but they achieve their high level expression when ZTA comes along to cooperate. And then finally, there's some genes, and of course, all of the late genes fall into this category because you don't get DNA replication without both. But there are also some early genes that fall into this pattern where you really don't get any expression due to R alone, and you really need both at the same time in order to get expression of these genes. And so I'll be using these different names. We're calling the first category RTA responsive. This synergistic thing is appropriately called RTA synergy. And then finally, just RTA plus ETA genes for the latter. And so I'll just give a plug for Alejandro's paper on our UTS method, which really helps deconvolute all the overlapping transcripts. And so we didn't have to do cage-seq to get relatively reliable quantitation of this. This was just with RNA-seq itself. And we were able to assign the three different categories over most of the genes. And so again, just to highlight, the ORI-LITs are the only thing coming on with ZTA. RTA is turning on some genes quite well by itself, and then we see another category of genes here where ZTA is contributing substantially, and then there's a few of these where you really require ZTA to get any significant expression. These are all just expressed at quite low levels, and so we weren't able to make a call in that instance. And so really the sort of hemi-conclusion here is that at least in epithelial cells, EBV is far more similar to KSHV than initially appeared, where we have OR50 in KSHV, which is the RTA homolog, of course, is the dominant early gene activator, and the BZIP protein is really just functioning at ORI-LIT to support DNA replication. That really appears to be what's going on with CTA, although it's also doing this other thing, it's synergizing. And so for the balance of my time here, I'm going to talk a little bit about our mechanistic studies about how it is exactly that ZTA is able to synergize with RTA. And importantly, we did all of this in the context of the whole virus. Very quickly, we had the delta ORI-LIT, and since Z was only turning that on, the first thing we did was exclude the possibility that maybe these are just in some way this high-level expression of these ORI-LITs was having effects at other promoters. That doesn't appear to be the case, because even without that, Z is still able to co-activate with RTA for BMRF1. So the ORI-LIT is not required for this phenomenon. It's obviously required for late gene expression. So the mechanisms between this effect on early genes and late is probably different. K8, the KSHV homolog, does not substitute for this phenomenon. And then the next thing we did was perform CHIPS, which we spent a lot of time controlling our RTA and ZTA levels, as shown by these Western blots here. But there's really two things I want to point out. First of all is that at these promoters that Z is not activating, it is actually bound. So it's there and just not doing anything. And although we do see with co-transfection of RTA, so this is Z alone, and this is Z with RTA, looking only at Z binding here. So we see increased Z binding with RTA, but it really isn't specific to the ones where we see a functional effect. So it may be important, but it doesn't explain the selectivity of this phenomenon. Okay, and then we did the reverse with RTA chips, and essentially the story is the same, that we see some increased cooperative binding with both around, but there's no specificity for the promoters that are synergistic. All right. We really considered two broad possibilities in terms of mechanistic synergy here. The first would be transcript stabilization, and I won't go, in the interest of time, I won't go into the details here, just except to say that we considered an NMD inhibition, YTHDF2 CRISPR'd out, and that did not substitute for Z, and also P38 activation does not substitute for Z. So we abandoned that and refocused our efforts on trying to see if we could prove that there was this was actually at the level of transcription. And we did that using a BRU labeling assay. Just shown briefly here, where we pulse label with BRDU, IP it, and then can quantify in a sort of snapshot of instantaneous transcription. This is just a control Western blot, and these are my two conditions, transfected R compared to transfected R and Z at the same time. And what we see is that the RTA responsive ones, there's really no effect of Z, so the transcription is not greatly impacted by Z co-transfection, but that's exactly what's happening with these synergistic transcripts. So it clearly seems to be a transcriptional effect at these synergy promoters, which is interesting since ZTA is not having a direct effect on transcription when expressed alone. So key observations from the study. So in epithelial cells, ZTA effects are limited to ORI-lit. RTA is the main driver of early gene expression. ZTA synergizes with RTA to activate some early genes via transcription, not stabilization. And importantly, these are at promoters where Z is having no effect on its own. I didn't have time to show this, but it comes up in the question. I've got a few slides on it. But we've done a lot of mechanistic studies to look at what is causing the difference with Z co-transfection. And all the way up through to Pol2 recruitment, there's no difference. And so I think it's probably downstream of this. And the most likely thing is RNA Pol2 pausing is being relieved when you have Z. And then, of course, I'll just tease the fact that we're going to do this in LCLs, and the early indications are that ZTA is taking some credit for things the RTA is really doing. And so I'd just like to thank everyone involved in the study, particularly Ahmed Ali, our collaborators, and I will be happy to take any questions.
Okay, thanks. I see two hands. Maybe we can just do two questions so we have a little time for a coffee break.
Nice talk, Eric. Have you looked at S186A, because that synergizes with RTA to activate BMRF1?
Sure, yeah. So it does, as far as we can tell, that you don't require the ability to bind methylated DNA. So the S186A is fully competent everywhere we looked for cooperation with RTA.
But was it included in your RNA-seq, like to look at which?
We did not do RNA-seq on it. So really for economic reasons, for these mechanistic studies, we've largely relied on qPCR, but we've looked at a lot of genes. So it seems to be pretty broad.
And how about the BRF1, like the NA protein?
So it's dysfunctional because of, it's very different. We essentially couldn't delete it here. We couldn't make this deletion without impacting that gene. And so it probably has a efficiency effect on replication, but it's not absolutely essential for us to make virulent particles.
Thank you.
Intriguing findings, Eric. So you've looked at Pol2 recruitment and it doesn't seem to make a difference. Have you considered looking at the balance between PTFB and NELF, perhaps?
No, but we need to start looking at some of those. My instincts have been bad. We have done so many negative studies, and I think we're finally getting close to nailing this mechanistically, but Ahmed's been a real trooper to put up with my bad guesswork.
Okay, thanks very much,
Yep.
Eric. We'll break for a 15-minute coffee break.
We don't fly at weekly on Friday mornings. Oh, okay. So you can have a day tomorrow. Yes.
Any instructions?
Are you in the next third? The second and the third session.
So in the last,
The la-
the last sessions.
Afternoon?
Afternoon, yes.
What is the session? Five.
That's fine. Just a second. So is this a PP?
PDF on PowerPoint?
This is a PowerPoint. Should be a PowerPoint.
Okay.
You just have a look at it. So maybe not on the screen.
Okay. The logo of the mouse.
Can you turn
This.
the thing off?
I'm sorry, but
Yeah. Okay. I just
And
I
the
just
button.
take it look like.
I'm sure. So after that, I don't even bother.
and yes, but the mouse is low. Okay.
No, I don't want to show it here. So
You want?
just here.
Sorry, don't understand.
Just scroll so
Okay.
that I can see the rest of the other.
Yes, but
Okay, so that doesn't work.
my opinion is because
Or I just. This also doesn't work.
my opinion is because the technique. Ma il mouse perché non va? Di nuovo. Adesso che sei arrivato funziona. No.
I think that's fine.
Mamma mia. Ogni tanto logga, capito?
I wasn't sure when.
Adesso funziona.
Magic
Considera
fingers.
che questa pennina ha la portafogli di un braccio.
Ok, ok.
Quindi non lo spostare quanto pare.
Magic, magic.
Probabilmente
Okay.
le garba se la allontano.
No, I don't want to show all the slides.
Ok, sorry.
Le diapositive se le devono modificare.
I just want to see whether everything is okay. It's fine.
Okay.
Can switch it on.
Okay, bye.
Okay.
Your...
Thank you.
Next.
Aspetta che ho sbagliato, eh.
La seconda.
Era... Siamo
Seconda
qui.
sessione di oggi. Ah, pomeriggio.
Sì.
Chi è?
Lui si chiama?
Chiesa. Chiesa. Eccola.
Questo era lui?
Sì. Guarda, c'è scritto, era un fondo alla...
Ok.
Ci scrivono 70.000 cose, scusa.
Ok. What is your turn?
It's the one that was done today, this morning.
Okay. And what is your session?
It's the, no, it's actually it's this one, 14.
Okay.
This is me.
Okay. Yeah, Jeffrey Kahn.
Yeah. With a very, my name spelled a little differently. That's
Oh, I see.
Cohen.
Yes. Like the famous person, right?
Professor.
Like the famous person, right?
Oh.
No?
Not related. If we can just open it up real, like you can just make sure it's, yeah, I'm just going to scroll down real quick. So this is how we move it. Okay, I have extra slides, but I'm going to stop there. Perfect.
Okay.
Thank you.
Thank you.
Thank you. Next.
I'm in the next session. My name should already be on there. It should be, yeah, PowerPoint. Cheerio. Yeah. Wonderful. Thank you.
Okay. Aspettami, ma Cohen is the third session. Is this? Okay. What's your name?
Alana.
Okay. Good.
Would you mind?
Yes.
Thank you. Appreciate it.
Cerchiamo qua in mezzo. Ecco, era la prima, sì. Sempre per la prossima sessione.
Ok. Vuoi dare un'occhiata?
Guardo solo se perché aver messo l'ultima versione. Sì, va bene, va bene. Grazie.
Grazie a te. Il pennino. La prossima sessione ci sono due.
Okay. All right. I'll see you in the break. Yeah. Okay. Yeah. And yeah, this
Okay.
okay.
Beta system.
Yeah, many folders.
And now this is it for the Bethesda system.
Yeah. Yeah. This is yeah. I find for you. Yeah.
No, but
no, this one. Oh, it's the Java side. Yeah. Oh.
The system.
Oh,
the distance
okay.
is the. Read it.
Many folders. I hope it's a 20. This folder is to be.
This is the date of the.
The date, yeah, I know.
The last
yeah.
minute.
Yeah. So many things there. Oh, up. Is there?
Are you sure? Can you find
This one here?
the work of the file?
Let me see. We're close, I think. 20. Oh, here. I think it's this one. This guy. Yeah.
Check.
Yeah. Check it. Yeah.
Is this?
Yeah. I'll go over quickly. If you. Yeah. Just yeah.
This slide?
Yeah.
Remove.
Should be good. Okay.
Okay. Thank you.
Sure, yeah. And the one top is using this mouse to
Yes,
control.
but I understand that the distance is ready.
Okay, don't move too far. Oh, I see the distance, and then the
Yes,
the,
if you
the slides here
in this
so
position, the mouse.
now this mouse
tension.
well, right? Okay, got it. This is here, and it
Or
is
you can slide the.
I see. Yeah, yeah. This also like show the full screen, right? I mean, like on your presentation.
Okay.
And let's see. Yeah. Oh, good.
Or
Small guy. There's everything. Oh, this mouse works.
O così, si va avanti. O così.
Oh, I got it.
Can you
Try
go?
this one? Okay. Oh, got it. So this is like okay, cool. Okay.
Okay.
Thank you.
You're welcome. Ok, allora adesso chiudiamo. Questo qui.
There's still very limited information on how these antibodies were used. Right. And we did some in Parkinson's disease. Because in Parkinson's, this alpha-synuclein, which is the pathogenic protein there, has an epitope that is repeated 4 times in LMP1. So if you have LMP1 specific antibodies, which occur during IM, and they disappear in some patients, they come back in Parkinson's patients. And you have this autoantibody response to the lead pathogenic protein. Right. And thus there is no single key protein that is the pathogenic protein, but it's a number of them. But having one mimicry, I think, plays really a very important role.
Even though what is always a little bit difficult for me to understand is so the B-cell depleting therapies, right? They work actually very well on the memory B-cell compartment and the naive B-cell compartment, right? But they usually leave the plasma cells and the oligonucleotide band untouched.
Have to change.
I guess I'm the first, right?
You're the first
one, right? Yeah.
Then make sure. Was this your first appearance in the EBV Association or not? You were not earlier in the NPC, in the EBV.
I was,
Yeah,
I
yeah.
was, but I don't remember which one was my first.
Maybe the fact that active IM or primary EBV infection is essential in your US military cohort is like it's the one first step that, okay,
Yeah.
there must be something linking.
Yeah, yeah. No, I was, when was it? Was it in Germany? Which year was Germany?
Germany was in the early 20s, early 2000s, I think 2004 or so.
What was this small town? I forgot. I remember.
Regensburg?
Regensburg. I remember Regensburg for whatever, but it may have been there before.
Yeah,
Okay.
Went to the Oktoberfest.
yeah, Oktoberfest.
Regensburg,
Yeah. I climbed the Zugspitze.
Anyway.
What year was the one in Australia?
There were two. One in Cairns, which is I think 2002. Yeah, and there was one later on in Brisbane, but I don't know if that was Brisbane.
Brisbane was 14.
Yeah, okay. Yeah, it was later. And we had an NPC meeting also in Brisbane on the beach.
The Cairns meeting was memorable, right? Because we went to the Great Barrier.
Yeah. Oh, it's boat ride in Cairns. Yeah. A lot of people were sick on the boat.
Oh, no, I didn't go to the Australia one, too far.
Yeah. Well, it was really great. Snorkeling. You stayed healthy on the boat, but many people just were so sick on the boat.
And there was one in Italy also.
I don't know. EBV. Yeah, I remember my first herpes virus meeting was in Bologna. That was 1980.
And then.
But the EBV meeting, I can't remember if it's in here.
I may be wrong, yes.
Yeah, could be. You have
We
not been
don't
to all
publish
these.
the list.
I wonder where it would have been, but maybe I missed it.
Yeah, maybe, I maybe just.
Higher water temperature and maybe lower oxidation.
There are two
Oh,
depictions
you're
of
in
tank.
Boston. I thought you were in New York.
No, I'm in Boston.
Right.
I think this meeting would be too technical for me, because you guys have used all the abbreviation and things and so on.
We
Me,
are.
it's just.
Yeah, of course. But this is a virology group. And
Right, but,
we should more go to a clinical orientation. We used to have more
You see,
dedicated.
my study is from variant, right? The infection, MS, and NFL.
It's just, but I will ask you a question later on.
200.
How do you think it works? How do you think EBV causes?
Well, that's, you should answer that
Yeah,
question, not me, right?
I tried in my paper in November.
I've seen it before. I haven't read it careful, but the one where you
And it's
talk
good.
about
It's the lead-
HHV-C.
Yeah, because it's the leading, I think indeed, that's a good title, leading cause, but there may be additional.
Well, there are always other things, it's, I don't know any disease that is caused by one, maybe rabies, I don't know.
No, it's the genetic makeups that makes you susceptible to these EBV-induced abnormalities. And genetic, you may also include your endogenous viruses, which are reactivated by EBV, which is sort of part in your genome. So that makes you a genetically susceptible person too.
Even though that is, these indigenous retroviruses, especially the superantigens, right? Their imprint is difficult to find, right? Because, for example, where you have that shaping the T cell repertoire quite dominantly, so in wild normal mice, right, like 6 and about C, so they're constantly active, there you can actually see when you look at the V beta distribution, right? So the superantigens selectively expand certain V beta, you can see this imprint. You can see this imprint, right? You see basically
in your humanized mouse model?
No, in normal black
Oh,
six
yeah.
mice.
Okay.
So there it's constitutively, the herbs are constitutively. But when you actually go to humans in IM or in other disease settings, you don't get a biased T cell repertoire to certainly better.
Yeah. So in my hypothesis also, it's like EBV is the leading cause. In some individuals where this endogenous retrovirus promoter is susceptible to being activated, there you might see this effect. The same for HHV6. It's only, I don't know exact percentage, but not everybody has HHV6 in the telomeres incorporated. But when you have, that might give an enhanced inflammation signal, which driving MS.
And you would think that is then activated and trained by T cells or?
By cytokines or by EBV. But cytokines, I think, play an important role. And EBV is the initiator of this cytokine response, T helper 17 T cells. An interesting thing is also this endogenous retrovirus encoded membrane protein. It's syncytin-1. That's a protein that can go to the blood-brain barrier in the endothelial layer and sort of make some leakage. That's a very interesting phenomenon that after that, then T cells may go into the brain even easier. But it's just a very complex area.
And you find antibodies against this?
This by others, Italian groups have done it a long time. And so there is, but it's not like in EBV, it's 100% serological link. And it's ABNA1 antibodies that are different. It's not VCA or EA. There's no lytic component clearly in it. It's typically ABNA1. And ABNA1 probably drives the mimicry to induce autoimmunity, which is also part of the MS.
Do you think that's an imprint because there's somewhere a reservoir that is less potently controlled?
No, we know, EBV homes to the head and neck area. That's its basis. And now we learn there's much more than the lymph nodes in the tonsils and so on. There's a lymphoid circuit in the brain, in the skull even. And so in those little lymph nodes in the brain area, that may be special regions where EBV reactivation from latency may occur under moments of stress, hormone-driven, glucocorticoid-driven. And that's been described quite a lot, that hormonal changes will activate latent EBV. But it may be quite a local event, which then triggers the MS, or at least local inflammation, which can lead to MS in DR 1501.
Yeah, so usually there's a person here who uploads them. I'm not sure where now. Are you in the next session?
Not this one, yeah.
So I think he will. Yeah, but this is usually the right
Just
place.
come here.
I mean, what is a little bit, this infiltrate of EBV infected B cells is quite a contentious area, right, in the setting.
Yeah.
And what is also a little bit unclear is indeed, for the B cell depleting therapies, they work quite well in the absolute limiting disease, but not in the progressive phase. Even though in the progressive phase, then actually also blockers of the immune system into the brain don't work anymore. You could argue, right, then something has sort of set up in the ZNS and does the thing, right?
It looks like in PPMS, then the blood-brain barrier is more closed. It's really a disease that's
right.
Only here.
Yeah. Don't get in.
Yeah, they don't get it. Yeah, exactly. And now there are trials with intrathecal injection of ocrelizumab and rituximab, and they seem to be effective in the PPMS stage also. So B cells, you can knock them out in the brain, but I think it's still a safety issue whether that's possible or not. It's in the trials at least.
Yeah, I also like this hypothesis, but it has been over time.
Yeah. But we should look at HEPNA1 DNA immune complexes circulating in the blood of the CSF of MS patients. They should be there as a sign of EBV B cell reactivation and apoptosis. Because I agree with you, we don't see much B cells, EBV positive B cells, directly in those inflammatory spots in the brain. At least we cannot reproduce the Italian data, and other people have also not been able to do that. Yeah, it's over. In my view, it's really, I think, overstating, because we also do parallel blocks, exactly the same blocks that they used, we used also, and we cannot reproduce the data. But occasionally we find a quarrel.
We probably have to start as soon as.
Yeah. I think
we still have 3 minutes. We still have 3 minutes. We get used to it. Oh, it's about 21. So I go somebody really.
Sorry? Yeah. Whenever you just trying to put my timer because I understand there is no time tracking device. You know, I'll put my phone off. You know.
I can tell you
Yeah.
so 5 minutes before then.
Yeah. No, I'll put the timer.
Good morning. Welcome back to this session on Epstein-Barr virus and multiple sclerosis. It's a pleasure to see you all in this sort of new event where we expand the association of EBV with acute infectious diseases, chronic infectious diseases, cancers, now with autoimmune diseases, and one particular one, multiple sclerosis, will be the topic of this morning. And it's my very pleasure with my co-chairman, Christian Muntz, to introduce Alberto Ascerio from Harvard in Boston, who has published recently very exciting data on the role of EBV or the involvement of EBV in the pathogenesis of multiple sclerosis.
Thank you, and thank you to the organizer for inviting me to this beautiful city. So it was really described in a beautiful way by Charcot already in 1879. It's demyelinating disease characterized by this lesion diffuse in space and time. The pathology is consistent with autoimmune mechanism, but the etiology has always been described as unknown. The possibility of infection was considered early on. And the disease is relatively rare, but it's still the most common disabling neurological disease in young adults. The lifetime risk among US women, just to give you an idea, is about one in 200. And over the world, there are about 2.8 million people suffering from MS. Many infections over the decades have been proposed as the cause of MS, and this has caused widespread skepticism that has been difficult to overcome. The idea that mononucleosis and EBV infection are specifically related to MS risk really go back to the 1980s, originally based on similarity between the epidemiology of mononucleosis and the epidemiology of MS. In addition, it was noted that individuals with a history of mononucleosis have an increased risk of developing MS. And this is a rather specific association, even if you will find dozens of papers on chickenpox, measles, rubella, and risk of MS. If you look carefully at the epidemiological data, there is no evidence that any other disease increase in a consistent way the risk of developing MS. The increase in MS following mononucleosis is not huge, it's between 2 or 3 times, but has been confirmed in multiple studies. Now, as you know, mononucleosis is a very narrow epidemic curve because by the age of 2022, everyone is basically has already been infected. And you are aware also this gradient, socioeconomic gradient that exists in the age of infection with EBV, related to the sanitary condition and hygiene in different parts of the world and socioeconomic group. So this observation led to the hypothesis, the hygiene hypothesis. So according to this hypothesis, the reason we see an association between mononucleosis and MS is that both diseases are associated with high hygiene in childhood. So this low burden of infection in childhood would prime the immune system toward more pro-inflammatory responses, thus increasing MS risk. And at the same time, by delaying the age of EBV infection would cause mono. So this would be a typical example of confounding, with high hygiene being the common cause of MS and mononucleosis. Now, this was 20 years ago, and we thought, well, if this is true, then a prediction of this hypothesis is that those individuals who escape EBV infection because of high hygiene, they should have a high risk of MS. Right? So we start looking at the literature, and it turned out that, if anything, these individuals who were not EBV positive, who escape EBV infection, they seem to have an extremely low risk of MS. So that was prompted the hypothesis, well, how do you go from that middle column, EBV negative, extremely low risk of MS, to the right-hand column? After mononucleosis, your risk is even higher than those who are EBV positive. So you have a jump in 30, 40-fold in risk, going from being EBV negative to being EBV positive. And these individuals come from the same set of individuals who escape EBV infection in early childhood. So the logical hypothesis is that the EBV infection is what causes that increase. Now, the low risk of MS in EBV negative individual was inferred from retrospective studies, including individuals suffering from with MS for many decades. So there were huge methodological limitations. So what we needed to do is to test the prediction in a longitudinal way that individuals who are not infected with EBV will not develop MS. So it's the study I'm going to present conceptually is exceedingly simple. We want individuals who are EBV negative and compare them with individuals of the same age, sex, socioeconomic status, as similar as possible, and see who gets MS and who doesn't. So, and of course, if you are EBV negative, if you do a longitudinal study like this and you are EBV negative, you have to take into account the possibility that you will become infected with EBV during the study. And if the hypothesis is true that EBV is causing MS, then you would expect to see no MS in individuals who are EBV negative or individuals who are EBV negative until after EBV infection, in which case you would expect that the risk would increase. Because MS has a prodromal phase, in addition to looking at the relation between EBV infection and the onset of MS, you also want to look at the relation between the time of EBV infection and the elevation of serum neurofilament, that is a sensitive marker of neurodegeneration and probably a better marker of the very beginning of the pathological process leading to MS. And this is just an example showing that even 6 years before the clinical onset of MS, there was a significant elevation in some individuals in NFL level. So validating NFL as an early marker. So as I say, the study is conceptually very simple. The challenge, of course, is there are very few adults who are EBV negative, young adults who are EBV negative that you can follow longitudinally for MS onset. And the study was made possible by the unique resource of the Department of Defense Serum Repository. These are blood samples collected repeatedly on average every 2 years from 10 million young men and women that we could follow longitudinally for the incidence of MS through the Physical Disability Agency of the US Army, Navy, and Air Force. So we reviewed the medical record of these individuals who were suspected of having MS to confirm the diagnosis according to rigorous research criteria, and particularly to establish the date of the very earliest neurological symptoms. And as you see in the bottom right, the age of this individual is around the age of 17, 18. That is the typical age of recruitment into the military. So being unable, of course, to measure EBV serology in a 60 million sample, you do this study using a nested case control study. This is a standard approach in epidemiology. It's very simple. So when an MS case, there is an incident MS case, you look at the risk set, all the individuals who were at risk at that point in time, and you select control matched on sex, age, race, ethnicity, the date of blood collection, and the military branch. So the result of this study would be identical to those that you would observe in a longitudinal study measuring the EBV serology in the entire 60 million sample, except for a bit more random variation due to the smaller sample size. EBV serology was measured by ELISA as a screening and then confirmed by Western blot. And an important consideration in observational study is the possibility of confounding. So you want to make sure there are no spurious association. And use infection with cytomegalovirus as a negative control. The idea being that if the association that we may see with EBV is due to like a difference in behavior, in number of social contact, in exposures to saliva, we would see a similar association with CMV because it's transmitted in a manner similar to EBV. So these are the main results of the study. Sorry, just was looking at my timer, but the telephone does go off. So at the beginning of the study, 6.8% of these young men and women were EBV negative. We documented 801 cases of MS, of which 35 current individuals were EBV negative at baseline, and only one in an individual who was EBV negative at the time of onset of symptoms. And in terms of seroconversion, you can look at the left. Individual with MS in green, the seroconversion was close to 100% before the onset of MS compared to controls. 50% of the controls seroconverted during the follow-up at a rate that is compatible with that seen in similar population. And as you see on the right, there is no difference between MS cases and control in seroconversion for CMV. In terms of risk of MS, relative risk, if you take as a reference individual who remain EBV negative on the left, those who seroconverted for EBV, the risk of MS 32-folds higher, and those who were already baseline at EBV positive at baseline, the relative risk was 26-fold higher. As you see on the right, CMV infection, either positivity at baseline or seroconversion were not associated with an increased risk of MS. In fact, individuals were CMV positive at baseline at a slightly lower risk of MS, as you see on the right. So confounding is often invoked as a potential explanation for observational studies. It's not always appreciated that a confounder, to explain a relative risk of 32, need to have a relative risk with a MS of over 60-fold, and at the same time increase by over 60-fold the risk of becoming infected with EBV. There is simply no plausible candidate or imaginable candidate that could explain an association of these trends. Another alternative explanation, alternative to causality, that of reverse causation, the possibility that individuals with MS, because of immune dysregulation, are more likely to get infected with EBV. We address this, this question in two ways. One, measuring NFL to be sure that we are really capturing the beginning of the pathological process, and the other by looking with virus scan, looking at the humoral immunity against the entire human virome. So the NFL data presented here, these are within-person changes in NFL. As you look at the left, the baseline is taken at the reference, 0%. At the time of the first EBV positive sample, there is no elevation in NFL in individual who went on to develop MS, and an elevation is only evident in the second sample collected after EBV seroconversion, so clearly establishing that EBV infection precedes the NFL elevation. You see on the right, individuals who did not develop MS didn't experience any NFL elevation. Virus scan, I will not get into the detail for question of time, but basically it's just a way to look at humoral immunity against overlapping peptide that cover the entire human virome. So it's over 100,000 peptides that are being screened here. You can get a sense of the immune response against this peptide, 56 amino acid peptide overlapping by 28. And we did this in 30 MS cases, in 30 closely matched control, with 2 samples for each individual, one before the onset of MS and one after the onset of MS, because the idea was to see if there is any other infection that could trigger the onset of MS in individuals who were EBV positive. And as you see here, it was actually surprised by this data, because they expected a lot of noise when you look at 100,000 endpoints with a serology like this. But all the peptide, virtually all the peptide that elicited an enriched response in individuals with MS as compared to control were all EBV peptide. So the signal for EBV was so strong that basically dwarfs everything else. An interesting observation that there was a lag time between EBV infection and the onset of MS. So the onset of MS occurs several months at least, and often years after EBV infection. And this is just food for thought, trying to understand what is going on. So our conclusion that MS is a rare complication of EBV infections. And obviously there are other factors, there always are. There is no situation in which a viral infection is sufficient by itself to cause a disease, and we investigated the other factors before. But I think the most important in question, and that's why I'm happy to present to this audience, because you're the one who can give the answer to this, not me. How does EBV cause MS? So I think we established the epidemiology that there is a causal link, but we don't know the mechanism. Just a few considerations just to initiate thinking about this. If you look at the top left, those are individual or EBV positive. So we're looking at a cohort of young adults, EBV positive, healthy, and we're looking at their serology years before the onset of MS. And as you see, the anti-EBNA complex titer, here measured by indirect immunofluorescence, seem to be the strongest predictor of future risk, with a relative risk going up to 36. You don't see association of this magnitude in epidemiology unless you're looking at something closely related to the disease process. As you see on the top right is anti-EBNA 1 titers. Whether the difference in the strength of this association, this could be due to technical factors. So I'm just throwing it there. Be happy to follow up with discussion with any of you. Is EBNA complex a better predictor for good reason, or are we just looking at technical differences between these things? Just to finish with some suggestions, some clues from epidemiology on what we could say about the mechanism. So MS risk is not increased with immunosuppression. The EBV viral load is only modestly elevated. There is a strong association with anti-EBNA titers. The number of infected B cells is tiny. And there is evidence of EBV reactivation in MS lesion. That would be really the key point. And you will hear later, I think, from our colleague La Sapienza in Rome. We've been in the Istituto Superiore di Sanità. The striking results showing EBV reactivation in MS lesion, but other labs have produced less convincing or inconsistent results. So that remains an important question to clarify. Overall, all this suggests the immune response to EBV is playing a key role. Also, EBV being an oncogenic virus, I wonder whether the transformation of B cell, oncogenic transformation of B cell could play a role. Anti-CD20, we already basically treat MS by depleting EBV with anti-CD20 until almost depleting the EBV with rituximab and ocrelizumab. Functions of EBV are likely to be important: antigen presentation, humoral factors, the interaction with T cells. And there has been demonstration of dually specific T cells recognizing EBV and myelin being increased in MS. And the chair of this section is also, as you know, an animal model on EBV infection in humanized mice that has provided interesting clues. We'll finish saying this work took more than 20 years, very simple study to, in design, a very challenging to perform, and just acknowledging my co-authors. Yeah, thank you very much.
Thank you very much for this revealing story. I guess there are lots of questions. Jeffrey?
So I'll ask a question for which there's no answer, but could you just speculate a little bit about your thoughts about molecular mimicry or B cell activation or other aspects of, what do you really think is the most likely sort of mechanism, realizing, we really don't know, or maybe there's some other hypothesis?
Yeah, no, the strong association with the anti-EBNA titer, right, would be consistent with molecular mimicry. How do we explain a 30-fold increase? Also, this, I'm not an expert in there. When I read the study on molecular mimicry, including the most recent GlialCam, it doesn't seem to really explain the full thing. 30% of the cases seem to have this cross-reactivity, other cases have other cross-reactivity. It's possible that there is heterogeneity. The molecular mimicry could be an explanation, but not with all the patients recognized. But, it's, it's not a very immediately, explanatory, yeah.
Paul Farrell, just behind.
So I'm just trying to understand about the link to mono. And you mentioned the old data with the 2.2-fold relative risk of nucleosis, but in your relative risk, where you had the 26-fold relative risk, the risk for people who had EBV from childhood or the people who seroconverted in the study seemed to be the same. So I'm trying to understand whether it's just EBV or there is actually a specific relationship to having had mono as well.
Yeah, and unfortunately, epidemiologically, we can only compare history of mononucleosis versus no history of mononucleosis in most large investigation. In this study, individuals were EBV positive at baseline, which means at the age of 17, 18. We know from other epidemiological data in the US that most of them were infected in childhood. About 50-60% were infected in early childhood. And the relative risk was 26 compared to the 32 in the one with seroconverted during the study. But this is a moving target. If we go on, probably those who seroconverted, the risk would go even higher up because we didn't exhaust all the time, all the cases of MS in that group. Yeah.
Two questions. First, the time from infection to MS onset. You have in your paper about 7 years. Do you think that that's the general experience in the population, given what's happening with other studies and the fact that the military is fairly constrained about when those samples were collected? What do you think?
Well, it's likely if we look at the peak incidence of mononucleosis, it's around the age of 15, 16, and then the peak incidence of MS is the late 20s, early 30s. So there is clearly, you just look at the two epidemic curves, that there is clearly a delay, yes.
And what do you think explains why multiple studies show that the cytomegalovirus is protective?
Well, I think probably CMV infection can modify the immune response to EBV, would be consistent with that hypothesis. In our study, the relative risk is 0.8, so for CMV, it's a modest protection. And I think it has to do with heterologous immunity and the way that people infected with CMV may respond to EBV infection.
Shankar?
You may have said this, and if so, I apologize, but of the people who seroconverted, that is, became infected during that period from the beginning of the prospective measurements, was the average age of infection for the number that developed MS different from the average age of infection for those who became infected but did not develop MS?
It was, in a sense, if you individual were EBV negative, because they don't get MS unless they're first infected with EBV, their onset of MS is delayed in time. So if you have two cohorts, EBV positive, EBV negative, in the EBV negative, you don't see any MS. Even if they are started the same age, the EBV negative, you don't see any MS until after EBV infection. So that causes a delay in the age of infection. It's actually a consequence of the causal role of EBV. Does that make sense? So you need two hits. You need first to get EBV infection, and then you become at risk of MS.
I have a
okay.
Yeah. Maybe I have a quick question, short answer possible. In the cohort studies, were you able to analyze whether DRB 1501 is more prevalent in the people who are become EBV positive and develop MS versus the ones that become EBV positive do not develop MS?
Not in this study, but we know that that would be true, right? The RB1501 is associated with 2-3-fold increased risk of MS. I'm sure will be true in this population as any other population. In terms of this main finding, we only have one individual who was EBV negative. Whether that was the RB15 positive or negative wouldn't change our...
That wouldn't change, no, but it's just the pathogenic and the induction of auto response.
Right, the people who are the RB1501 positive tend to have higher EBNA titers. I don't know, there is no study looking at their propensity to become infected with EBV. That would be a difficult question.
With regard to the protective effect of CMV, did you see a protective effect of any other herpes viruses or seropositivity for other herpes viruses?
No, we didn't do the same, longitudinal analysis. The individual in CMV negative will compare individual who are CMV negative at baseline and seroconverted. We didn't do those analyses for other viruses, but the viral scan doesn't really show any hint of possible protective viruses.
Paul Lieberman, last question.
So I'm just curious if there's any anti-correlation with outcomes like Hodgkin's disease, which is also associated with late EBV exposure and mononucleosis. Do we see individuals who are likely to get Hodgkin's less likely to get MS?
No, unfortunately, we don't have this data. It's very difficult to work with these databases for several reasons, and so we're very constrained. There have been studies on Hodgkin lymphoma and lymphoma in this population, and I think they find that elevated anti-EBV titer are associated with the risk of EBV positive Hodgkin. And one difference with MS is that I think they see EBNA, if I remember well, an EBNA2, EBNA1 inversion in terms of the prediction of Hodgkin as compared that we don't see with MS. We see high EBNA1 being predictor of MS, with high EBNA2 being predicted of Hodgkin, I believe.
Thank you.
Okay, let's finish this presentation. Thanks very much. And we'll move on to the next talk by Jessica Allenach from Vancouver, Canada. Epstein-Barr virus infection promotes T helper 1 mediated disease in a humanized mouse immune model of multiple sclerosis.
Great. Thank you so much, everyone, for being here. As you may notice, I am not Mark Horowitz. Unfortunately, he wasn't able to travel today because he was not feeling well enough. But he does send along his gratitude and appreciation to the organizers for having us today. And so, following up on Dr. Shiro's really helpful talk, we're going to be speaking about our lab's attempt to try to uncover some of the basic mechanisms underlying EBV's relationship to MS. And we've done this through the development of a humanized immune system model of multiple sclerosis. And just because we don't have as much time to go over all of the data that's included in this project, the draft manuscript is on bioRxiv if you are interested in seeing more of the data. Okay, and so just to cover the basics of multiple sclerosis, it's a chronic neurodegenerative autoimmune disease that is characterized by the infiltration of lymphocytes into the brain as well as the spinal cord, and it's primarily mediated by CD8 cytotoxic and CD4 helper T cells. These T cells coordinate to demyelinate the CNS and promote axonal degeneration, which then leads to motor and cognitive decline for people who are affected. As far as the cause of MS, obviously we've just spoken a little bit about that, but it's not entirely well understood, and it's thought to be a combination of both genetic and environmental factors, including HLA alleles. Heritable risk accounts for about 20% of the risk for MS, which means 80% is left to environmental. And this includes a history of poor diet, obesity, smoking, as well as viral infections. And so thankfully, I don't have to rehash all of this background information. Thank you, Dr. Shirio. But just to summarize, there's been years and years of clinical and epidemiological evidence suggesting that EBV plays a role in the development of MS. And the fact that B-cell depletion therapy is so effective for many with inflammatory subtypes of the disease suggests that these therapies could be working in part through depletion of the viral reservoir in patients. The problem, as this audience is well aware, is that EBV is a human tropic virus. And so most animal models of MS utilize either primates or rodents, which constrains our ability to experimentally address the mechanism behind this relationship. The Horowitz lab has employed a twofold approach to overcome this issue, the first being to model the virus to suit the mouse. In this case, we take regular C57 black 6 mice, and we latently infect them with gamma herpes virus 68. And although not a perfect homologue for EBV, it replicates a lot of the latent infection of B cells and immunomodulatory features of EBV. What we do is we infect these mice for 5 weeks to establish a latent infection, and then we induce EAE. EAE stands for experimental autoimmune encephalomyelitis. It's the most widely used autoimmune model of multiple sclerosis. And how it works is you inject mice with myelin antigens, in this case, myelin oligodendrocyte glycoprotein. This is the immunogenic epitope, emulsified in complete Freud's adjuvant. And this combination recipe results in a T cell infiltration in primarily the spinal cord as well as the cerebellum of the brain, which leads to demyelination features and clinical deficits similar to MS. And I don't have time to get into the exact details, but what we have observed previously is that gamma herpes virus infected EAE mice show features of immunopathological damage that is more similar to MS than uninfected mice. And this includes greater brain involvement, more demyelinated lesions, and CD8 T cell involvement in the brain. So I'm going to be speaking about the second approach that we are using to overcome this problem as a part of my PhD project in Mark's lab, which is to model the mouse to suit the virus. And this, of course, calls for the use of humanized mice, which in our lab refers to mice with human immune system components, including human B cells, which are susceptible to infection with EBV directly. And so our experimental design was to use human peripheral blood mononuclear cell, or HUPBMC mice, in order to assess donors who were endogenously infected with EBV. And we have 3 blood donor groups here: individuals who developed relapsing MS who are all EBV seropositive, healthy donors who are EBV seropositive, as well as healthy donors who are EBV seronegative. We then isolate PBMCs from these blood donors and inject them into NSG, so immunodeficient mice, who then take on the PBMCs from the donors. We wait 3 weeks for the engraftment of these cells to appear at detectable levels in the blood. And then we induce EAE using a combination of 2 myelin antigens. And as far as our donors, we collected 3 to 4 female 20 to 40-year-old females for each of these groups. For individuals with MS, they had a relapsing MS diagnosis. They had never been on any treatment, had a short disease duration, and a low disability score. So we're looking for individuals who are near the earlier stages of their disease in order to capture that more inflammatory phase. Looking at this EBV status of our donors, A and B here just show serology curves for anti-EBV VCA IgG as well as EBNA1 IgG. And consistent with other reports, all of our MS patient donors had elevated antibody responses to both these antigens as compared to our healthy donor EBV positive group. And of course, our negative group had no response to either antigen. In C, we also assessed just total PBMC DNA for viral load. And what we noted was that none of these donors had significantly elevated levels of EBV DNA in their PBMCs that would suggest that they're experiencing some type of EBV-associated disorder like mono or a reactivation event. So this is looking at a history of EBV infection rather than an active EBV infection. We then transferred these PBMCs into mice and induced them with EAE. And what we did is we measured the clinical score for EAE. And that's is based on a grading system looking at the degree of paralysis that these mice experience and the severity on the right here. And for anyone interested or familiar with EAE, they seem to resemble a nod mouse with EAE. And so we can see that our mice from RRMS EBV positive donors experience a more severe disease as compared to both healthy donor groups. And if we look at disease incidence and symptom onset, we can see there's like this stepwise increase in disease incidence when we have donors who have EBV infection and that have also been diagnosed with MS. And this includes delayed onset for individuals who donated who had never been exposed to EBV. We also assessed some other risk factors that have been proposed for MS as far as our donors go. We first looked at CMV IgG and noted that there was not a substantial difference between the group cohorts as far as serology to CMV. There was a slightly significant difference between our MS and our healthy donor EBV negative groups, but not so much to explain the phenotype that we're seeing. We also made sure that none of our, especially our MS patient donors, had existing reactivity to the myelin antigen that we were using to induce this model. Anti-MOG disease can look like MS in some cases, so we confirm this is not the case. And we also did not observe differences in serum vitamin D levels between our donors, suggesting that the phenotype we're seeing is likely related to a history of EBV infection. So that raises the question of, well, what is the cause of EAE symptoms in a cross-species model of disease? So first thing we did was assess demyelination of the spinal cord. EAE symptoms, the way that they're measured clinically, is usually a reflection of damage to the spinal cord. And so on the very left here, we have this purple bar, which is NOD control mice, so regular immune competent mice with EAE, who have significant demyelination as compared to the far right bar here, NSG mice that are not engrafted, so they're immune compromised, who do not have any demyelination. And what we observe is that our MS EBV positive and healthy donor EBV positive groups also show significant demyelination of the total spinal cord area as compared to our controls. But this is not the case for healthy donor EBV negative mice who appear to be better protected from demyelination, having not been infected previously. This raises the question of, are human T cells that we're transferring actually mediating demyelination in the CNS of these mice? And on the left here, we have a control NSG that was not engrafted. On the right, we have a PBMC humanized mouse with EAE engrafted with MS patient donor cells. And the green myelin staining here is eroded in the induced sample. And we can also see the co-localization of human CD8 T cells in these areas. We can also see this in the cerebellum of these mice, where areas where there is degraded, that green myelin staining has dulled. We also see human CD8 T cell infiltration. And by flow cytometry, we know that these CD8s are granzyme B and interferon gamma producing. I don't have a ton of time to get into the immunopathological features of this model. If anyone's interested, please feel free to ask. And as well as human CD4 T cells that express a large amount of interferon gamma. So this raises the question of, well, which cell types are causing these group differences in clinical scores? So the first set we looked at was B cells, obviously. EBV is a B cell infecting disease. And immediately what we noticed, perhaps not surprisingly to people who are familiar with humanized mouse models, is that PBMC engraftment of NSGs does not lead to strong engraftment of B cells. And so in the brain and the spinal cord at endpoint, we really only had about 25 to 100 human B cells total infiltrating. And this is in comparison to the 10,000 to 1 million T cells that we're getting to these same organs. Interestingly, we did seem to observe increased CNS migration from the spleen to the brain and spinal cord in our MS group, which I think is interesting for future study. But for the most part, most of our B cells were in the spleen. And so if we were looking for EBV infected B cells, we did not want to bother looking in the CNS where it was unlikely to be able to observe infected B cells. So the next question is EBV reactivating as a result of EAE induction? And in the spleen, we did the same assessment of EBV viral genomes. And with the exception of one lone mouse, we did not observe EBV reactivation in the spleen that would otherwise explain why these groups are experiencing such different clinical outcomes. In C here, we also evaluated whether or not we could detect EBV VCA IgG. And so we would see this IgG if a memory B cell specific to VCA was transferred into the mice and reactivated as a response. So in general, we don't think, at least for VCA specific memory B cells, that they are transferring and activating in response to induction. So then we focus our attention on T cells, which we know are present in the CNS. And the first thing we saw was that in our brain and spinal cord samples, that human regulatory T cell counts are significantly suppressed in EBV positive healthy donor mice, and then again, even more so in our MS EBV positive mice. And this defect in the regulatory T cell accumulation in the CNS is highlighted even more so when you express this as a ratio of interferon gamma producing Th1 CD4 T cells compared to Tregs.
You have about 1 minute.
Oh, sure. I'm almost done. So we have a stepwise increase in the ratio of Th1 to Treg cells in all 3 organs as related to EBV and MS status. And just to note that this isn't a result of taking mice
that were differentially affected by the disease. It wasn't that one group had been sick for longer, and this is why we're seeing differences. They had pretty similar disease burden at the time of collection. And this page is just to show that we also see differences in cytotoxic CD8 T cells. And so mainly the point that I'm looking to convey to you is that pre-engraftment in our donor PBMCs, we do not see a big difference in the proportion of CD4 T cells that express FOXP3 or Tregs. After engrafting these mice, so before inducing them, we already see a deficiency in the expansion of Tregs in our EBV positive and MS EBV positive groups, and that this deficiency sort of relocalizes to the CNS following induction, suggesting that any immunological challenge to these PBMCs results in a hampered expansion of Tregs when the patient has been infected with EBV or has a diagnosis of MS. And because Tregs are deficient in other autoimmune diseases, we suggest this may be a common mechanism through which EBV could predispose to many autoimmune diseases, this general immunomodulatory effect on Tregs. And just to rehash the significance, we developed a humanized mouse model of MS where we see that EBV infection history alters the balance of effector and regulatory T cell infiltration to the CNS. And we hope to further optimize this model to assess other aspects of MS, including treatment of disease, assessment of HLA type, other genetic risk factors, and things of that such. So I'd just like to thank everyone in our lab for doing all kinds of work for this project, all of our funding sources, and all of you for listening, and I'll be happy to take any questions.
Thank you. We have time for 2 questions.
That was really beautiful. I've question. All right, I'll try one. Even though it's highly unlikely, did you do Eber staining to look at whether the B cells in the brain-spinal cord were EBV positive?
We have not. Unfortunately, we only had access to 4 MS patients so far, and so we needed to collect samples for flow cytometric analysis. We're also not entirely optimistic that we'll be able to find EBV-infected B cells in these mice, just because I'm sure Dr. Veroni will be able to explain a little more thoroughly that the accumulation of B cells in the MS brain takes time. Yeah. And because this is a relatively short-term model, we're not sure it's enough inflammation over time to accumulate those B cells.
Can I just, one really quick, have you tried the Cooperzone model rather than EAE, either
currently,
in MHV68? You are currently?
Currently, yeah, ask me in about a month. I'll have that answer for you.
Jeffrey?
So you use normal control PBMCs, and I'm wondering whether you've tried PBMCs from people with other autoimmune diseases, and perhaps the question is whether this is an MS-specific finding you're seeing, or is it just because people or is it associated with, let's say, patients with SLE or RA, which you see the same thing?
Yeah, great question. We think that because it's a more broad effect on Tregs, that likely if you induce with a different antigen for a different tissue, that you might see a similar effect. It'll be difficult to recruit all those different types of disease patients, but I think it's worth doing in the future. And especially to compare autoimmune diseases that are and are not related to EBV, like type 1 diabetes, for instance.
Thank you very much. I hand over to my co-chairman.
This presentation will be by John Harley from Cincinnati, and gathering from his title, he will talk about cross-reactivities of EBNA1 specific antibodies to autoantigens.
Well, thank you very much, and thank the organizers for inviting me to speak today. And if I can figure out how to make this go. So this is our model of how we think EBV generates both lupus and multiple sclerosis. On the bottom here, I have the transcription factors, the cofactors that EBV makes. We published EBNA2, thank goodness to Michelle West, who provided a wonderful ChIP-seq data set that we used to basically change my investigative life. And more recently, we can show that the other cofactors from EBV also do the same thing. We don't understand how they connect to these diseases, but both diseases are highly associated with the risk loci that we find doing genetics. Today, I'm going to concentrate on the top here how molecular mimicry works through the EBNA1 response to generate both disorders. And we'll see what the audience thinks of that. So this paper we published was published in January and presents the model that we think for lupus, where the idea is that lupus autoimmunity appears to go through the anti-EBNA1 heteroimmune response. And so I'll review the data for that. Within a day of that being published, this paper was published, showing that EBNA1 also is a mimic for glial CAM, and the data is fairly powerful that at least a proportion of multiple sclerosis patients operate by this sort of mechanism also. And so if you look at the SMBB prime, which is the spliceosome, and the EBNA1 sequences, glial CAM and the SMBB prime are almost identical sites in the EBNA1 sequence. Very, very curious. We'll come back to that. Traditionally, we have used the Mario Gleason peptide synthesis strategy to dissect the structure, structure of the antibody response in lupus. The 4 panels that I show here on the bottom show a patient over about 2 years developing the anti-SMBB prime response. 120 amino acids overlapping, 8 MERS overlapping by 8. The secret to this, making this a success, is that the density of peptides is extremely high, making homonymous bivalency operational and providing, meaning that if we have an affinity, an intrinsic affinity of about 10 to the 4th, that the homonymous bivalency can raise that to 10 to the 8th, and we can get a good response for even a partial epitope. Our work shows that in SMBB prime, this proline rich is first, another proline rich is second, a third proline rich is third and fourth, and then it distributes all over the molecule, and most patients develop exactly the same fine specificities. We've also done this in the anti-Rho response. Here's the overlapping peptides from EBNA1. And despite there not being any sequence similarity between EBNA1 and the Rho 60KD autoantigen, there is immunologic cross-reactivity that is convincing that we published now almost 20 years ago. And we can generate from the EBNA1 sequence, we can generate very complex autoantibodies in animals who develop findings that are reminiscent of human lupus. Then this animal has leukopenia, rising creatinine, and thrombocytopenia. So this is the molecular mimicry model. You develop anti-EBNA1 antibodies, it cross-reacts with self, the immune response takes off, you have epitope spreading, it doesn't, after it gets started, you don't need the EBNA1 any longer, and we published this before with the intention that it was for lupus. If that brought us to do the epidemiology, this is one of our paper in pediatrics showing a 50-fold odds ratio, and these data now, there are about 20 studies showing that Epstein-Barr virus infection is concentrated in lupus patients. So we have now 4 different, maybe 5 if you include double-stranded DNA, epitope autoimmune systems that look like they are coming out of the EBNA1 immune response. The EBNA1 immune response is very strange, but it leads us to the hypothesis that it looks like the EBNA1 antibody response is what's required to develop these autoimmune findings. And if that's true, then in the normal population, only somewhere between 70 and 90% of individuals develop anti-EBNA1 antibodies. If it's true, you would predict that individuals with these diseases, if anti-EBNA1 is required, would have a higher frequency. So we tested that hypothesis in 2 studies, and this is in lupus, and in both studies, we have a near complete EBNA1 presence in lupus, with an attributable fraction of 90%. And so I don't have time to go over this in detail, but if anybody has more data that has both VCA, anti-VCA, and anti-EBNA1 data in lupus patients, then we would love to see it. If you look at the anti-EBNA1 threshold for positivity in MS patients, we've been able to find 14 studies, all of which, except for one maybe, that show the relative risk of about fourfold for anti-EBNA1 positivity. But that's really not the question. The question is, what is it in those people who are EBV infected and convincingly affected? Do we have the same story happening? And we've been able to find data from 3 studies, and hopefully some of you in the audience who have MS data can help us expand this, that MS does the same thing, where the among just those EBV-infected people and only infected controls, the anti-EBNA1 response is higher than you would expect. But that only takes care of about 30% of the risk, leaving 70 or more % of the relative risk from going from a normal person to a person with one of these 2 diseases having, and so you have all of this area to work with. Smoking, being female, vitamin D. I noticed in the last study that almost all the patients they worked on had a vitamin D level that was deficient. And everybody in British Columbia probably needs vitamin D. And the peptide. So we have this system. Well, so what's going on here? You are interested in what's happening with this anti-EBNA1 system to set things up so that you have the cross-reaction. I will remind you that the anti-EBNA1 response is late. It's the last immune antibody to develop in EBV infection. There are fascinating studies on the T cell side that shows that the GA repeat in the center of the molecule inhibits the proteasome and prevents EBV peptides from being put on the surface. There is guanine stacking that inhibits microRNA processing, and the nuclear localization of EBNA1 is thought to also contribute. It means that there's virtually no surface peptides on the EBV-infected B cell. There's a very interesting study that Alan Rickinson presented last year showing that if you break the EBNA1 up, you can find strong responses on the T cell side from peptides, but not from the whole molecule in an infected B cell. And so if you look at the molecule, then lupus has these 2 on the N-terminal side and 2 on the C-terminal side. There's no immune mimicry happening in the GA repeat, which covers what, 295 amino acids of only glycine and alanine that is thought to inhibit antigen presentation. And multiple sclerosis has 4, and they're all in about 35 amino acids, this tiny little section. One of them I showed you earlier. And then there's one for rheumatoid arthritis, and there's one for, of all diseases, preeclampsia has a molecular mimic with EBNA1. And so this is the molecule with the amino acid sequence. Here's the N-terminal side. All of these are presented. Lupus has SMD, Rho, C1q has been done in the last couple of years, a very interesting autoantigen in lupus, and SMBB prime. MS has glial cam, the alpha B crystalline, myelin basic protein, and anoctamin 2. And rheumatoid arthritis has citrullinated beta fibrin, that's a cross reaction, and preeclampsia has one paper that GPR50 looks like it was discovered in a phage display library effort that disintegrated after that first paper was studied, and the investigators have not followed it up. There's a lot more to talk about. This is the paper about GPR50. I would submit that there are, a few hundred, maybe 1,000 idiopathic human diseases that we don't know where they come from. There's at least 150 inflammatory disorders. EBNA1 is such an incredibly strange antigen and it stimulates the immune system in such an odd way. The initial response, it looks like you need degradation of the infection in order to represent EBNA1 to start an immune response. The anti-IgM that starts looks like it is a B cell, T cell independent response to begin. And so there's a lot of immunology to be done there. Our conclusions are the data are consistent with anti-EBNA1 heteroimmune response initiating autoimmunity for the great majority of those afflicted with lupus and multiple sclerosis. And the anti-EBNA1 heteroimmune response may serve as a foundation for autoantibody generation through molecular mimicry for other diseases. And so I need this is the virus that we all love to work on. And my colleagues who've made a difference to this work throughout my career, and the people, the funders who by accident have decided to fund some of this work. And if you have, and if anybody in the audience has data that would contribute to this, I'd appreciate being connected.
Yeah, very nice. Thank you. Questions? Maybe I can start. So what always puzzled me a little bit with this cross-reactivities at the antibody level is that in MS, the B-cell depleting therapies that work actually quite well, right, at least in the relapsing remitting phase, usually leave these antibody levels untouched because the plasma cells that produce the antibodies are not depleted during the therapies. So how do you explain that? So these are then different mechanisms by which B cells...
So the B cell therapies don't work as well in lupus.
Mm-hmm.
And the two diseases immunologically are dramatically different. Mm-hmm. You need a tiny amount, nanograms of antibody, probably in the wrong place in the brain to create the lesions that we see in multiple sclerosis. And lupus is this overwhelming systemic response with huge amounts of antibodies. I've measured 80 milligrams per mil of specific antibody against a single antigen. That's 5 times more than anybody in the room is making against the entire universe of things that you need to defend yourself against with your antibody response. So there's an enormous amount of difference there. I think it may be that the B cells, instead of plasma cells, are more important in multiple sclerosis and in the brain lesion itself. And in lupus, there's more, the B cells are more mature and more established.
Mm-hmm.
I've given myself anti-B cell therapy to lupus patients, and it does not touch the autoantibody response.
Mm-hmm. Thank you.
Yes. So the two peptide regions of EBNA1 that you are focusing on, have very intriguing properties, one of which is that they're rich in arginines and maybe post-translational modifications. The other might be RNA binding or something along those lines. Have you considered whether that might be a feature that's unusual about EBNA1 peptides in this particular case?
Well, you're the world's expert on EBNA1 structure, so maybe we can share some idea-generating discussions. I think if you take the GAGA repeat in the center and put that on any other antigen, the data are that it'll inhibit peptide presentation on the surface. And so it looks like the EBNA1 needs to be broken up first and represented in order to generate the immune response that's characteristic. I haven't seen, and if somebody in the audience does this, people that do more T cell work, I haven't seen the T cell response. The classic antibody response has EBNA1 happening late. And I wonder whether that's also the time course for the T cell response.
I have some comments to make on that. This MS-related EBNA1 epitope sequence is relatively non-structured. We did detailed mapping of autoantibodies, and we have published that already in 2010, and in a recent paper in Frontiers Immunology last November, where we proposed this hypothesis of how EBNA1 might trigger autoimmune responses.
Mm-hmm.
And so we cannot go into details too much, but certainly I have additional data for you that
awesome.
might be helpful. One last question. Okay, you do that, Chairman.
So in mononucleosis, we have this huge heterophile response, heterophile antibody response. And I'm not sure that we still know what this heterophile response is really directed against. Is there any idea, I understand that the Ebner response evolves later and it takes a while, and the heterophile response may actually be, is probably earlier. That said, though, do you think there is any link between these heterophile responses and some of these responses that you're seeing against Ebner-1 and potentially other antigens of the virus that persist, perhaps, in those who develop these autoimmune conditions?
So the heterophile response is generally an IgM response, right? And it's early after infection. And so I was able to find 5 cases of infectious mononucleosis that morphed into lupus, and nothing that goes the other direction when I did that a few years ago. But I think, you know, there's usually, like MS and lupus, I think there's years between the time that the initial infection happens and the disease onset. And so there is continual stimulation of the immune system that's happening. And I think that's critical in order to develop the cross-reactive antibodies in order to make the molecular mimicry mechanism operate. And it's a lottery. You know, you're throwing a dart at the wall every day with a new stimulus. And then you have a low proportion. You know, but in both diseases, the twin concordance is not very high, for example.
Okay, thank you. I think for the sake of time, we have to move on. Thank you again. The last presentation will be by Caterina Veroni from Rome, and she will probably talk about cells that are EBV positive and in lesions in the brain.
So thank you for the introduction, and thanks to the organizers of this meeting for giving me the opportunity to present and share with you our data focusing on the detection of EBV DNA and RNA in biological samples from multiple sclerosis patients. The model of multiple sclerosis on which our research group is working for more than 15 years could be defined as a EBV-centric model in which the virus is the driver of the immunopathological response leading to central nervous system inflammation and tissue damage in MS. This disease model implies that a persistent EBV infection in the central nervous system stimulates a cytotoxic T-cell response that aims at eliminating the infected B cells in the brain and leading to the bystander tissue damage. The 2 main findings support this model. On one hand, the presence of EBV-infected B cells in the MS brain, and on the other hand, the presence of EBV-specific T cells in the MS brain. And most of the evidence supporting the presence of EBV in the multiple sclerosis brain was provided using either in situ hybridization and immunohistochemical techniques. I was mainly involved in verifying the presence of EBV in the MS brain using PCR-based techniques to detect EBV nucleic acids in the laser cut immune infiltrates from post-mortem MS brain tissue. And I've also extended this study on cerebrospinal fluid cells from therapy-naive multiple sclerosis patients and to peripheral blood samples from both MS patients and healthy donors. To detect EBV RNA, we have used a real-time PCR method with a preamplification step that allowed to increase the accuracy and sensitivity in detecting EBV transcripts in samples with a very low amount of RNA, such as cerebrospinal fluid cells and laser capture microdissected samples. In this study, we have analyzed the expression of 7 EBV genes, 5 related to EBV latency and 2 related to EBV reactivation. to detect EBV DNA in cerebrospinal fluid and peripheral blood cells. We have used droplet digital PCR, and the BAM-HIW region that is repeated about tenfold for each EBV genome was selected as target region to maximize the sensitivity of our assay. In the first part of this presentation, I would like to show you already published data obtained in immune infiltrates isolated from the MS brain. To this aim, MS tissue blocks showing good RNA quality were extensively and accurately screened for the presence of large immune infiltrates, and finally the B-cell enriched immune infiltrates selected were laser capture microdissected, and the EBV nucleic acids analysis was performed. Here you can see the results showing that EBV RNA was detected in 9 out of the 11 MS cases analyzed, and in a substantial number of the immune infiltrates analyzed, about 30%, most frequently in the meningeal infiltrates compared to white matter perivascular infiltrates, and latency transcripts were more frequently detected than lytic transcripts. In very preliminary experiments, we have also analyzed the expression of EBV DNA in a few immune infiltrates isolated from the MS brain, and using different set of primer and probes targeting EBV DNA polymerase gene, and we have found DNA in 3 out of the 9 MS patients analyzed, and only in the meningeal infiltrates, but not in the white matter perivascular infiltrates. So these data confirm the presence of EBV in the MS brain, corroborating the results obtained in situ hybridization, immunohistochemicals, and also DNA studies performed by our research group, but also by other independent research groups. Now I'm going to show you unpublished results obtained using the same PCR techniques in samples from cerebrospinal fluid samples and peripheral blood samples from multiple sclerosis patients. This table summarizes our cohort and the demographic, clinical, and radiological data of the MS patients and healthy donors enrolled for this study in 3 different Italian multiple sclerosis centers. In the cerebrospinal fluid cells, EBV RNA was detected in a small fraction of samples, about the 8%, but it's important to highlight that all the samples with detectable EBV RNA contained 1 or more transcripts related to EBV dysregulation. The very low amount of RNA extracted from CSF cells, together with the fact that B cells are a minor component of CSF, may represent major limitations and may explain the absence of signal in most of the samples analyzed. We have also the opportunity to analyze EBV DNA in 3 CSF samples that contained enough cells to perform both DNA and RNA analysis, and interestingly, we found that 2 out of the 3 samples analyzed had detectable EBV DNA levels, and the viral load in the cerebrospinal fluid cells was about tenfold higher than in paired PBMC samples. Concerning peripheral blood, we didn't find any differences in the frequency and in the levels of EBV DNA in MS patients compared to controls, and this is consistent with previously published studies. And despite no significant differences were found Also, in the frequency of EBV RNA in MS patients compared to LT donors, it's important to note that about the 80% of the EBV of PBMC samples from healthy donors with detectable EBV RNA levels contained EBER1 only, and only 2 samples contained also LMP1. While conversely, about the 80% of the EBV RNA positive samples from MS patients contained genes related to EBV dysregulation, and only the 20% contained EBER1 only. Here you can see that EBER1 and LMP1 were the transcripts more frequently detected, while other transcripts related to latency, such as LMP2A, IBNA1, or IBNA3A, and the 2 genes associated with EBV reactivation were detected less frequently, but only in MS patients and never in the healthy controls. So overall, the frequency of the transcripts associated to EBV dysregulation were significantly higher in MS patients compared to control subjects. So in conclusion, in the small fraction of cerebrospinal fluid cell samples with detectable EBV RNA, from multiple sclerosis patients, the transcripts, EBV transcripts detected were always associated with viral dysregulation. In the peripheral blood samples, the transcripts associated with EBV dysregulation were more frequently detected compared to in MS patients compared to LT donors. And this data confirmed that MS patients may have a problem with the control of EBV infection. The central nervous system is confirmed as the site where EBV is detected most consistently, and where could be the target of the immunopathological response leading to central nervous system inflammation and tissue damage in multiple sclerosis. Before I conclude, I would like to mention another potentially attractive site to study EBV infection in MS, despite not easy to study due to ethical reasons, but this is the CNS draining lymphoid tissue, and this is suggested by a study from our group demonstrating the presence of an accumulation of infected B cells in the cervical lymph node of a patient of MS patients, but also by other study demonstrating the trafficking of B cell clones between the deep cervical lymph node and the central nervous system. And finally, an interesting ultrasound-based study demonstrating that MS patients display cervical lymph node with higher diameter compared to controls. Finally, I would like to thank my research group directed by Francesca Loisi and Barbara Serafini, Corrado Fagnani for the statistics, and all the clinicians and the biologists who have recruited MS patients and analyzed and collected the biological samples, the UK Tissue Bank at the Imperial College of London, of course, most importantly, all the donors who contributed to the study. Thank you very much.
Thank you. We have time for 1 or 2 questions. Maybe I can start. So do you think this infiltration of EBV positive cells into the brain is due to an inflammatory setting in this organ and would not occur in other inflammatory settings in other organs? Or did you ever compare?
We have analyzed the expression, this infiltration in the brain, in the postmortem MS brain tissue. We have analyzed the expression, the presence of these cells also in other samples from control patients in the brain, and I think this accumulation in the brain, I think that is specific for MS patients.
Mm-hmm. Would you see it also at other sites?
In my experience, not other different from the peripheral blood or the central of the CSF and the cervical deep cervical lymph node, as I shown. But I think that in MS, also the fact that I didn't find any significant differences in the EBV DNA load, and that EBV RNA was detected in a very small fraction of samples, suggests that in MS the EBV infection is really compartmentalized and is associated to brain inflammation.
To the cellularness. Okay, if there are no more questions, then thank you. There's one more. I think there's one more. Last question.
Hi, great talk. I was just curious, you observed that the EBV levels in PBMCs aren't different between healthy donors and MS patients. Did you look at all in plasma, or do you know if anyone has looked in plasma to do the same comparison?
No, I really don't understand. If you can repeat, please.
So I was just wondering if you compared, or if you know if anyone has compared EBV levels in plasma between healthy donors and MS
I don't know.
patients.
We didn't do this, and I don't remember any paper on this.
Okay,
Thank you very much.
So thank you. With that, I would like to close the session, and we have now lunch. And please, everybody, be back at 2 o'clock for the next session. And I thank all the speakers one more time.
Well, actually, CD4 German. I mean, some CD8 activities are very strong, right?
You need to move something on here? Yeah, I wanted to see if, just really quickly, if it would let you do like presenter mode. Do you know if this is possible? Yeah, I don't need them, but I always feel a little bit better if they're up there. I think it does let you. Let me just see really quickly. Sorry, I'll be just a second. Then I'm going to come back and... Oh, it's all in Italian. That's great. Let's see. Yeah, so it does. Okay. So you can go ahead.
I don't know how to do it. I don't know where. Yeah. I'll let them do it. Sorry. So I'm in the first. Oops.
Allora. Please.
No, West Siena talk.
Okay. What's your name? West?
Yeah.
It's all right.
Yeah, that looks fine. Good.
Okay.
Or do you have an email that's already up? Send it. Or I can just log in. If that's easier.
So the crystal structure is only from we made additional sites outside of the
Faccio uscire. If you want, login.
Thank you. Then I have a question. How do you pull up the presenter view? Do they have that? You know presenter view? Maybe I can show you. Italian. This one.
Yeah. No, it's this.
We met in detail with MS Sierra, where everybody was on that level.
It's here if I just click it.
Yeah, this is the hypothesis part of this. So how is the role of FL1? This is like previous clinical days, so okay, well, I apologize. No, there's no problem. If you have ideas or you have to know more.
Of course, well, I think this is I think that its immune response is centered on EBNA1.
Ah,
It's really.
okay. Do you mind if I leave it like that?
No.
Yeah. Yeah. Do you think it'll? Yeah, that's fine. For the next session, will that be confusing for people?
You can
leave.
write and
DNA capturing that's
Huge system.
So if I'm here, how do I do it? If I start here, oh, so here, yeah, inverted. Okay. So let me just practice. I changed it, right? Before it was like this. Duplicate this. So we'll take a second. There you go. So it's okay if I change it when I do it myself, yeah? Oh. I'll just leave it. I don't need to use it. It's going to be too complicated, I think. So,
yeah, connect. No. Do the screen. Okay.
yeah, because I wanted to do it like this, but it's difficult to do here. Yeah, I'm just going to move it over. I think it's fine. I'm not going to try to plug it in. I'm just going to download it now. So I think I'm done. Thank you. I'm sorry.
That was the other side that you can
I just have to send it to myself.
The keyboard is different.
I see it.
Yeah.
This is shift?
Yeah.
Okay. Yeah, so this
Different Italian keyboard to escape.
which is caps or shift? It's like capital?
This.
Oh, so. Enter?
Oh.
Okay. Thanks.
It's sort of like more circumstantial evidence.
Well, to see it was, I would say, more about
you can move the presentation with the cable with
Yeah,
USB pen to the
I'm just emailing it to myself.
Bluetooth.
I'm just emailing it
Okay.
to this email.
Okay.
So it's just taking a second.
Any way you want.
Give me a moment. It's just slow. Because it doesn't have USB.
USB-C, no.
Yeah, there's no USB-C. You know what I mean?
If you want the USB pen. No. Can you invite mail?
Yeah, you can send it to my email.
Or
Oh, got it.
so.
No. Give me a second.
Your file in this section.
Sure. It's third session? Two? How do you rename it?
Rename with your
I just want to make sure everything was okay. Sometimes the Mac to the.
Oh, yes.
The format will. Yeah, I have to go fix some things. But if you want to go first, that's fine. So I have bosses and they ruin them. Sometimes it'll do that.
Do you want the presentation with the...
I just find this way. I think for that you have to extend it, but I know how to do it and get figured it out. So if you want to do it this way, then you have to go extend it, and then it'll pull it out. Probably want to enter. See, now it's still this one. I wouldn't worry about it. I'm fine just doing it the regular way. But I do need to go ship the boxes.
So you need to fix it.
Yeah, but you can move your stuff over first. Okay. We have time.
Which is the button?
Yep.
This.
Oh, let's go.
Sorry.
Can we just look at this one?
Success slide.
This is correct. So do you want to put my name?
Okay.
Okay. Thank you.
Yeah, I wonder if it's even worth it, because I feel like I can try it, but I think it might not work well. So this, presentation. Which one was it?
Do you want here this screen and
Yeah.
here the presentation?
Yeah, it just wasn't. Where was that thing before? Can you show me how to do it? So 60.
You duplicate the screen when you
Oh, so then when you go to present. I think it has to be. I think it has to be extended, not extended.
Extended, but if you extend it, here is a clean screen.
No, I'll show you.
Okay.
So I'll show you. So if you extend it, give it a second to load cycles. Okay. So now it's extended. Now you go here. And then which one was it that you said? It's here. There.
Okay.
So I might just leave
This
it.
way.
I'll leave it like this. And then let me just see what happens if somebody opens one. So when they open it themselves, they'll have to go through that. Each person. I don't think it's the default.
Okay.
Do you see the problem? So if somebody opens theirs, it's not automatic, you see?
No.
So they might have
Here?
Yeah, but for anybody else, yeah.
Because it's embedded.
Yeah. So should I change it back to what it was before? Like,
If you when open the presentation.
yeah, because other people are going before me. I'm in the middle.
Yeah.
So, like,
Yeah,
I'm
yeah,
here.
yeah.
These people might get confused.
I
You understand?
say the technique that when it's your turn, he don't move the mouse, remote mouse, and you can control it.
Oh, does he normally do it? Or does he normally control it?
For the other, the technique move remote move the control. Okay.
To the next slide?
No, to open the presentation. To
Okay.
For your presentation, I say that you can take hold. Okay.
Okay, I think I don't want to make it too complicated. I don't need the notes. I think just leave it how it is, the same as everybody. I don't need this. So no.
Okay.
So I want it the same as everyone, just the duplication, yeah. Yeah. So I think for everybody else, if you just do, yeah, there you go. Or actually, can I see that? So,
In this way.
yeah.
Sorry.
How did you change that?
In this way.
Okay, yeah, that's fine with me. That's fine.
Okay.
Yeah, sorry. Sometimes it's easier. I don't want to do too much, you know? Thank you. Close it.
Ah, sono lì, perfetto.
Allora. Paola, io decido che faccio la precisione.
C'è da perché
Se troviamo un professore va bene, allora mettiamo un video. Se non troviamo nessuno.
Perché fate i così esatto.
essere che sia caduto qui.
Vabbè, ma secondo me se chiediamo a Roberto qualche cosa.
Allora io direi che questi qua, vabbè, tanto forse a voi sapete caricare avete tutto.
Noi ce l'abbiamo.
Io sto
Buongiorno. Buongiorno, si possono indicare? Sì, certo, adesso finiamo di caricare una cosa 2 secondi.
Scusa.
Non si preoccupi.
Stavo finendo di fare una cosa. Finisco di fare una cosa. Hai finito?
Facciamo. Fai subito.
Qua? Sì, sì. Ma io sarei più basta, ma che mi frega? Sì, alle 2:30, però ha detto un pochino e basta. Ma infatti c'è allora. Dove la danno la verità? Sì. Quindi c'è da ricominciare. Che mi danno 2 euro? Ah! Aspetta la parola. No, chi era Lorenzo? No, era adesso, è più grande. L'ha detto. Mammona, mi chiedeva dove stava la chiesa. No, io però ti dico che forse se te ci dai il senso.
So you are involved? Okay, I understand. Not this time. Where are we? I got student in from. You're 29 to 30. And your time to be Florence? No, this is. But that time we are going to Florence. Today you have time to go around? No, not of it. I know the one going close to the station. Okay. Tomorrow morning, go to the morning? Yes, tomorrow, around 12 o'clock. Okay. Are you flying from Florence? And where? Which state is that? No. Paris. Paris, France, and. Paris. Yeah. And COVID test was okay. Thank you very much. Oh, good. You have a certificate? Yeah. Oh, yes, thank you. Good stuff. And they also build a home for Japanese folk. Wow. Very nice. Oh, wow. There is also Japanese. Yeah. Yeah, I'm not sure to the Japanese, when I am entering, I'm sure. Yeah. They don't tell me, so they accept us. They accept us. Yes. But the home is for Japanese. And then, they took my passport and checked. Thank you. Okay, no, it's difficult in Japanese.
Ho capito, si sbagli qui, si può sbagliare qui. Hanno cambiato. Sandro, siamo andati lassù, se non è più. Ah, vabbè. No, è impazzita, gliel'avete cancellata? Perché poteva impazzire. La mandiamo su tutti i social. Se il cinese e gli hanno detto di cancellarla e non la cancella è morto. Congo com'è? No. Sì. È già tanto che, vero, voglio mettere posto già nei polsi. Cosa dicendo? Non l'avete dato con voi? A medicina. Occupato.
Siamo niente.
Mi
pare mangiato, mi pare che non siamo niente.
Posto di pozzi ci sono io.
Io ti pizzicoti.
Va bene, giù.
Te quanto sei stata, hai dovuto subire in vita tua? Che un tempo le donne non venivano rispettate sul luogo del lavoro. Oggi siamo tutti così. Ma lei ha dovuto subire.
Lui.
Quanto hai patito?
Tanto.
Ma non dire cazzate.
Ah no. Mi faceva piangere, un giorno sì, 8 no.
Piangeva, c'aveva il fidanzato.
Un giorno sì no, veramente le fattacce. Me le facevi te.
Che fai? Ops, vedi? Beh, lì c'è questa interruzione del passo.
No,
però-
è fatta male la scala.
Sì, perché c'è un gradino
Perché ci sono degli architetti che sono intervenuti. Io
Sì.
quando questa era una sala di pediatria
Quando ci mettono in mano gli architetti è un casino.
è un casino, non sanno prendere misure.
Grazie perché a Binder se no
Leo, aspetti un attimo. Che bravi. Resistete.
Ormai è più facile finire che non tornare.
Lei dove è di Genova?
No, è più in Veneto.
No, dico,
Non si sentiva, è un po'...
non è proprio...
Ma dopo proprio un po' un mix di cose.
Iris. Greetings. La medicina.
No, yes. I met him last
He's
night at dinner.
professor of microbiology.
Yes,
here at
yes, we
Università.
sat next to each other at dinner last night.
Ok, so,
Thank you.
Giannino. Mi raccomando, se fanno troppe, insomma,
No,
teniamo tempo.
let's be mean.
Not a problem.
No, but okay, same time, there is some discussion.
Yeah.
So let's recommend synthetic talks in space for discussion. I was looking at the abstracts, and
it seems to me the first four are more. I don't know.
You want me to do the first four and you do the second four? Is that what you're
Yeah,
saying?
yeah, that's
That's
probably
good.
that.
And you can introduce Jeff if you want.
Well, I actually don't know him. Probably you're better suited for introduction. Yes. So this is also the last.
Yeah, last session.
Last session. So you went to Johns Hopkins in
And never
70
left.
and never left. Well,
I had no idea it was going to be a lifetime sentence.
you never do these things. I enrolled in the medical school here in Siena and I was sure, to become a psychiatrist. And it was 76, and my professor of microbiology, she was way ahead, and she was working with phage lambda plasmids, and genetic engineering was being born there. So I got in and never left. I've been all over the place, actually, because I've been a lot in the US and Belgium, several places, and also in Italy. I've been in Padua, Verona, several universities. But at the end, I came back here, and last time I was here, I was in an oral exam as a student in pediatrics.
Interesting.
I was sitting there.
Interesting.
And so they renewed it a bit, and then this is law, political sciences. So now everything is at the hospital. But yes,
So
still.
Jeff, let me introduce you to Gianni.
Hi.
Jeff.
Gianni. Hi. Nice to meet you. And I think, Richie, you should introduce him. So we're getting.
So there's no timer here, it's just you're going to let me know.
Yeah.
It looks like there's a timer right in front of you. I don't know if that is or is not. Is that a timer?
It's not activated at the moment. Maybe they're doing
It looks like a timer. No.
it. Anyway, just let me know if I get close.
Let's say, at 15,
Yeah,
we
timed
let you
it.
know so that they
When I
will
timed
really-
it, it was like 13 and a half minutes.
I was going to say, I've heard Jeff talk many times. He doesn't run over. He's got postdocs where it's been an issue.
And that postdoc knows that you also ran the meeting in Geneva. We practiced his talk. His was under time, and I said, if they tell you, if it goes, if it's getting longer, just go to the conclusions and it'll be fine. It was, we remember. And I was really embarrassed.
No, your talks are always quite wonderful and always show very serious preparation. You always
You
have,
need time to be
thoughtful,
synthetic.
I just, when you're the first person and you run over, then everybody else suffers. It's not fair.
Yeah.
This is not fair.
But certainly, there is an old saying where you say, I'm sorry that I'm writing you a long letter. I didn't have time to write you a short one, So that's the same goes with talks.
Right.
Because besides, there is only so much that you can convey, and so much attention that you can hope to induce in the audience. So, but let me tell you.
Why don't you open up and make sure you can open it up? And
They're doing it.
They're
As soon as
doing
you- They must have heard you.
Okay.
They just loaded.
Cool.
They just loaded it. It's not in presentation.
I guess we'll get there in a second.
Come va, va bene? And so you went to Maryland from where?
I was an undergraduate at Harvard. I grew up in Detroit, Michigan.
Certainly a prestigious medical school, Johns Hopkins. Once you're there, you don't want to leave it. But
I've had a lot of fun. I've enjoyed what I've done. Certainly have no regrets. Looks good. Shall I go ahead? I want to welcome people to the last session. And it's my pleasure to introduce Jeff Cohen from the NIH, who's going to talk to us about strategies to prevent EBV-associated diseases.
Okay, thank you all for coming, and I appreciate the kind invitation to speak here. So as Dr. Ambiker said, I'll be talking about the strategies to prevent EBV-associated diseases. And as we all know, there are a number of reasons why we might want to prevent EBV infection, mononucleosis, and of course, EBV is associated with a large number of different malignancies. And all told, there are 200,000 cases of EBV-associated cancer and 140,000 deaths related to EBV cancers each year worldwide. And of course, as we heard this morning, EBV is strongly associated with multiple sclerosis, another reason for developing an effective EBV vaccine. Of targets for a vaccine, the usual suspects, of course, are the major viral glycoproteins on the surface of the virus. And GP350, as you all know, is important for attachment of the virus to cells. And then for B cell entry, there's a complex of 4 glycoproteins, GH, GL, GP42, and GB, which form the fusion apparatus, allowing the virus to fuse to B cells. For epithelial cells, there's a different protein that's thought to be important for attachment. And the fusion apparatus for epithelial cells is GH, GB. So 3 different glycoproteins. So most of the work historically is focused on a vaccine using GP350. And really the first study that showed that a GP350 vaccine might be effective in an animal model was actually done by Tony Epstein. And you can see on the bottom, this was done 21 years after he discovered the virus. And what he found was the cotton-top tamarins, which are now an endangered species, when he vaccinated those animals with membrane antigen, which is GP350, purified from B95 8-cell membranes, and then challenged the animals with EBV, he could show that those animals were protected from lymphoma. So in the first set of experiments, he vaccinated 4 animals, and you can see the animal that the highest levels of antibody to GP350 had the highest ELISA titers, highest neutralizing titers, had no lesions, had no tumor after challenge. But animals that had lower levels of antibody developed multiple tumors. He then did a second confirmatory study where he vaccinated animals 17 times with this membrane antigen, developed very strong, high levels of neutralizing antibody. And those animals, when challenged, had very transient lesions that resolved spontaneously, whereas the control group that were unvaccinated all had multiple tumors. So again, the first evidence that a vaccine might be effective. Now we move forward to the largest clinical trial to date of an EBV vaccine. And you can see that 22 years elapsed between that tamarin trial and the phase 2 clinical trial. Here, individuals, students in Belgium, received either a vaccine
or placebo. And this was given in an adjuvant at 0, 1, and 5 months. And you can see the vaccinated students had a 78% reduction in mononucleosis compared to those that got the placebo. However, the vaccinated students still became infected. So this vaccine did not prevent infection, but it did prevent or reduce the rate of infectious mononucleosis. And it's unclear, but the company did not pursue this vaccine further, potentially because it did not prevent infection. So we decided to see if we could try to make a better mousetrap, a better GP350 vaccine. And instead of using a soluble GP350 as was used in that clinical trial, we decided to use GP350 in a nanoparticle. And we know that hepatitis B vaccine and human papillomavirus vaccines are highly effective and produce very long-term immunity. And these vaccines are multivalent, they're symmetrical nanoparticles, and they have a repetitive array of antigens. And we decided to try to convert GP350 into a similar type of nanoparticle. So we use Helicobacter ferritin, and we have GP350 fused to each monomer of ferritin, and it self-assembles into a 24-mer. And when we made this protein or particle and looked at by EM, you can see the ferritin core in the center, and you can see GP350 radiating from the center of the core. When we vaccinated mice with either the soluble GP350, which was virtually identical to what was used in the clinical trial, or the GP350 ferritin that we produced, you can see the mice have much higher neutralizing titers if they got the ferritin construct than the soluble GP350 construct. And these mice, we found, had antibodies that persisted for nearly a year after vaccination. So it gave both high levels of antibody and persistent levels of antibody, which are very important for a EBV vaccine. Now, why might that be the case? Well, when we did cryo-EM, or I should say we collaborated with the late Michael Rossman and did cryo-EM, we found that the receptor binding site, the CR2 binding site on this particle, which is shown here in the pink ovals, is spaced about 50 to 100 angstroms, which is the ideal distance to cross-link the B-cell receptor. So this gives one very high levels of antibody, and it also focuses the immune response on what's really on the surface. So it focuses the immune response to neutralizing CR2 binding site antibodies. So with that in mind, we have started a clinical trial of our EBV GP350 ferritin nanoparticle at the NIH. It just really opened, we just recruited our first patients last month. And we're doing a study of 20 seronegative and 20 seropositive volunteers. These are young individuals. They'll be receiving 3 doses of the ferritin vaccine. And we chose the Matrix M adjuvant, which is a saponin-based vaccine based on comparisons with some other adjuvants. And what you can see here, again, it was a number of years, 15 years between that phase 2 trial that I just showed and the next major clinical trials, which is both NIH and I'll mention the Moderna trial as well. Our primary endpoints in a phase 1 study, of course, is safety, 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 but we also will be looking, of course, at neutralizing antibodies to EBV. And secondary endpoints are GP350 antibody titers and CD4 T-cell responses to GP350. So what about other vaccines for EBV? What other targets might be important? So I focused on GP350, but there are other glycoproteins. And to try to figure this out, Wei Bu in my laboratory made cell lines expressing each of these glycoproteins and then incubated human plasma. He actually used IVIG, which is plasma pooled from over 1,000 donors, so we get a very representative response. Incubated plasma with each of these individual glycoproteins, and then saw what was the contribution of neutralizing antibody. So what this slide shows that 50 to 60% of the neutralizing antibody in human plasma is directed against GP350. And if you look at GHGL GP42, that makes up nearly all, if not all, of the remaining neutralizing antibody for B cells, indicating these would be important targets for a vaccine as well. For epithelial cells, you can see that 75 to 80% of the neutralizing antibody is directed against GHGL. So with this in mind, we think a vaccine targeting GP350 and GHGL GP42 should induce antibodies that neutralize infection of both B cells and epithelial cells and might prevent EBV infection and or disease. So we went ahead and made GHGL GP42 nanoparticles with ferritin. We vaccinated monkeys this time, and you can see that the ferritin nanoparticles outperform the soluble vaccine, both in terms of neutralizing titers in B cells and epithelial cells. And to try to find out whether those animals or whether animals will be protected from a challenge, we vaccinated mice with a combined GP350 ferritin plus GHGL GP42 ferritin vaccine, collected serum from the animals, purified the IgG, and then immunoglobulin from the vaccinated animals or animals that were unvaccinated, naive, were given intraperitoneally to humanized mice that were subsequently challenged. And what we found is that the animals that got the immunoglobulin from naive mice, when challenged, all became viremic, whereas animals that got the immunoglobulin from the vaccinated mice, only one was transiently viremic. More importantly, when we looked at these animals and dissected them, you can see that 50% of the animals that got the immunoglobulin from naive mice developed EBV lymphomas. You can see here T cells infiltrating the liver, CD20 positive, EBR positive, whereas none of the animals that got the immunoglobulin from the vaccinated mice had lymphomas. So to summarize this portion of the talk, combined GP350 ferritin and GHGL GP42 ferritin nanoparticle vaccines may provide both a quantitative improvement over the soluble GP350 as well as a qualitative improvement because we've added different antigens. A vaccine may or may not induce sterilizing immunity, and if it doesn't induce sterilizing immunity, but it reduces infection of B cells and epithelial cells, it might result in a lower viral load, and it might prevent mononucleosis. And we heard this morning that mono is a risk factor for multiple sclerosis, and we know it's a risk factor for Hodgkin's lymphoma. So we may not get sterilizing immunity. As I mentioned, a trial is underway at the clinical center with the GP350 ferritin, and we have a GHGL GP42 ferritin vaccine online that we expect to start sometime early next year, and we'll try that either alone or in combination with GP350 at the NIH. Moderna, as I mentioned, has a GP350 GHGL GP42 mRNA vaccine being tested at multiple sites right now, and this vaccine also began enrolling in 2022. So we have two different vaccines that are being studied right now. Now, importantly, immunocompromised patients, transplant recipients, individuals with primary immune deficiency that can develop severe EBV disease may not respond well to an EBV vaccine. So we have a plan for them as well. And that brings to the second part of my talk, EBV monoclonal antibodies. So prophylaxis with an EBV monoclonal antibody might prevent or attenuate EBV infection. And to date, we've isolated monoclonal antibodies from humans that have high neutralizing antibody titers or animals for which we've vaccinated with EBV vaccines. And humanized mice who are given, or we now give them monoclonal antibody intraperitoneally. You can see the different time points here, challenge them with EBV, and we can follow them for survival, viremia, lymphoproliferative disease, or lymphoma. And this protocol is really based on a study from Dorothy Crawford a number of years ago. So we've isolated multiple GHGL monoclonal antibodies, 3 of which are shown here. And when we looked at neutralizing titers of these antibodies for B cell infection or epithelial section or fusion inhibition for B cells or epithelial cells, B10 in general, you needed the fewest micrograms per mil to neutralize infection or to block fusion. So this looked like among the best antibodies here. We then did the humanized mice where we gave them the antibody and challenged them. And you can see the animals that got the B10 here had the highest rate of survival. The animals that got the B10 had the fewest number of animals that had EBV viremia compared to PBS, C2, or F7, the other 2 GHGL monoclonal antibodies. And we also looked at these animals pathologically, and you can see that animals that got the PBS, the F7, or the C2 had tumors. You can see in the tissues, these white blood cells are lymphocytes that are resulting in tumors. And we look at either atypical infiltrates, lymphoproliferative disease, lymphoma, or the combination. Only the animals that got B10 had no evidence of lymphoma, atypical lymphocyte infiltrates, or lymphoproliferative disease. Finally, we collaborated with a group at the Vaccine Research Center that did crystallography of the antibodies combined with GHGL, and we model here GP42 onto that. And you can see that these 3 monoclonal antibodies bind to 3 discrete sites on GHGL. So to summarize, GHGL monoclonal antibody B10, and I haven't had time to show you GP42 monoclonal antibody A10, but Wei Bu will be giving a talk in just a few minutes on that. Both of these antibodies potently neutralize EBV infection, block fusion, and protect humanized mice from death, viremia, and lymphoma. And we think these monoclonal antibodies might be used to prevent infection or disease in immunocompromised patients who might not respond well to our EBV vaccines. So really, the people that did all the work, Wei Bu has really been leading much of the work in my laboratory. We have a graduate student, and over the last 6 or 7 years, we've had a succession of postbacks who've worked on these projects. Dr. Durkee Schock is a clinician that works with me, who's the PI on our clinical trial. And as I mentioned, this is a collaboration with the Vaccine Research Center. So I'll stop here, and if there's time, be happy to try to answer some questions.
Thanks, Jeff. It was a great talk. I have one question on each part, if that's okay. So on the vaccine part, you mentioned GB in the introduction, and I was just kind of curious. It seemed to be conspicuously absent in the rest of the talk, including the depletion study. See if you can comment on why. Would be good, because I mean, obviously, as I know that you know, for HSV and CMV, GB has been a focus. On the second part, the mapping of the antibodies just visually looked like they map roughly to the previously known epitopes, or E1D1, and maybe some of the CL antibodies. Could you comment on that as well? Great.
Sure. So part 1, what about GB as a vaccine immunogen? So GB, as you know, as well as I do, has different forms, a post-fusion and a pre-fusion form. We had difficulty expressing really good levels of GB on the surface of cells to deplete the antibody. It may very well be important as an immunogen, but we can't tell that from the depletion studies. And as I showed you, 80 to 100% of the neutralizing antibodies were depleted, but we do think GB is something that we want to look at, in the future as an immunogen. So I think that's a really important question. Of course, if we could get the pre-fusion form of GB, that's sort of the holy grail in terms of a GB immunogen. The second question about is the mapping of these epitopes. So our most potent antibody, B10, actually maps to a site that's similar but not identical to AMO1, which the McGuire group has shown also to be very effective as a monoclonal antibody. So coincidentally, those 2 antibodies, were quite potent. One of the other antibodies, as you mentioned, maps to a site that's been previously been shown with some antibodies that were made by the NCI and studied by Lindsay Hutt-Fletcher. And the third antibody maps to a novel site. We have 2 other antibodies that also map to different sites. So unlike GP350, where there seems to be a major antibody, a site of for antibodies to the CR2 binding site, GHGL seems to be very different, and there are antibodies, epitopes in multiple sites that have neutralizing activity.
Yeah, thanks, Jeff. One could argue about whether one really wants sterilizing immunity, perhaps because of how long it would last. But if assuming that you wanted to achieve sterilizing immunity or the most preventive vaccine, it would seem that an IgA response is critical. Now, do these elicit, do they elicit much IgA, and are there efforts to try to develop vaccines that would result in really good mucosal immunity?
So that's a really important point. This virus infects through the mucosa, and having IgA would be useful. So we have considered doing vaccinations at different sites as opposed to intramuscular vaccination, or some different types of vaccines that might give mucosal immune responses. So I think that's important. Potentially you might get enough transudation of antibody at the mucosa. The human papillomavirus vaccine, of course, which is also is a mucosal type infection, is given intramuscularly and seems to be highly protective, and it's thought to be due to transudation of antibody. And then there's this observation from Jim Sixby many years ago that IgA can potentially enhance infection at least in studies that he published a number of years ago. But I think it's important to keep in mind, keep a broad, open mind about this. And it's something that we plan to look at, but I'm not sure that our vaccine is ideal for inducing, it's probably not ideal for inducing an IgA response. Again, I am skeptical if we're ever going to get a sterilizing vaccine against a herpes virus. The varicella-zoster virus vaccines, of course, don't induce sterilizing immunity, but it's something I think IgA is important to look at.
Last question.
Jeff, the process is aiming at a particular, or using a particulate type of antigen, which sort of triggers the B cell receptor directly. But I wonder whether you really develop good T cell responses, whether there's a memory induced, yes or no. Do you know anything about that?
The short answer is no, we don't know about that. Again, we're sort of modeling things off of successful the HPV vaccine. We just think that long-term immunity is going to be particularly important, and that's why we're sort of focusing on this approach. The DNA vaccine from Moderna, I think, is going to be better for T cell responses than ours will, but I'm a little bit concerned about the duration of the immune response and don't want to shift mono to a 30 and 40-year-old population. Yeah.
Thank you. And our next speaker is Michelle West from the University of Sussex. She's going to talk to us about natural variants of EBNA1.
Thank you. Thank you very much. I'm presenting this work on behalf of my postdoc, Vishwa, who wanted to attend visa complications. So unfortunately, you have me instead. So a little bit of introduction to EBNA1. So we've heard quite a lot about EBNA1 already. So to remind you, it's a dimeric DNA binding protein that's required for viral replication, genome maintenance, and segregation. It can also regulate transcription of both viral and host cell genes, and interacts with a number of host cell proteins to promote the survival of infected cells. It's the only viral protein expressed in all EBV-associated tumors, so this makes it an attractive therapeutic target. So this is just a cartoon of the EBNA1 domains, and just to draw your attention here to this C-terminal location of its DNA binding and dimerization domain. So the structure of this DNA binding domain was solved. So EBNA1 binds to an 18 base pair motif as a dimer. This is the dimerization interface shown here. And it has a flanking domain, which includes these key flanking helices, which I'm going to refer to again later. So it binds to multiple copies of its consensus in the origin of latent replication, or EP. So there are multiple copies in the family of repeats and 4 copies in the dyad symmetry region. The spacing between the consensus sites in the family of repeats is wider than that between the dyad symmetry sites, and we saw nice cryo-EM structures from Paul Lieberman earlier on in the meeting that showed that very nicely. And those cryo-EM structures show what the X-ray crystal structures have also shown, which is when 2 EBNA1 dimers are sitting side by side on 2 of the dyad symmetry sites, there's only a few nucleotides in between, and actually they bend DNA, and there are important interactions in this dimer-dimer interface between these flanking helices. So our interest in EBNA1 and EBNA1 variants came from a collaboration with Paul Farrell, who sequenced a large number of EBVs from across the world. Around 241 are shown in this phylogenetic analysis. And one of the new pieces of information that came out of that study was that he identified additional variation in the EBNA1 protein sequence that hadn't previously been reported. And this separated nicely on this phylogenetic tree and identified 5 non-adjacent amino acid changes in the DNA binding domain, where the sequence of PSMVT, which you find in the B958 reference strain, is replaced with QCIGP. So I'll be referring to this as the QCIGP variant. And what he found was that this strain is present in around 75% of the EBVs that were sequenced from the EU and Australia samples, and around 96% of African samples. So it's a prevalent, naturally occurring variation. So there had previously been variation in EBNA1 noted at this additional amino acid 487, which is also in the DNA binding domain. So in B958, this is an alanine. There is a variation at this position to valine, and you find this in the M821 strain. And this has actually been shown to have an impact on reducing viral genome replication and maintenance capacity compared to B958. So these variant amino acids can have implications for the function of EBNA1. So interestingly, when we find these 5 non-adjacent changes, this QCIGP, they are only ever found in the presence of either a leucine or a threonine at position 487. So the starting point of Vishwa's project was to determine, and a previous postdoc in my lab, Rajesh Ponusamy, was to determine the impact of the variant residues on the structure of the EBNA1 DNA binding domain and to try and understand the structural basis for their co-association. So why do these 5 non-adjacent amino acids co-occur? Variants in these amino acids co-occur. We then wanted to have a look at how this might impact on druggable pockets that have been identified in EBNA1, and then to look at the impact of this variation on the properties of EBNA1, so replication, genome maintenance, and any protein interactions that might map to that region. So to do this, we expressed and purified EBNA1 DNA binding domains from representative natural strains containing these variant residues. So the QCIGP 487L variant is actually the Raji sequence, and the QCIGP 487T variant is actually a Dowdy sequence. So we took these natural sequences. So you can see the positions of these 5 variant amino acids. But just to note, there are a few other changes in these strains, but these weren't identified as clustering phylogenetically, so they're just random changes, and most of these are changes to conserved amino acids. So Rajesh solved the X-ray structures of B958. This has already been published, but we resolved it at a slightly higher resolution, and he solved the structures of the QCIGP L and T variants. And as you might expect for naturally circulating viral strains, the structures overlay very well. They're very, very similar. And what you'll note, you can see on this slide here is I've colored in orange the locations of the variant residues. So you can see one of them is in the dimerization interface, but the remainder of the changes occur on this flanking helix. So you can see 2 of them here, and the other ones are easier to see on the other monomer, the other side here. And this is the position of this additional variation, the position of this 487 amino acid, which is alanine in B958. So if we look at how this might impact on druggable pockets, there was a very nice computational study done by the Lieberman group where they identified potential binding sites for drugs, so kind of hydrophobic pockets that you might be able to slot a drug molecule into. And they identified a number of sites that with this potential characteristic that they called secondary, tertiary, quaternary, and the primary site is here. It's not indicated on this slide. So if we zoom into where our variants are sitting, so the tertiary site is actually a dimerization interface. So you can see that this methionine to isoleucine change that we have in these variant strains actually makes quite a difference to the space available in this pocket in the dimerization interface. And then at this quaternary site, which is at the end of the flanking helix on the opposite side to the DNA, you can see that in B958, we have a serine opposite a valine. And in order to accommodate this slightly larger cysteine side chain, which we find in the variants, this valine is changed to glycine. So this gives us the first piece of information that we were interested in trying to understand the structural basis for why these 5 changes might co-occur. So you could understand how this glycine here would help to accommodate this larger cysteine side chain. So I just want to point out the importance of bearing in mind natural variation in strains. I guess we're not the only virology community that likes its reference strain and kind of does a lot of experiments with that one strain. But if you look actually at some peptide inhibitors that have been developed and fine-tuned more recently, you can see that the premise of these peptide inhibitors is to disrupt dimerization, targeting this central sequence in the dimerization interface. And you can see in our variants, actually, one of the central amino acids that is being targeted here is actually changed to an isoleucine. So it is important that we take this into consideration. So because of the location of the variant amino acids on this flanking helices, and the crystal structure that was published by the Lieberman group of this dimer-dimer on the 2 dyad symmetry sites, we also solved the structure of the B958 dimer-dimer, again, repeating the work that was published, although actually using dyad symmetry sites 1 and 2 rather than 3 and 4. They're very similar. And we've also solved the structure of the dimer-dimer on for the variants. So if we zoom in at what's happening at this flanking, at these flanking helices, I think this gives us more information on why these amino acid changes co-occur. So if you focus in on the central part of this dimer-dimer interaction interface, you can see that this threonine 585 to proline change is right in the center of the interaction interface. And you can see that the 487 amino acid is also right in the center there. So by changing the threonine to proline at this position, you will lead to a smaller side chain in this pocket and a bigger space in this interface. So I think this shows us quite nicely why an alanine at this close by 487 position isn't tolerated in the variants, and they actually need a larger side chain here to kind of pack out this interface and stop it kind of moving too close together or slipping about. So this is why we only see leucine or threonine at this position. You also note that the other variants are at either end of this flanking helices. So we know that proline at this position here is a helix breaker, so it would help to kind of lock the end of that helix in position. And actually serine can also serve the same job because its side chain can hydrogen bond back with the main chain in the polypeptide. And indeed it is doing this in this structure. So we think that providing some flexibility for this flanking helix to be able to accommodate these 2 central changes by changing this proline and the serine is important. So that doesn't explain why we have this change in the dimer interface. So we think that the change in the center here, which would be located here, might be important to provide some rigidity for the dimer so that it actually keeps those flanking helices pushed together. So we think that the crux of the co-association is in maintaining this flanking helices interaction in the dimer-dimer that forms at the dyad symmetry region. So we then asked whether these variants showed any difference in the DNA binding properties. So for this, we used a fluorescence polarization assay where you just have a fluorescently labeled DNA oligo and you can measure binding of recombinant EBNA1 to it. So we saw no difference at binding to family of repeats. These are high affinity sites. But at the dyad symmetry region, and we chose the one particular sequence from here, we see that the L variant has a lower affinity. We also see this if we look at the dimer-dimer interaction. So this is showing an electromobility shift assays, and we've quantified the dimer-dimer formation here for the different variants. You can see we also see reduced binding of the L variant as a dimer-dimer. Now, this can't be the result of just these 5 amino acids because they're the same in the T variant, and we don't see this property. In actual fact, the T variant does show a slightly higher affinity for binding to both the double site and the single site. So we think it's the result of some allosteric effects of the other variation or a combination of the other amino acid changes plus the QC-IGP, and we're investigating this further. Because the Lieberman group had published this EBNA1 inhibitor, which disrupts DNA binding, we also examined the sensitivity of these variants to inhibition by this inhibitor, which we had synthesized by our medicinal chemists at Sussex. And consistent with the L variant showing a low affinity for binding to DNA, again, this is assessing the quantifying the dimer-dimer bands here. We see that it's more sensitive to inhibition by VK1248. And Vishwa is just in the middle of completing a number of replication and genome maintenance assays. So the first results that we have here are from the replication assay, where actually we don't see, and this is using full-length EBNA1 in cells and monitoring OREP-dependent plasmid replication. We see that the L variant isn't actually impaired in the context of the full-length protein, but the T variant does seem to show a slightly enhanced capacity for replication. And as I say, we're just completing this set of work now.
1 minute.
That's fine, that's my summary. So I've shown you X-ray structures that reveal that the variants have differences in druggable pockets that we need to bear in mind when we're designing inhibitors and for drug discovery. We've shown some information on why, on the structural basis for those 5 co-associated variant residues. We've shown you some information that certainly the DNA binding domain of the L variant looks as if it doesn't bind as well to dyad symmetry sites, even though, and is more sensitive to inhibition, even though the drug binding pocket is conserved, and that the T variant may bind to, certainly in vitro, to OREP dyad symmetry sites at a slightly higher affinity. So Rajesh Ponusamy did all the X-ray crystallography in association with our X-ray crystallography facility manager, Mark Rowe, and our collaborator, Erica Mancini, who's a structural biologist. And Vishwa has done all the cell-based assays supported by Helen. And we had a new addition to the team during lockdown, Bob the kitten, who used to like to join our Zoom meetings by sitting on my shoulder or lying on my mouse. And he was instrumental in keeping us sane while we had far too many Zoom meetings. Thank you very much.
We have time for questions. Very well.
Were most of the sequences that you sequenced, Paul, type 1? I don't think you picked up any additional variation in type 2. Yeah, the differences between type 2 and type 1 are driven by changes in EBNA2 and the EBNA3s.
Thank you.
Joanna has a question at the back.
Do you see any correlation with the variants that you've looked at and where you see that reduced DNA binding like the Raji variant and the level of EBNA1 in those cells that might be necessary for the viral propagation, I mean, for viral persistence in the cell line?
That's a really interesting question, actually. So kind of does it compensate by kind of expressing a bit more? I mean, I guess we could, it's difficult, isn't it? I mean, we could look at Raji, Dowdy and kind of compare it. I mean, I suppose, you know, the level's quite low. There's probably going to be some cell line variation. And we're trying to do our replication assays and make sure that we've got, you know, the same expression level because it would be impacted.
Yeah, I guess you'd have to look at quite a lot of cell line, representative cell lines to get an idea.
Yeah.
Thank you.
Our next talk will be by Waibu from the NIH. He's going to talk to us about a GP42 monoclonal antibody.
Okay, good afternoon. So I want to thank the organizer for this opportunity to present our work here.
Just, I think there is
Hello,
No,
Remove?
just this here. Yeah.
Okay. Yeah. So as we know that EBV B cells and epithelial cells using a different combination of glycoproteins. For the B cell entry, we know that viruses, glycoprotein GP350 bind to the cellular receptor CR2 or 3035 as the first step, to allow the viruses to attach to the cell surface. After this binding, GB42, binding to HR class molecules, and here is GB42 tethered to GHDL, forms heterotrimer. And after this binding, this fusion is triggered and accomplished by the glycoprotein B. So the GHDL, GB42, and the GB are composed fusion apparatus for B cell entry. Different from B cell entry, so instead of using GB350 as attachment molecules, it's using BMF2 to attach viruses to the cell surface. And here, just without need help of GB42, the GHDL and the GB compose our fusion apparatus. So in this talk, we will focus on the antibody against GB42. And we know that GB42 is a type 2 glycoprotein, and there are two types of GB42 has been detected on infected cells. So the full-length GB42 and also the other versions, the soluble GB42, this is a signal sequence and transparent domain was removed by the cellular protease. And also based on the crystal structure, we know that GB42 is tethered to the GHDL using its N-termini to form heterotrimer at ratio 1:1:1. And also we learned that GB42 is bound to HLA, is essential for our entry into B cells. And also the B cell fusion is mediated by binding of the GB42 to the HLA class 2 molecules. And we did literature search, find very limited reports on, GB42 antibody. And only one reported GB42 neutral antibody has been reported is F21. And this antibody has been, isolated from mouse immunized with EBV infected cells. And this is paper published about 40 years ago. And we found this clone, F21, is able to neutralize B cell infection. And in our hands, this antibody doesn't neutralize epithelial cell infection. So isolation of human GB42 specific antibody is pretty challenging. So because usually people using the fluorophore-labeled GB42 as a probe to fish out these B cells, specific B cells. However, this method is not working because we know GB42 band, HLA is band all of B cells. So we have too much background. It's unlikely to use in the probe to fish out the GB42 antibody. So there's so far no human GB42 antibody reported. And we're trying to use a different way to isolate GB42 antibody and using the single plasma blast cells from the immunized monkey receiving GHDL GB42 previously. In this study, sorting since they have limited in antigen for the cyno monkeys, so we did tons of native gating. So the first is we did live cell and singulate and gate out the NK cell on the monocytes and gate out T cell and early B cells and gate out HLA positive cells and gate out dendritic cells. So finally, we just sort single blast into 96 well plates, and then we're using the primer for the antibody high-reach land chain to amplify the viral region and express them and test its binding to GB42 and the neutralizing activities. So luckily, we got one clone that name is A10, looks like it's binding to GB42. So we test this antibody and immobilize the GB42. We test this antibody. We immobilize the GB42, GHDL or GB42 in the sensor and deep into the wells contains antibody A10. And we see that the A10 is binding to GB42 and binding to GHDL GB42, but failed to bind to GHDL. The binding profile is the same as F2-1. We know that is GB42 antibody. So from this binding, we confirm that this is a GB42 specific antibody. Also, we did kinetic assays to measure the K-on value and the KD value, and to evaluate, to determine their affinity. So we found this A10 antibody has about 6-fold higher than F2-1 to binding to GB42, and this A10 has a 17-fold higher affinity than trimer than the F2-1. Sorry, and then F2-1 binding to the trimer. And after that, since we have two antibody now, it's F2-1 and our A10, we want to know if these two antibody target to the similar epitope or different epitope. So we're using cross-competition assays. So here we just immobilize sensor with ligand, is GB42 ligand or GHDL GB42. And then we dip this the sensor into the well containing prime antibody is A10 F2-1 isotype control. And then we dip this one go to the next file, contains the second antibody. So the top one shows the raw data, and based on the raw data, we summarize the computation, the experiment like showing here. So from this computation, the calculation, we know that A10 can compete F2-1 to binding both GB42 and the trimer. So that suggests that M21 and A10 target to an overlapping epitope on GB42. And the next question is very important, is this A10 is able to neutralize virus infection or not? So we did the B-cell neutralization assay. And here you can see this line is F2-1, and this line has the blue dot is A10. We see that A10 is very good to blocking B-cell neutralization compared to F2-1. And also we including the most potent GB350 monoclonal antibody 781 as our control. It's showing this A10 and 781 has a similar potency to blocking B-cell neutralization. And this is IC50 titer, and this is the actual value. So from calculation actual value, we clearly know that A10 is about 12-fold more potent than F2-1 to neutralize B-cell infection. And since I mentioned GB42 is the components for the B-cell fusion, so next we want to see if this A10 or F2-1 able to blocking EBV fusion. So here we use the glycoprotein mediated fusion assays. So in this assay, we compare just about using this antibody, we found that A10 is amazingly better than F2-1 to blocking B-cell fusion. And also here's IC50 titer, and this is actual value. It's showing that A10 is about 60-fold more potent than F2-1 to blocking B-cell fusion. So besides this study, we want to know what is mechanism, how this antibody neutralizes virus infection. The most common idea maybe they target receptor binding site, so the blocking HLA receptor binding. So to test this hypothesis, we just using the recombinant HLA protein and mobilize to the sensor. And then we dip into the well contain GB42 or GHGB42 plus with isotype control A10 or F2-1 antibody. And here is the data showing from the one experiment, and the blue one is our isotype control. And we have here summarized this blocking experiment in this 3 independent experiment. From this summarized data, it's clear to see that A10 is much better than F2-1 to blocking the GB42 binding or GHGB42 binding to the HLA receptor. Besides using this, it's a recombinant protein, we're asking about how about our antibody blocking the GB42 binding to the human cells. So we do the fact-based computation assay. In this assay, we use the fluorophore labeled GB42 and mixed with GB4 antibody or mixed with the anti-HLA antibody, and then we just load it on the human PBMC, and then we did FACS. So this results from the one donor. So we see GB42 is binding B-cell very well. And then we see that anti-HLA antibody, A10 and F2-1, reduced GB42 binding to the cell surface. And this is isotype control. And here we summarize our results from 4 independent blood bank donors. It's the results showing that A10 is the most potent, followed by HLA class 2 antibody, and then F2-1 to blocking the GB42 binding to the human B cells. And we did the same experiment. Instead of using GB42 probe, we used the GHGL GB42 probe. And it is also from one donor, and this is from result from 4 independent donors. The results very consistent. So the A10 is the best, and followed by anti-HLA antibody, and the F2-1 is blocking this GHGL GB42 probe binding to the B cells. So besides this in vitro study, we next want to evaluate if this antibody are able to protect humanized mice from EBV infection. So we give the this purified antibody to mice by IP injection at 6 doses, and the virus challenge at day 0. And then we collect blood weekly, and until the end of studies of week 15. And also we did real-time PCR to quantify the EBV copy numbers in the blood. So for the PBS group, it's clearly we see this all the 5 mice have infected, and the peak level is at either 3 or 5 weeks, and then the viral load is reduced. And for the F2-1, we know that at week 9, all of 4 mice got infected, and also this 1 mice keep higher the titers during the course of study. However, for the A10, we have this EBV the DNA is under detection level. So from this comparison, we know that the mice receiving A10 has lowest viral load in blood after challenge with EBV. And at the end of the study, we euthanized mice and collect the organs. We find there's a tumor, it's the lesions, in the PBS group, and also we find this tumor tissues in the mice in the F2-1 group. And we send this tissue section for pathology, And clearly, they are confirmed, this tumor are lymphoma. Also, we did EBR1 in situ hybridization to look at the, infiltrated EBV positive cells. So we found 3 out of 4, at least 3 out of 4 organs, from the PBS group. And these mice have, EBV positive cells are detected. And we have 1 mice, in 2 organs have the EBV cell detected. However, so A10, we didn't see any EBV positive cells. And this table summarized the mice with the disease. And we know that 80% mice, in the PBS group has microscopic lesions, and 20% had, LPD, and 6% has lymphoma. The 1 mice, like 20%, has this, microscopic lesions, but it confirms lymphoma. However, for A10, this group of mice is disease-free. And in conclusion, just we, isolate a novel GB40 antibody from cyno monkeys, and we did biochemical and biological comparison about GB40 antibody, A10F-1, and we found that A10 is more, there's a higher affinity to binding GB42 and the trimer, and here is showing the fold, this, A10 over F-1. And also A10 is very potent to blocking, GB42 and the trimer binding to the human B cells, and A10 is potent to blocking, the B cell neutralized viral infection, and also potent, just to blocking, B cell fusion, compared to F-1. And also we learned that A10 F-1 target an overlapping epitope on GB42, which is crucial for HLA receptor binding, using this mechanism to neutralize virus infection into B cells. The most important thing is that we learned the humanized mice receiving A10 were protected from EBV, viremia, and lymphoma. Thank you for your attention. I'll stop here and answer your questions.
Questions for Weibo?
Thank you. Very nice work. Obviously the antibody is very potent. That's pretty clear. So I want to go back to the original depletion study you did, where you used GP42 only, I think.
Yes.
Have you ever repeated it or thought about repeating it with GHGL GP42 and see if there's a difference between that and GHGL?
I remember doing that study, just we didn't try the trimer. We have a GHGL individually, GB40 individually. The reason is that, some GB42 based on quick structure, the epitope is hidden. So that means if you use the trimer, possible you will missing some like antibody against GB42 targeted epitope, there's a contact with GHGL. And also GB40 antibody based on depletion is not allowed, compared to GHGL to contribute based on neutralization. So we believe the reason maybe GB42 very tricky protein, the binding B cells all by HLA, maybe not like a very good antigen to elicit the very good immunogenicity, produce antibody against GB42.
So is the expression of the target sufficiently leaky in any of the latency phases, 1, 2, or 3, that you can detect the latency antibodies, the latency cells of this antibody?
I think we are interested to work on that as this hypothesis to see possible this antibody can become therapeutic. So that is able to prevent a radical lymphoma is able to to kill the lymphoma or something to control this cancer.
Mind LCLs?
That one we didn't test it, so sorry, we didn't do that. Yeah, it's good to try. Yeah.
Thank you.
Thank you.
Our next speaker is Silvia Gianco from the University of Padova.
Good morning, good afternoon to everybody, and thank you to the organizing committee to give me the opportunity to show our studies on the anti-proliferative effect of telomerase inhibition on EBV positive malignant B cells. So I start with an introduction on telomerase in DBV crosstalk. Then I will show our last results using TERT inhibition on LCS in vivo and in vitro models. As you know, maintenance of telomere length is a must for cancer cells. In the vast majority of tumors, is provided by the reactivation of telomerase. Telomerase is a ribonucleoprotein complex with an internal RNA template and a catalytic subunit with telomere reverse TERT with a telomere reverse transcriptase activity, which maintain telomere length, thus preventing cell senescence and apoptosis. Also, EBV-driven malignancies maintain their ability to grow indefinitely through inappropriate reactivation of telomerase. Indeed, only EBV-infected B lymphocytes with the sustained telomerase activity are truly immortalized and become continuously proliferating lymphoblastoid cell lines. In addition to its canonical role in maintaining telomere length, TERT may contribute to EBV-driven tumorigenesis also through extratelomeric functions. On this ground, our previous studies demonstrated that the latent protein, EBV latent protein LMP1 activates TERT at transcriptional level through NFKB and MAPERK pathways. On the other hand, high levels of ectopic or endogenous TERT expression in established LCLs were associated with the latent EBV stage and prevented the reactivation of EBV lytic cycle. Consistently, the TERT inhibition by short-terpene RNA triggered the EBV lytic cycle with the expression of early and late EBV lytic genes, and with the release of complete virions in the culture supernatant. The reactivation of EBV lytic cycle was associated with a propoptotic effect with the death of infected cells. Mechanistically, we found that TERT contributes to EBV latency through the Notch2-BATF pathway. In particular, we found that ectopic TERT expression activates Notch2 in a dose-dependent manner, and this effect was mediated by the NFKB signaling. Consistently, ectopic TERT expression in TERT-negative LCS increased the expression of Notch2 and its target genes, including BATF. BATF is a transcriptional repressor of BZLF1, the main inducer of EBV lytic cycle. Thus, TERT through the Notch2-BATF pathway appears to be a critical regulator of the balance between latent and lytic stages of the virus, and its inhibition could be exploited to promote the death of infected cells and sensitizing to antiviral prodrug as ganciclovir. More recently, we have studied the effect of short-term TERT inhibition, also employed a chemical inhibitor, BIBR 1532. BIBR is a synthetic non-nucleoside compound which selectively inhibits telomerase catalytic activity. In our LCS model expressing high endogenous or ectopic TERT, BIBR efficiently inhibits the catalytic activity of telomerase. Using this approach, we have demonstrated that the short-term TERT inhibition by BIBR induces cell cycle arrest with a significant accumulation of cells in the S phase, and also induces apoptosis. These effects were observed in TERT-positive LCS, but not in TERT-negative cell lines. The antiproliferative and pro-apoptotic effects of short-term TERT inhibition in LCLs were associated with the activation of ATM and ATR DNA damage response pathway. Interestingly, the DNA damage response was unrelated to telomere length and dysfunction, as cells treated with the telomerase inhibitor show the same telomere length as control cells. And importantly, The majority of gamma H2AX damaged foci were randomly distributed in the genome and did not co-localize with the telomere probe signals. The evidence of extratelomeric function of TERT in cellular kinetics and in resistance to apoptosis strongly support the telomere length independent therapeutic effect of TERT inhibition. And then to explore the possible clinical application of TERT inhibitors in combination with antineoplastic drug, we treated established LCS with the two drugs employed for the treatment of B-cell malignancies, cyclophosphamide and fludarabine, alone or in combination with the TERT inhibitor. Results show that the combined treatment significantly impairs proliferation, growth, and viability compared to the drug treatment alone. These important results need to be validated in an animal model. And to investigate the possible clinical application of TERT inhibitors in combination with antineoplastic drugs to counteract EBV-positive tumor growth in vivo. To do this, we chose a zebrafish embryo xenograft model and the flow cytometry analysis to detect the fluorescence injected cells. Using this model and this approach, we have already demonstrated that while control untreated LCS proliferate in zebrafish embryos, the cells treated with the TERT inhibitor show reduced proliferation rate compared to control cells. For the combined treatment experiment, we treated LCS with DMSO as control or BRBR for 24 hours. Then cells were CMDIL labeled and injected into the yolk sac of 72-hour post-fertilization zebrafish embryos. 24 hours after xenotransplantations, successfully engrafted zebrafish embryos with similar fluorescence intensity were divided into 6 experimental groups, and the drugs were added to the medium. The number of fluorescent cells were monitored in dissociated embryos at 24, 48, and 72 hours post-treatment. In each experiment, also non-xenograft zebrafish embryos was employed as control to set the experimental threshold. Results show the confirmed that LCS proliferate in zebrafish embryos throughout the experimental period. And while in comparison, BIBR treatment show a reduced proliferation rate compared to the controls. Cyclophosphamide in red and fludarabine in blue also show a significant decreased proliferation rate compared to the controls. And importantly, the combined treatment with BIBR and cyclophosphamide further reduced the proliferation rate of xenograft cells compared to the controls and also compared to the drug treatment alone. This cumulative antiproliferative effect was even more evident in the combined treatment with BIBR and fludarabine, particularly at 48 hours post-treatment. Mechanistically, we are investigating the molecular processes through which TERT inhibition impairs cell proliferation without affecting telomere length in vitro model of EBV-driven malignancies. To do this, we are evaluating the role of MYC, which is a key regulator of cellular proliferation. We observed that the VIBR treatment reduced significantly the expression of MYC, both mRNA and protein level. In addition, since MYC is a repressor of P21, an important regulator of cell cycle progression, an inhibitor of cell cycle progression, we evaluated the expression of P21 under TERT inhibition. And we observed that in established LCS, the inhibition of TERT was associated with the increased expression of P21, both mRNA and the protein level. In particular, Western blot immunofluorescence analysis revealed that the increased expression of P21 is localized at the nuclear level, thus allowing its cell cycle inhibitory functions. In conclusion, our in vivo results indicated that TERT inhibition increases susceptibility to antineoplastic drug, supporting the use of TERT inhibitors in combination with chemotherapeutic agents as an efficient anti-cancer approach for EBV-associated malignancies. In vitro data suggested that the telomerase inhibition alters cellular proliferation through the downregulation of MYC and the upregulation of P21. These findings support the concept that TERT inhibition can contribute to chemotherapeutic benefits beyond the telomere erosion. I want to thank Prof. De Rossi and all the lab staff and the zebrafish team. And also thank you for your attention. Sorry.
So your data suggests that TERT regulates MYC.
Maybe
right?
that.
Yeah. So how does it do so? I mean, there must be some information on that from the MYC world or the TERT world.
We are investigating the fine mechanism, and we have found that the NFKB signaling seems involved in this mechanism. We see that TERT contributes to P65, NFKB P65 nuclear localization in a subset of NFKB target genes, including MYC.
Thank you.
So do you know how commonly TERT is upregulated or is highly expressed in EBV lymphomas?
I don't understand, sorry.
How often is TERT upregulated in EBV lymphomas?
TERT is upregulating in all lymphoma because without TERT, the telomere continues to shortening and move to apoptosis.
Well, you can have the alt mechanism as well to maintain telomere length, but yeah. So sometimes it will be TERT, sometimes alt. Yeah, so I've got transcriptomic data showing EBNA3B represses TERT.
Thank you.
Thank you.
Arne Kaiser from the Helmholtz Center, Munich, is going to talk to us about LMP1 TRAF2 inhibitors, interaction blockers.
Thank you. First of all, thank you for staying here this afternoon, and thank you for the organizers for the opportunity to speak about our LMP1 inhibitor project. This project started about 7 to 8 years ago, and actually this is the first time that I'm reporting results to a larger audience. As you all know, EBV is a class 1 carcinogen, now also involved likely in multiple sclerosis. It is involved in eliciting tumors, cancers from B cell, T cell, and epithelial cell origin. And many of them express LMP1 and are dependent of LMP1. For example, PTLD, which is one of our main and primary targets. So I think you agree that EBV therapies are urgently needed. And currently, there's no drug on the market that targets EBV. And we have been involved in studying LMP1 signal transduction and function in cell transformation for almost 20 years now. And we're involved in deciphering this signaling network. So LMP1 is a transmembrane protein which resembles a kind of viral TNF receptor interacting with TRAF molecules and TRAT, mimicking CD40 receptor on B cells. And it is critical for transformation of EBV-infected B cells. And having all this signal transduction network in mind and working so long on these pathways in interactions, we thought taking this a step further and using LMP1 as a target for small molecules. And especially we came up with the idea to develop protein-protein interaction inhibitors of the interaction between TRAF2 and the CETA1 domain. So this interaction is critical for CETA1 function, for NF-κB activation by this domain, survival and transformation of B cells. By interference with CETA2, it also is required for NF-κB and junk signaling by CETA2. This interaction is quite well characterized, so the interaction site is well known. This is a PXQXT motif, which is also found in some cellular receptors. And of importance for such an inhibitor development project, the structure has been solved. So this is required for structure-based development of such drugs. One drawback is that this interaction has not a pocket where LMP1 is binding, but this is more a shallow surface. So this can be a challenge for development of protein-protein interaction inhibitors. But nonetheless, it's a validated target because when you mutate this motif, EBV loses almost all its capacities to transform cells. So first we had to come up with a suitable screening assay. So we started out with ELISA-based assays with purified protein components. These assays worked very well, actually, but they are not really suitable for high-throughput screening because of the many washing steps that are required. So what you would like to have for high-throughput screening is a mix and measure assay where you just combine the components and you get an immediate readout and can add your compounds and in one step you get a result. And we came up with an assay here that is based on an alpha screen system originally developed by Perkin-Elmer. So these are beads to which we couple a purified signaling domain of LMP1 and his tagged TRAF, in this case TRAF2. We have this system, we can use the system like a Lego system, so we have also other receptors, we have mutants, you get immediate results regarding KD values and so on, can test mutants, compounds, and so on, whatsoever. So yeah, these are the protein components, and this assay works very nicely, as you can see here. We titrated in a cetiran-derived peptide. You can see that this peptide blocks interaction between LMP1 and TRAF2 with an IC50 of 270 micromolar. So this assay had to be developed further to be suitable for high throughput screening. So automatization was required and downsizing to industry standards. So the final assay was performed in a plate in 1,536 well in 7 microliters. So all components in 7 microliters. Having in mind that variations of plus minus 5% of the signal readout is allowed for such a screen, it's quite a task to establish such an assay to a level where you can really reliably perform a high throughput screening. We have no washing steps, assay temperature 21 degrees, readout and proteins are very stable, and we have a decent signal to background ratio of about 50-fold. So this shows you here in the screening that we have set prime values close to 1, which is speaks for an ideal screening assay. With this assay, we went for a high throughput screening campaign, basically screening 2 libraries. The by far biggest library was a library of the European Lead Factory, which is a private public partnership of the EU FFP7 program and the Big Pharma. So here we had 350,000 compounds, a result 15 qualified primary hits, and 1 qualified hit entered further hit to lead development. But today I would like to concentrate on the result of our second screening, which was an in-house screening at Helmholtz Center, library of 30,000 synthetical small molecules, which had covered a huge chemical space. So this were pre-selected compounds. The result were 39 primary hits with an IC50 lower than 50 micromolar, and 1 qualified hit entered hit to lead development. And I want to show you now the results and further development of this hit. So the hit compound EN254 specifically inhibits LMP1 TRAF2 interaction that we showed with our screening assay, but also was backed up with an alpha ELISA assay. So we have for a primary hit quite decent IC50 of 21 micromolar. See a very nice blocking curve. This is, by the way, the structure. I cannot show you more of the structure because the patenting is still ongoing, but I think this is not really required for the talk. And this compound is specific for the GST TRAF2, as you have here, a parallel assay LMP1 TRAF6 interaction, which is also a direct interaction. By the way, we had a poster here demonstrating this. Also that this is this interaction, direct interaction, is absolutely required for B-cell transformation and LMP1 function, and is also now a validated target. But here, this inhibitor is specific for TRAF2, no effect on TRAF6. So, of course, the question is then, how does this inhibitor interact with the complex? Does it interact with LMP1? Does it interact with TRAF2? What is it doing? To answer this question, we performed an STD NMR, which tells you whether such a compound interacts transiently with a protein. And here you see a 1D spectrum of the compound with these peaks. And the same peak occurs here in the STD spectrum, where the protein is excited and transfers the energy to the compound only if the compound transiently binds to the protein. You see the same peak here. And this tells us that the compound interacts with TRAF2. To get more information on the binding site, because this could also be allosteric effect, we had a couple of mutations in the in TRAF2 at the site where LMP1 is interacting. Many of them, of course, affect binding to LMP1, so we cannot test those. But one compound here, the cysteine TREF2, the cysteine 469 mutated to A, still nicely bound to LMP1, but the effect of the compound is completely lost. So that means this residue is involved in TREF2 interaction. That means the compound binds to LMP1, to TREF2 at the LMP1 binding site. So we went then in a hit-to-lead development of this compound to improve the activity. And so first we did this by cataloging. So we ordered derivatives around this primary hit, which are not always the ones that you would like to have, but at least gives you some information. So there was one compound, the best compound in this cataloging with 8 micromolar activity. And then we started into a directed structure-based improvement of these compounds by new synthesis. And here we established novel chemical matter, went through iterative rounds of synthesis, testing, hypothesis generation, synthesis, and so on. So this whole campaign took several years, actually. And finally, we could improve the activity of these inhibitors by more than 2 logs. And our best inhibitor is now this SBL 425, which blocks LMP1 TREF2 interaction with 91 nanomolar. So this is really very good. But we have also other compounds here with slightly different chemistry, which are in a very decent range. So of course now now comes the point, are these compounds somehow effective on EBV transformed cells, on LMP1 signaling, are they on target? And to answer this question, we used a cell system that we've established, published here 2 years ago.
A minute left. Minute left.
Okay. And here we see a very nice blocking of phospho-junk pathway in these cells with an EC50 of about 100 nanomolar, and also a blocking of the IKB pathway. So we are here really on target with these compounds. They have also effects on PTLD and LCL survival also in the nanomolar range, where they have much less effect on unrelated cells. So then we went through ADME profiling to get an idea which of the compounds is suitable for mouse studies. Not all of them are actually, but one compound, SBL442, is favorable and is now in pharmacokinetic studies to prepare mouse studies. So this is my summary. So I make it short. We developed the first-in-class TREF2, LMP1 TREF2 inhibitor. Next steps is in vivo pharmacokinetics and toxicity testing in animals, also proof of concept studies, and then of course preclinical and clinical development. Most of this work has been pushed forward by my long-term first PhD student, now a postdoc, Fabian Gieler, and of course we have great help in a fantastic team with medicinal chemists, structural biologists, pharmacologists. So without these people, this work would not have been possible. So thank you very much, and I'm ready for taking questions.
Thank you. I think we have time for.
Thank you. Very impressive tour de force. So I actually have sort of 2 questions. So one is that so LMP1 is known to aggregate at the plasma membrane, and the TRAD interactions or the TRAF2 interaction, is that thought to be monomeric or multimeric? And your binding studies are presumably monomeric, so they might even be augmented in inhibiting a multimeric interaction. And then the other part of that is, given the channel activity that we see when the transmembrane spans and that the cytoplasmic tail with the or the CTAR domains appears to be regulating that, would you expect these inhibitors to alter the conductance?
First question, so this assay is based on a GST fusion. GST forms trimers. So this kind of imitates on a densely packed bead the clustering you have on a cell. So when you really use peptides, you have another KD, for example. So the KDs here are very low. To the second question, could well be. So if TREF2 and this interaction is really regulating or involved in closing this channel function, these inhibitors might well also be effective to block or activate, depends on how this is regulated, the channel function.
Joanna had a question.
So this inhibitor binds to TREF2. Do you know if that, or this drug, do you know if it inhibits the interaction of TREF2 with any cellular proteins, and particularly any that might bind in a
Yes.
similar region?
Yes, it does. We have tested that. So it also interferes with TREF2 interaction with CD40. So this is, of course, then the discussion, is this an off-target effect? Would you like to have that or not? I would say in the moment that it's beneficial to block CD40 because this is involved in tumor microenvironment interactions with the lymphoma. So you might even, this might even augment the functions or the function of the inhibitor. And there are also CD40 dependent cancers that have nothing to do with EBV, which might also be targets.
Just to follow up on that, did you have any potential leads that might bind to LMP1 instead, or could you not follow that up?
That I didn't understand that.
Did you have any leads that might bind to LMP1 instead of TREF2? Any of your candidates?
Bind to
No.
LMP1
No.
instead of TREF.
So all compounds we found in all screenings always bound to TREF2.
Okay, thank you so much. And now we go back to the vaccine approach. So our next speaker is Dr. Harman Mali. Immunization with a self-assembling nanoparticle vaccine displaying EBV GHGL protects humanized mice against lethal viral challenge.
Yeah. Not a doctor yet, but hopefully soon.
Okay.
Yeah, so just thank you to the organizers for giving me a chance to share my research here today. My name is Harman Mali, and I'm a fourth-year graduate student in the McGuire Lab at Fred Hutch. And today I'm going to be sharing my thesis project, designing a next-generation Epstein-Barr virus vaccine. EBV is orally transmitted through saliva and is capable of infecting both epithelial cells and B cells. It's not clear what immune response a protective vaccine would need to elicit, but given that the correlate of protection for most licensed viral vaccines out there are neutralizing antibodies, it's likely that an EBV vaccine would need to elicit neutralizing antibody against infection of both of these cell types. When it comes to designing a vaccine, obviously the most important thing you want to consider is which antigens to include. And a logical first step there is to focus on proteins that are involved in viral entry. The idea being that if you can prevent entry, then you can prevent infection from occurring in the first place. With EBV, there are several proteins involved in entry. Other speakers have already gone over them, so I'm not going to re-summarize that, but I do want to reiterate the point that most of these proteins that are involved in entry are actually targeted by neutralizing antibodies during the course of natural infection, and we believe that any of them could potentially be included in a EBV vaccine. In the past, most EBV vaccine efforts have focused on GP350. However, it has been suggested in the literature that the inclusion of other proteins might lead to a more protective vaccine. Our lab wanted to further explore which other proteins might be important for an EBV vaccine. So we set about trying to isolate monoclonal antibodies against EBV proteins in EBV-infected individuals. And through that, we were able to isolate AMA1. Unlike the other anti-GHL monoclonal antibodies, which had been previously isolated, shown here, AMA1 was capable of potently neutralizing both epithelial cell infection as well as B cell infection, which suggested to us that if you could elicit AMA1-like antibodies through vaccination with GHL, it might be an important EBV protein to include in a vaccine. Since then, the discovery of other potent anti-GHL monoclonal antibodies, such as 769B10, which Jeff Cohen shared earlier, as well as and 1D8, which targets the distal epitope to AMA1's epitope, discovered by the Zhang lab. The discovery of these further supports the notion that GHL might be an important target for an EBV vaccine. Our lab has shown that the passive transfer of AMA1 to rhesus monkeys protects them against challenge with RHLCV, which is the rhesus ortholog of EBV. Likewise, our lab has shown that passive transfer of AMA1 to humanized mice protects them against challenge with human EBV, and that was the case in 11 out of 13 mice. 1D8, the other anti-GHL monoclonal antibody I shared earlier, has also been shown to be protective against EBV challenge in humanized mice. Taken together, these studies really provide a proof of concept that anti-GHL antibodies, specifically AMA1-like antibodies, may be protective in vivo against infection by EBV, and that if you were able to elicit AMA1-like antibodies through GHL vaccination, perhaps that vaccine too would be protective. Antibodies elicited by that vaccine could also, confer protection. Several researchers have shown that through multimerization, you can improve the immunogenicity of protein subunit vaccines. We wanted to explore that further in the context of GHL, and we did that using a combination of both computationally designed as well as naturally occurring protein subunits. The former that we used was designed by Phil Bradley at Fred Hutch. The 7-mer we used was derived from the C4b binding protein from Gallus gallus. The 24-mer we use is the same H. pylori bullfrog ferritin used by the Cohen lab. And the 60-mer that we used was designed by Neil King's lab at the University of Washington. We fused GHL to each of these multimeric protein subunit backbones, and in order to ensure that they express with the expected structure, size, and antigenicity, we proceeded to characterize them. One of the ways we did this was by collecting electron micrographs of each of these constructs. And as you can see here, we were able to confirm that each of these multimers expressed as monodispersed particles with no aggregates or any sort of clumping forming. And as you might expect, with the increasing valency, we saw increasing size in each of these constructs. So they're expressing about as we would expect. The next thing we looked at was antigenicity using a panel of anti-GHL monoclonal antibodies. And in each case, we saw that each of these constructs retained their antigenicity, and in most cases, multimerization actually improved the antigenicity of GHL. Having confirmed the expected antigenicity, structure, and size of each of our constructs, we proceeded to immunize C57 black 6 mice with our constructs. We performed 3 immunizations and collected blood after each immunization in order to measure plasma neutralizing titers against EBV infection of both epithelial cells as well as B cells. I'm going to go over the epithelial cell neutralizing titers first, as measured here by reciprocal ID50s. The way to interpret this is, the higher the potency of the neutralizing activity, the higher value you'll get with the reciprocal ID50. And as you can see here, after the first immunization, you already begin to see significant differences between the monomer, where there was no measurable neutralizing activity, and the multimers, where you saw titers sometimes past 10 to the third. Post-second, you really see the clearest differences between each of our multimer constructs and the monomer here. You start to see some neutralization. However, neut titers are as much as 2 log-fold higher, and in each case, significantly higher in each of the multimers. By the third time point after that third immunization, we see a further boost in titers with the monomer, and titers sort of begin to converge across each of these constructs. The next set of data are the B-cell neutralizing titers. These titers, it's worth pointing out, they're more difficult to elicit, so we do see them show up later and at relatively lower values compared to the epithelial cell neuts. However, the trend of the data is pretty similar. Post-second, we see the clearest differences between the monomer, where there's really no neutralizing activity, and each of the multimers. And again, post-third, these begin to converge. We next wanted to sort of explore the relevance of these clear differences we see in vitro between monomer and multimer. We wanted to explore that in vivo, the relevance of that in vivo, and we chose to do that using the 6-steamer, which, as you can see here, has consistently high neutralizing titers in both of these assays, and we want to compare that with the monomer. And we did so using a passive transfer approach. We, in humanized mice specifically, we did this in immunocompromised mice engrafted with human CD34 stem cells. We chose that approach over direct immunization because, due to the fact that humanized mice are immunocompromised, they don't mount a very strong antibody response. And this experiment, it took place in 2 parts. In the first part, we generated that vaccine-elicited IgG by immunizing large groups of mice with either a GHL 6-steamer or monomer. We then purified IgG from the blood of those mice. We also included a non-vaccinated group to control for the passive transfer, which took place in part 2. In part 2, we transferred 500 micrograms of the vaccine-elicited IgG to each humanized mouse. At day -1, we then bled the mice to ensure that each mouse received roughly equivalent levels of total IgG. And then at day 0, we challenged them with a high dose of EBV. And we included an infected control there, which had no treatment or any transfer, as well as an uninfected control. We then monitored those mice for 10 weeks for any signs of infection. The first sort of metric of infection that we looked at was survival. In the infected control group, the mice didn't survive past day 60. So in all 5 of the mice succumbed to the lethal challenge, whereas the uninfected control, as you might imagine, survived through the length of the experiment. The next group I want to look at is the control IgG. These mice received IgG, but it wasn't any vaccine-elicited IgG, and they also received challenge, and they fared about as well as the infected control, as you would imagine. Now, the mice that received the monomer vaccine-elicited IgG, they fared a little bit better than the infected control groups. 1 of the 4 mice did survive through the length of the experiment. However, by contrast, all 5 out of 5 of the, it didn't show here, but this purple line here, all 5 of the mice in the 6-steamer group were protected against lethal challenge. So already you begin to see sort of the superiority of vaccine-elicited 6-steamer IgG compared to the monomer vaccine-elicited IgG in terms of protection against challenge. The next measure we looked at were DNA copies in the blood, or viremia. As we saw with the infected control group, we saw viremia as early as day 21, and by day 49, each mouse had detectable levels of viremia in the blood. And as you expect, in the uninfected control group, there's no detectable viremia through the length of the experiment. And it's worth pointing out, this agrees quite well with the survival data, where we saw none of the infected control mice survive through the course of the experiment. As with the survival, the control IgG group mirrors quite well what we saw with the infected control group. With the monomer IgG, the 3 mice that didn't survive the challenge had high levels of viremia. However, that 4th mouse that survived, while it did survive the lethal challenge, it did have detectable viremia at day 70. And then finally, the 6-teamer group, while all the mice did survive, at day 70, we did see 3 out of those 5 mice have detectable levels of viremia. However, compared to the infected control groups, this was delayed really until the final weeks of the study. The final measure that we looked at was EBV DNA and splenic extracts. I want to point out that the 2 mice that remained EBV negative in that 6-teamer group, one of them, we did have detectable levels of EBV transcript in the spleen. However, it was just above the limit of detection. And we did see a significant reduction in the levels of EBV DNA in the spleens of the 6-teamer group, despite having that positivity. So in summary, multimeric GHCl constructs, they elicited superior neutralizing titers than monomeric GHCl in vitro. And we also showed that passive transfer of IgG elicited by the GHCl 6-teamer, 1, provided superior protection against lethal challenge than IgG elicited by the GHCl monomer, and 2, delayed viremia in those humanized mice. And thinking towards the future, we really believe that immunization with multimeric GHCl is a viable strategy for eliciting neutralizing antibodies against EBV infection and may be important for designing an effective EBV vaccine. Finally, just want to thank everybody in my lab, as well as our various collaborators across these various Seattle institutions. And thank you all for listening.
Thank you. And it's open for questions. Please.
Hi, great talk. I'm wondering with your 6-teamer, if it's large enough to add other proteins that are involved in attachment and fusion, if you think that would be worth doing to improve reducing viremia past 70 days in those mice.
Yeah, so we do think that it would be important, perhaps a complex GHL with other important antigens such as GP350. As Jeff mentioned during his lecture, other groups such as Moderna and other labs are also doing that. It's probably going to be important in, if you're targeting more antigens, it would make sense that that might be more protective. In terms of whether or not it would delay viremia, we won't know until we actually do those studies, but I think, it's an important thing to look into and it's worth doing, yeah.
Please.
Could you comment a little bit more about the scaffold for the 6-teamer? Is that a sort of an artificial protein? Do you know whether antibodies are made to that could affect things? Is it a single protein or multiple proteins for that 6-teamer?
Yeah, so for that, it's a single protein. It's I3. I believe it's either in preprint or it's been published by the King Lab. We looked into whether or not we generated through those vaccinations any antibodies against that, and we weren't able to detect it. I don't think we've published that, or it's not in the paper. But we looked at that. We also looked at MDT, and we didn't really see any titers against the I3. But it is possible that you could generate titers against those backbones. So it's something worth looking into.
I have a couple of questions myself. Oh, sorry.
Yeah, quick question about the dosing. For the comparison in animals, and I'm sorry if I missed it, but what was the dosing? Was it based on GHGL or was it based on total protein?
Yeah, so that was based on total protein. It was 5 micrograms, and that was adjuvanted with, in the original set of data, where I saw the neutralizing titers, that was with SLA-SEs from IDRI. However, when we did the humanized mouse study, we used SAS or RIBI, and we compared the titers, and they were equivalent between those two adjuvants. They're both, lipid-based adjuvants, so it's about what you would expect. But yes, so it's total protein.
When you were looking at the neutralizing antibodies produced by the various multimers, the 4, 7, 24, 60, it looked like they were all better than the monomer. So I'm just wondering why you chose to go with the 60 for your further studies.
Yeah, so I think part of it was just the limitation of, how many mice you'd have in the study. It was really the 60-mer. It was consistently quite high. There were other groups that we could have chosen, and I think if we could have used all of them, we would have. But that's the one that we elected to go with.
Okay, my questions were about if it's done there. Okay, about when you compare this monomer versus multimer, you just mentioned now that you used a couple of different adjuvants in different experiments, but they were all the same kind of type of adjuvant coming from IDRI? Okay. So question is, have you thought about playing with different adjuvants of a different nature with the monomer and maybe obtaining titers that you would compare when you take the trouble with your 60-mer? Okay, first question. Second question. When you talk about superior neutralization titers, okay, the superior neutralization titer, you imply also the quality of the antibody response, say, for instance, in the total avidity of the sera. Have you tested anything of this type?
Yeah, so maybe this might be answering, so another thing that we looked at were binding titers. So in addition to sort of having those higher neutralizing titers, we also looked at binding titers, and it's similar to what we saw in terms of the trend with the neutralizing titers, where it was higher with the multimers. And that, if the title that I have there, we've published that data in that paper, and you can see we do see those differences there. In terms of looking at other adjuvants, I do think that would be really interesting. There's a matrix M, for one, that Cohen Enable, and that sort of group is using for the GP350 vaccine. I think saponin-based adjuvants also have, a lot of they're really great at adjuvanting or immune response. And there's obviously lots of other adjuvants out there that you can look at as well. And I think it's worth doing, definitely.
Okay, thank you so much. Any other questions from the floor? Okay, we go to the next speaker. Hi, welcome. Ayman El-Guindi, targeting Epstein-Barr virus-associated malignancies using kick and kill.
Yes, hi. Well, you guys are actually the real patriots here. Hold on, it's almost done. So I'm basically the chief scientific officer at Viracta Therapeutics, and we are interested in, at Viracta, we're interested in studying and finding therapies for virus-associated malignancies. And the first virus that we are targeting is EBV and its associated malignancies. And the title of my talk today is Targeting EBV EBV-associated malignancies using kick and kill approach. And so we all know that EBV accounts for about 2% of all new cancer cases globally. That is equivalent to about 310,000 new cases every year, whether it's lymphoma, nasopharyngeal carcinoma, gastric carcinoma, and that results in about 180,000 deaths a year. My talk today will focus on, I will focus on EBV subtypes, lymphoma subtypes, and we all know that there are various subtypes and they differ in their degree of positivity. And at Viracta, the system we're using to identify whether a certain lymphoma is associated with EBV or not, we use the EBRISH assay, which is considered the gold standard assay. And you can see some examples here of the EBRISH assay. And the reason we're looking, we all know this, the reason we're looking for whether EBV is present in a certain lymphoma or not is explained in the 3 figures down here. As you can see, if EBV is present in a lymphoma, it's always associated with inferior survival outcomes, whether it's diffuse large B-cell lymphoma. You can see here's the EBV negative, here's the EBV positive. This is the PFS. Peripheral T-cell lymphoma or Hodgkin lymphoma. And so at Viracta, we are interested in developing, or we are in the process of developing a kick and kill approach. It's a new therapeutic modality. And what we're using is an HDAC inhibitor to induce a lytic cycle. I'm sure most of you are familiar with this mechanism of action. So nanatinostat activates the lytic cycle. That triggers expression of BZLF1 and BRLF1. We heard about these in several talks. Expression of BZLF1 and BRLF1, whether it's synergy or through BRLF1, as we're learning from Eric, induces expression of BGLF4. This is a viral protein kinase, and that's what Shannon had shown, that it phosphorylates ganciclovir. This is our kill. So we consider nanatinostat the kick to activate the virus, and then ganciclovir is our kill. BGLF4 phosphorylates ganciclovir. The phosphorylation of ganciclovir activates it, allows it to become incorporated into cellular DNA and into viral DNA, blocks or inhibits viral DNA synthesis, and cripples cellular DNA synthesis, causing apoptosis. As I mentioned, many of you are familiar with this approach, and I'm listing here several of these papers that have been, that basically describe this approach in the past. It's quite validated. It's been shown that it's working preclinically. And so what we wanted to do was to take this approach into the clinic and see if it works or not. Our co-founder, Douglas Fowler, had done a short preclinical study, a short clinical study, and demonstrated some efficacy, but we wanted to use a stronger HDAC inhibitor. And so in the next few studies, a few slides, I will tell you more about our HDAC inhibitor. As I mentioned, we call it nanatinostat, or for short NSTAT. And what I'm trying to do here is to differentiate it from the current approved HDAC inhibitors. And there are 4 main points that I want to highlight. The first one is that nanatinostat is not a pan-HDAC inhibitor. It's actually a specific HDAC inhibitor. It targets HDAC 1, 2, and 3. And we use it at nanomolar concentrations preclinically. And clinically, we use it at concentrations that are basically 1/4 or 25% of the, or even less, of the MTD, or the maximum tolerated dose. This is unlike all the other FDA-approved HDAC inhibitors that are used at their maximum tolerated dose. Perhaps because of the specificity and the potency, and using it at lower concentration, it's a very well-tolerated drug. And we don't see any of the heme toxicities or the hepatotoxicities, or it doesn't come with or any of the black box warnings, as in panovinostat, for example. So we see it as a well-tolerated HDAC inhibitor. More importantly is that nanatinostat is an oral drug. So unlike romidepsin, for example, or belinostat, which are infusion therapy, nanatinostat is an oral drug. So the patient basically takes 2 pills in the morning, nanatinostat and valganciclovir, and that's it for them. As I mentioned, it's a potent inducer of the EBV lytic cycle. And here's an experiment. We're comparing the capacity of nanatinostat to induce a lytic cycle in this Burkitt lymphoma HH514-16 cells. We're comparing nanatinostat with sodium butyrate, and you can clearly see that 200 nanomolar nanatinostat activates the lytic cycle, or significantly activates the lytic cycle in these cells. We're looking here at expression of the BZLF1 protein, or the zebra protein, as those who were trained with George Miller usually calls it. Sodium butyrate, basically 3 millimolar, only activated 40.2%. I would say more or less the same, but we're looking here at 3 millimolar versus 200 nanomolar. It's about 15,000 fold concentration difference. We looked at other cell lines, B cells, MUTOR1 and LCL, and you can see again that 200 nanomolar nanatinostat activates the lytic cycle more or less equal to or better than sodium 3 millimolar sodium butyrate. Nanatinostat induces the expression of both BZLF1 and BRLF1, the two immediate early proteins. It also activates expression of BGLF4, the viral protein kinase that phosphorylates ganciclovir. In a combination experiment, you can see that nanatinostat strongly inhibits or reduces the number of live cells. We're using here two different assays. One is propidium iodide using FACS, and the other one is the good old method of using trypan blue and just counting the cells, and you can clearly see an effect for the combination. Okay, we have many more preclinical data, but I just want to move quickly into the clinical data. This is a phase 1b trial, a phase 1b/2 trial for relapsed refractory EBV positive lymphoma. Basically, all histology was accepted with one or more prior therapies. The endpoints were rate response, response duration, safety, and clinical benefit. It was based on the phase 1b trials that we had a recommended phase 2 dose that goes like this. It's nanatinostat. We use it 20 mg per day, 4 days a week, so 4 days on, 3 days off, in combination with valganciclovir, where we use it at 900 mg per day, and that goes every day throughout the week. And basically, the patient undergoes a PET scan every 2 cycles. Each cycle is about 28 days. These tables here are basically describing the patient characteristics and the types of EBV positive lymphomas. And I want to direct your attention here. We have almost 80% of the patients with 2 or more prior lines of therapy. And about 75% of these patients were refractory to the last therapy. And they all, 96%, exhausted all therapies per investigator. We have a nice variety of EBV positive subtypes. We had diffuse large B-cell lymphoma. We had TNNK non-Hodgkin lymphoma. We had immunodeficiency-associated lymphoproliferative disease, and we had Hodgkin lymphoma. The total number of patients was 55 patients. So this is a summary of the data that we obtained. We had 43 evaluable patients, and across all the subtypes, we have seen 40% overall response rate and 19% complete response. And I just want to looking at this data, I want to highlight a couple of things. First, if you just look at diffuse large B-cell lymphoma, ANKTL and PTCL and AITL, you notice that we have here 63 to 67% overall response rate. We don't have much response rate. We have about 10% with the Hodgkin lymphoma. We had 4 responders with the diffuse large B-cell lymphoma. 2 of these had failed R-CHOP and basically had refractory diffuse large B-cell lymphoma. They came on the study and they were complete responders. And we also had 4 responders with the PTCL, AITL, and 3 of these were complete response. You might be aware that some of these, that for PTCL, romidepsin lost its approval because of the fact that it didn't show any advantage when it was combined with R-CHOP. This is the swimmer plot, and I want to drive your attention to 3 points here. The first point is that notice most of the patients who responded basically had obtained, or we observed the response within the first 2 months of starting the treatment, and you can see that clearly here. I want to direct your attention to these 2 patients here with T-cell lymphomas, and these 2 patients basically were unqualified to receive stem cell therapy. However, they came on the study, they improved, and then they became qualified for stem cell therapy and basically withdrew from the study. So it's bad for the study, but it's good for the patient, which is what's really important. Patient number 11 here actually had a complete response in the first year, and then for some reasons that are unrelated to the study, he stopped, and we tracked him down and were able to do PET scans, and we are more than 34 months, because this is basically in October 2021, still doing well. We want to think that he was cured, but we cannot say that. Our median duration is about 10.4 months, and this is the safety profile. I'm not an MD, but Ivor is here. He is an oncologist by training, and he's the CEO of Viracta. So if you have any questions about the adverse events, you can definitely ask him. But just to summarize, this is a table that shows the grade 3 and 4 treatment for patients, for 3 or more patients, or about 5%. And I believe that all what we're seeing is cytopenia or uncomplicated thrombocytopenia. And there are some other mild effects, adverse events. This is the last slide. I want to end with this slide. This is a case of 37-year-old female. She has extranodal NK T-cell lymphoma. She was refractory for 2 lines of treatments, for SMILE plus radiotherapy, and for ASCT. She was basically considered at this point for palliative care. She was accepted in the study. She had partial remission within the first 2 months and of starting our drug, and 28 months and counting. She's still doing well. She's saying hi. So the other patient is a 50-year-old male with peripheral T-cell lymphoma. He had the first line was CHOP, romidepsin with stem cell transplant as a second line. Third line is romidepsin, but progressed after 5 months, came on the study as a partial response and stayed with partial response for 10.6 months. That doesn't include the 2 months at the beginning before they show the response. The reason I'm mentioning this case here is that this patient had romidepsin, which is an HDAC inhibitor, so we're not seeing any resistance as a result of treating with an HDAC inhibitor. This is my conclusion slide. Nanatinostat is a potent HDAC inhibitor specific for HDACs 1, 2, and 3. It activates the EBV lytic cycle, including BGLF4, the viral protein kinase that Shannon had shown at phospholase ganciclovir. We believe it's a novel EBV targeting therapy with clear anti-tumor rationale in EBV positive lymphomas. The phase 1b demonstrates that the combination of nanatinostat and valganciclovir is well tolerated. The median duration of response is 10.4 months. We believe it's safe and oral, and we are already enrolling a multi-center, global multi-center phase 2 study we're calling it NAVAL-1, again for relapsed and refractory EBV positive lymphoma. I want to thank first and foremost the patients and the families, as well as the investigators at the different sites who supported or carried out the trial. like to thank also the collaborators, Sumita and Michael and Ibikun, and the Viracta team, particularly Dr. Ivor Royston, who actually joined me all the way from San Diego to here. And I would like to take any questions.
Thank you so much. Questions? First, okay. Excuse me, Professor Cohen. Please.
Oh, okay. So you had an efficiency of EBV activation about 50% in the cell culture.
Yes.
So what would you estimate is your required efficiency in the patient?
Well, remember the 50% is a one dose in these cells.
Yes.
So it's not, in the patients we're dosing, as I showed you, we're dosing daily for 4 days, and then we give them a break for 3 days, and then 4 days again.
Okay.
So the 50% is just one dose. And we've seen responses within 2 months of starting the medication.
Okay, you answered my second question. So you go through several kick and kills.
Exactly.
Okay,
exactly.
Thank you.
Sometimes we learn a lot from patients who don't respond, and I'm wondering whether the individuals that didn't respond, was their pharmacokinetics different in terms of drug levels? Did the drug not, activate EBV? Anything known about the individuals who don't respond?
So we are actually in the process of, we have a biomarkers plan, and we're in the process of assessing. It's a very interesting question. We're definitely interested in finding out, multiple biomarkers, viral load, expression of BGLF4, as well as some of the other mutations that are associated with these lymphomas. Yeah.
I don't see anybody for a question, and I would like to thank you.
Thank you.
We can proceed to the next speaker, Maher Gandhi, the tumor microenvironment and genetic profile of ABV plus primary CNS lymphomas in amenable to combination third-party EBV specificity, ibrutinib, and rituximab.
Thank you. And I'd like to start the talk by thanking the organizers for such an excellent meeting. So the focus of the talk today is immune evasion, and it's in EBV positive primary CNS lymphoma, which is arguably the most devastating of all EBV diseases. Immune evasion in lymphoma can either be mediated by the malignant B cell or the tumor microenvironment. Loss of antigenicity by the malignant B cell can occur due to loss of the MHC peptide complex, and a reduction in immunogenicity can occur through a number of mechanisms, including PD-L1 and 2 gene amplification, for example, as mediated by EBV. In the tumor microenvironment, there are an upregulation of immune checkpoints, an infiltration of regulatory T cells and tumor-associated macrophages, all of which to serve to make EBV-specific effector T cells less potent. So the overarching hypothesis of today's talk is that there are distinct mechanisms of immune evasion that are operative in lymphomas that look histologically identical, but that are etiologically different, and that we can use this information for the rational design of combination therapies. And the lymphoma in question is primary CNS lymphoma that has diffuse large B-cell histology. The non-viral cases are, for the purposes of this talk, EBV negative, and they occur in the HIV negative setting. And the tissue positive ones are ones that are AIDS related and those that are HIV negative. So talking about the non-viral cases first, they are 1% of all non-Hodgkin's lymphoma, and they occur in the brain, the eye, and the CSF, and they're about 90% of all primary CNS cases. The median age is 65. The treatment is still fairly barbaric. You have to give very high doses of methotrexate, which is done as an inpatient, and this is given so that it can cross the blood-brain barrier, and it is consolidated with a transplant or radiotherapy. Even so, relapse is 50% within a year, and the median survival is only 5 years. Up until now, we haven't known very much because the biopsy material is very challenging, but we now know that there are recurrent mutations, particularly in the B-cell receptor and NF-kappa beta signaling pathway, and also in cell cycling mechanisms. The cell of origin is activated B-cell type, And there are deletions, particularly in HLA class 2, which is interesting because we call the brain an immunoprivileged site. Moving on to EBV tissue-positive cases, these primarily occur as a result of acquired immunosuppression, typically iatrogenic, for example, in PTLD, but also AIDS-related lymphoma. There are no accurate figures because it is so rare, but we believe it is less than 10% of all primary CNS lymphomas, or about 0.1% of all lymphomas. It is very clearly an unmet clinical need, and yet these patients are excluded from clinical trials, in part because they do badly, because they can't tolerate conventional therapy, and the optimal management is therefore unknown, and the median survival is only 10 months. And the molecular characteristics of these cases is completely unknown. So in an enormous collaboration around the world, we managed to obtain 92 samples. About half of them were non-viral, and then a quarter were AIDS-related, and the other quarter were EBV tissue-positive but HIV negative. We performed sequencing, nanostring on the transcriptome, and copy number alterations. And the results were very striking indeed. There's clearly a distinct mutational landscape between EBV tissue-positive cases and the non-viral. So here we have the non-viral, and if we focus firstly on NF-kappa beta, epigenetic regulation, cell cycle adhesion, and B-cell differentiation, you see that compared to the EBV tissue-negative cases, there's a clear enrichment, as you would expect in the non-viral cases, primarily missense mutations, but there are very few of these mutations in the EBV tissue-positive cases. We also, in our targeted sequencing panel, looked at antigen presentation and immune function, and there were, generally speaking, very few mutations, and most of those that did occur were in the non-viral setting. We then focused on the hallmark mutations, which are MYD88, CD79B, and PIM1, and they occurred in 80% of all cases of non-viral PCNSL. In fact, more than 2/3 of cases had 2 or 3 of these mutations, whereas there wasn't a single mutation in any of the EBV tissue-positive cases. And the overall number of mutations was clearly much higher in the non-viral cases. But if you look more closely at the EBV tissue-positive cases, you see that there are more mutations in the HIV-positive setting. Arguably, this may be expected because of work led by Riccardo Dolcetti regarding the HIV matrix protein P17, which appears to be oncogenic. You probably, in this audience, would expect that the general absence of mutations is consistent with EBV's known oncogenic properties, but of course, in order to prove this more conclusively, we need to do a more extensive sequencing panel to definitively rule out if there are other mutations present or not. We then looked by nanostring at the cell of origin. This is a measure of the B cell stage of differentiation in which transformation occurs, which is typically the activated B cell in non-viral cases, as we found here, and yet it very rarely was activated B cell in the EBV tissue-positive cases, clearly showing a difference in biology, and is also in line with some of our recently published findings in other settings. Moving on to copy number loss, there is clearly a very high amount of copy number loss of HLA class 2 and to a lesser extent HLA class 1. We saw very little of this in the EBV tissue-positive setting indeed, which is intriguing. It suggests that these proteins can potentially be targeted by EBV-specific T cells or other forms of immunotherapy. But it also means there must be some other mechanism of immune evasion. So by nanostring, we found that there was an upregulation in immunosuppressive molecules such as CD163 and PD-L1 and LAG3 and TIM3. HIV, of course, had low levels of CD4, as you would expect. So potentially, we could give combination therapies that can cross the blood-brain barrier, such as EBV-specific, viral-specific T cells, and also small molecules that can also cross the blood-brain barrier, such as ibrutinib, which is something that is known to polarize T cells towards the Th1 phenotype and has also been shown to kill EBV-transformed B cells in vitro. Interestingly, this drug is commonly given in chronic lymphocytic leukemia, where we know that there are a number of inactivating mutations, particularly CARD11, but as we've just shown, these mutations are absent in EBV-positive PCNSL. Our group was the first to show that third-party EBV-specific T cells can cross the blood-brain barrier. This is the very first patient, and she's still alive today, and this data is from 2007. EBV-specific T cells, in this case alone, was sufficient to induce long-lasting remission in a patient who was refractory to the first 2 lines of therapy. There is a facility in Sydney that makes third-party EBV VST using the Miltenyi closed system and the Miltenyi proprietary peptides. And this approach is fairly safe and has low toxicity. It can also be combined with monoclonal antibodies such as rituximab, but I just want to stress it can't be combined with PD-1 blockade because PD-1 blockade is contraindicated in PTLD because of the risk of organ rejection. So we treated 2 patients. These patients are treated off study on the government-approved compassionate access that is available in Australia. The first patient, he was 42, he was a grandparent, and he was actually deemed unfit for high-dose methotrexate therapy. This was because he was on hemodialysis, and he had had prior disseminated Cryptococcus and Pseudomonas bronchiectasis. The other patient, a 30-year-old female, had had previous methotrexate, and she was refractory. Both patients had no mutations in HLA class 1 and class 2. They both received ibrutinib, and at week 10, they received third-party EBV VST, and they also received a number of doses of rituximab. Both patients went into remission fairly quickly. They tolerated the therapy reasonably well. I say reasonably well. They did have some side effects. Remember, the first patient was very prone to opportunistic infections, and he had a number of inpatient admissions, but they got through it. And this slide is out of date. They are now in sustained remission for over 3 years of therapy. I mentioned that the first patient was on hemodialysis, which of course is a problem because it removes the ibrutinib. We performed PK analysis on dialysis days and on dialysis off days, and we performed this simultaneously in the CSF and the plasma. And we found that although the ibrutinib was being removed to a fair extent on dialysis days, even on dialysis days, there were still detectable levels, at reasonable levels, in the CSF of ibrutinib. The first part of the data was published last year in March, and the second part, the 2 patients, was published last year in October. And on the strength of these findings, we went on to perform a clinical trial. This is a national trial, and it began prior to COVID, shortly prior to COVID, and the cooperative group, the ALLG, that ran it prioritized this study when COVID happened. So it was full steam ahead during COVID. There was a great deal of cross-referrals amongst the 10 referring sites across Australia, so much so that we actually finished our accruement 1 year ahead of schedule. Patients receive ibrutinib for 18 months. They receive the VST at 10 weeks, and they receive a number of shots of rituximab at various time points. And the last patient will receive their last dose of ibrutinib in December this year. Data cutoff will be in 23, and we hope to write off and complete then. A second study, which will be in non-immunosuppressed patients, which is a different combination, will start in December 22, and that gives autologous EBV VST with an anti-PD-1 blockade antibody, and that is for patients with systemic disease of EBV-positive DLBCL. I'd just like to
2 minutes.
thank everyone. I've got 2 minutes to thank everyone, but in particular, Tan Hong, Com Keen, and Emily Bly. And thank you very much for listening.
Thank you. Questions?
So I like the approach. I think you've got great results, but quibble with the horrible outlook for these patients. So we did a study in the early 90s where, in fact, the average survival was 2 months, and it was very common before there was effective antiretroviral therapy. In the era of effective antiviral therapy with the combination of methotrexate, which can only be used in people with good renal function, and with aggressive use of rituximab, patients who respond to the antiretroviral therapy do very well. And my clinic has about 4 patients, all of whom are several years in remission, none of whom have relapsed, in contrast to my non-HIV patients. And I'm telling you my anecdotal unpublished results, but the group at UCSF has put together a retrospective look with some similar data. I believe there's a group at UNC who's putting together similar data, and an informal poll of the AIDS Malignancy Consortium, and I lead the lymphoma committee of the AIDS Lymphoma Consortium, says that many investigators around the US are having similar results, all with their own somewhat different formulas, but it's not the grim disease that it used to be. There are failures, and I think you have a great approach for people who can't tolerate methotrexate.
If I could make a comment to that. So the 10 months that I quoted were from the largest World Series. I led the Australian component, but that was of PTLD. So we are talking about 2 different diseases. So I fully agree, and I'm aware of your work, and I'm a great fan. But in the PTLD setting of an international study, I think of 93 patients, which has been updated. The updated study has not been published, but the original study was published about 5 years ago. It was 10 months.
Yeah. So that all makes sense to me, and it's testimony to how wonderful antiretroviral therapy is, that in HIV, the world has changed.
Hi, excellent talk. So with regard to the mutational landscape observed between the viral and the non-viral lymphomas, I can't help but wonder might that have something to do with the clonality of the EBV lymphomas compared to the non-EBV lymphomas?
Interesting question, but difficult to obtain data because of the quality of the samples. I presume you're saying that you're more likely to have poly or oligoclonality in the cases, and I've actually never thought about that. It's a very, very interesting question. It could do, but if you're suggesting that that would make us less likely to pick up mutations because they may be at a lower level and more dispersed, I think given the level of coverage that we had, I think that would be unlikely. I think what's more likely is that our targeted sequencing panel missed some notable mutations which have been presented through this meeting, which may well be present. STAT3 is an obvious example. We never had STAT3 on the panel.
Okay, if there are no other questions, we go to the last speaker. Vijayendra Dasari, a lymph node targeted molecular adjuvant and engineered subunit antigen vaccine, promotes potent cellular and humoral immunity to Epstein-Barr virus in HLA-expressing mice. Please.
I would like to thank organizing committee for giving me this opportunity. I'm going to show you our vaccine approach using lymph node targeted adjuvant and engineered subunit vaccine. Basically, this is a collaborative project between QIMR, Elisio, and Atara Bio. Just a little bit of background. After infection, EBV establishes 2 different life cycles, lytic and latent phases of infection with unique viral gene expression. In some people, lytic infection can cause infectious mononucleosis, which is a high risk factor for MS or chronic active EBV. Depending on the immunocompetency of host, during latent infection, EBV can cause various types of B-cell cancers and epithelial cancers. Therefore, in order to reduce the EBV-associated disease burden, various vaccine formulations were developed and tested in human clinical trials. These vaccine formulations were developed to induce humoral immunity by targeting GB350 antigen or cellular immunity by targeting EBNA and LMP proteins. First two vaccines were tested in university students because this is considered as a highest risk group for infectious mononucleosis. Most importantly, both vaccines were able to reduce incidence of IM, but unfortunately they didn't prevent asymptomatic infection. Therefore, subsequently, in order to improve the efficacy of vaccine, there are two new generation vaccines in developmental stage. Both these vaccines target multiple glycoprotein complexes in order to improve breadth of humoral immune responses, as we heard from Professor Jeff Cohen. While these vaccines are progressing into human clinical trials, we wanted to follow a different approach. EBV-specific antibodies play a critical role in primary infection. Usually, these antibodies are raised against multiple glycoprotein complexes induced in lytic phase. After lytic phase, EBV entered into establishes a latency. This latency phase is very well controlled with cellular immune responses induced against LMP proteins or EBNA proteins. Indeed, in various adaptive immunotherapy clinical trials showed that reconstitution of T-cell immunity, especially in transplant patients and MS patients, can prevent the outgrowth of EBV-infected B cells. Therefore, we hypothesized that if any vaccine is designed to induce both humoral and cellular immune responses against multiple antigens expressed in both lytic and latent phases of infection, likely to provide better protection. However, targeting multiple antigens into a single vaccine formulation may not be a feasible approach. And also some of these, especially these latent antigens, are associated with oncogenic potential and also known to interfere with antigen processing and presentation. In order to overcome such issues, we have developed a novel recombinant protein called EBV poly, which consists of 20 CD8 T-cell epitopes derived from 8 different lytic and latent antigens. They target all major HLA alleles worldwide. Each epitope is linked with a proteasomal liberation amino acid sequence in order to improve the processing and presentation of these epitopes. After purifying this protein, we tested immunogenicity of EBV poly in vitro by stimulating EBV seropositive donor PBMC with EBV poly. After stimulation, we could see rapid expansion of EBV specific CD8 T cells restricted to multiple epitopes from all the donors. After confirming the immunogenicity, we developed a vaccine formulation with 3 different components. First component is the EBV poly, which is a target for CD8 T cell responses. Second component is the GB350 protein, target for neutralizing antibodies, CD4s, and CD8 T cell responses. Third component is MCPG, which is basically an adjuvant, consists of CPG molecule linked with albumin binding lipid, which is just to target to link draining lymph nodes. After immunization, at the site of injection, this MCPG binds to albumin. Then the albumin delivers this antigen adjuvant to lymph nodes. In the lymph nodes, this adjuvant activates antigen-presenting cells and B cells in order to induce humoral and cellular immunity against these 2 proteins. Initially, we looked at the ability of MCPG to enhance the delivery of vaccine components to both inguinal and axillary lymph nodes. As you can see here, mice immunized with MCPG vaccine enhances delivery of EBV polyprotein into both the lymph nodes compared to mice immunized with EBV vaccine formulated with CpG. Similarly, AMCpG also induces strong innate immune responses compared to CpG in both inguinal and axillary lymph nodes. Then we tested immunogenicity of EBV vaccine by formulating these EBV poly and GP350 proteins with either AMCpG or CpG. It's labeled as a soluble CpG here. Then we tested immunogenicity in 4 different HLA transgenic mice by giving 3 doses of vaccine. As you can see here, mice immunized with AMCpG vaccine induced a higher magnitude of EBV polyspecific CD8 T cells and GP350 specific CD4 T cells compared to EBV vaccine formulated with CpG. Interestingly, these cells were able to secrete multiple cytokines such as interferon gamma, TNF, and IL-2. Then we tested the ability of AMCpG vaccine to induce durable immune responses. As you can see here, mice immunized with AMCpG vaccine induced a higher magnitude of EBV polyspecific CD8 T cells and GB350 specific CD4 T cells either after 3 doses or 2 doses of vaccine. Then these responses were maintained at a detectable level for beyond 29 weeks. In addition to cellular immune responses, AMCpG vaccine also induced GP350 specific antibody secreting cells, then GP350 specific antibody titers, and also these antibodies were able to neutralize EBV virus. However, there is a decline in the neutralizing antibody titers by week 29. But when we boosted these mice with a third booster dose, we could see again a boost in the neutralizing antibody titers to its normal level observed in week 7, indicating that memory responses can be recalled with the subsequent booster dose. So these are all my conclusion. Epitopes included in EBV poly can be processed and presented by human PBMC. AMCpG enhances targeting of EBV poly to draining lymph node and also activates strong innate immune responses. AMCpG induces robust polyfunctional EBV-specific CD4 and CD8 CD8 and CD4 T cell responses and antibody responses in HLA transgenic mice. Then these responses were maintained for long term. So these are my acknowledgements. I would like to thank my supervisor, Professor Rajiv Khanna, for his tremendous support, and other colleagues. We would like to thank Eliseo for providing AMCpG adjuvant and their experimental contribution. We would like to acknowledge funding from Atara Bio. Thank you for listening.
Thank you. It's open for questions. If there are no questions, I'd like to ask a question about your adjuvant. Beside the data that you showed, is there this has been used for other experimental vaccinations, trials?
Yeah, Eliseo is testing this adjuvant in human clinical trials as well.
Yeah, and
this is basically a CpG 7909. It is already being tested in various human clinical trials from GSK and other companies.
It's interesting because CpG by itself, it doesn't work as an adjuvant in humans.
It will work a little bit, because it's a small molecule. It can get lost in the blood circulation. The amount of CpG reaches to the lymph node is much lesser if you don't
add the lipid moiety.
Lipid moiety.
Okay. Okay, if there are no other questions, thank you very much. And thank you very much, everybody, for sticking up with us.
Okay, perfect. So, as we say in this country, the train has slowly arrived to its final destination. And Lorenzo has asked me to sum it up a little. And I thought that the scientific sessions were full of many, many exciting talks. And there were some pretty lively and sometimes even animated discussions and questions. So that part was pretty good. As for the social events, all the profane videos that I received from that sacred place yesterday, where people were dancing away their worries to the tune of Funky Town and You Spin Me Right Round, we can safely consider that part also a big success. So we have learned so many things over the last 3 days, and I thank you all for your participation and coming from near and far afield. And there are so many, we learned so many things, but there are so many other things, so many issues that have remained unresolved and will require our impetus to go back to our labs and work on them. And just to remind you, this statement from my PhD mentor, Professor George Klein. So with this sobering statement, I hand over the mic to Lorenzo for his comments.
Thank you for these comments, but I just, since it's late and all the people, I think we are tired, I just want to thank all of you for coming in presence in this meeting and to contribute to this meeting. And I really hope that you enjoy the scientific part of this meeting. And this also some collaboration came out from different people in different labs. Then I want to ask all the people who helped me in the organization of these events. That was after 2 years of pandemic, so that was a little bit difficult to start. And a particular thank to the staff of the University of Siena, of the Department of Medical Biotechnology, and Roberta Corsi and Sandra Maggi. And a particular thanks to the scientific organized committee, local organized committee. And in this, I want to thanks personally, and because they give a great contribution and great help that otherwise I could not have done the organization of the meeting. My two PhD students, Salvatore and Maria Chiara. And also a particular thanks to the students that have helped in organize, and to also my residents that have contributed also to the organization of the meeting. And here I see Domenico and yes, so thank you.
And have you finished?
Yes,
Okay,
I finished.
sorry. And I'd just like us to put our hands together and really thank Lorenzo and Pankaj and all the staff and students that Lorenzo's mentioned for an excellent meeting. Thank you so much for having us.
So we will meet again in Boston, and then I really hope in Nairobi in 2026.